I thought. Slides. Do we want to be sharing a second? And we just do a quick mic check. Yeah, so the mic should be working. You don't hear yourself very much, but this captures essentially the speaker, and it broadcasts. Testing? Yeah, it broadcasts it through those speakers. Okay. So I'll just double check. Yeah, so the link is showing. I'll just. Skip this one from the some place where so let's start at 9:22.
9:22. Okay. Okay, I'll just draw.
We'll be getting started at 9:22. Just 9:22.
And then. Or you can just go grab coffee and come back. Is it okay if I hasn't started yet? We're starting at 9:22. Sorry. Sorry. The tape is kind of acting weird. There we go.
Everybody needs to access the Hayden or lecture has to come through there.
Yeah.
Okay. Yeah, exactly. Would you want to close the doors at some point or leave them open? Leave them open. Because there is some noise. Yeah, I guess there's also some noise from the construction. Yeah, maybe close it once we start the speakers. So until 9:30. Yeah. Also, one thing that speakers typically ask me whether they can use their pointer. So once you start, say, did you want to close this?
No, keep that open. Yes, that's a different presentation. You don't say that one.
Yes, when you start presenting,
yeah.
So we'll set up the presentation when it's working, you have the presenter here, and this is what we broadcast on the big screen. And then if you want your monitor to be visible, right click. Yeah, it's simple. Yeah, that'll work. Now I think Kenneth has to show the open it up for you.
And so, so I can load it from this room or download it except for the next keynote, which we don't have yet. But yeah, so I think just after each talk ends, I'll come in and then I'll exactly you come in. And then as soon as I press from beginning, then it'll play here.
Okay. The second screen. If for any reason the speaker doesn't want to present your view, you can uncheck that.
Okay.
But I think most of them will want.
Yeah. There we go. Nice. Yes.
I'm saying this part, and then you'll say some general things that I myself, and you're saying this.
Yeah. Do we have
Plus canteen.
plus canteen? Yes.
Like we just need to mention.
Yeah.
Throughout the day, the food will be available all day.
Yeah. Okay. Yeah. Okay, we got that here, and then
Cannon is the overflow.
yes, exactly.
We have that.
Florence Buchanan here and got canteen here.
Yeah. We just have to mention Florence Buchanan and canteen. We will help people get us started there.
Yeah, exactly. Yes. Well, I think in terms of like getting this word, so we'll tell them to go up.
Up to the stairs.
Yeah. I don't know if we should say the rules word. Right, let's give it another minute.
There's going to be a lot. Did the speakers get breakfast and stuff?
I don't know if they maybe they'll go.
The secretary wants to speak to everyone about access to the growth. He's the only one. That's what she was saying.
Do we have signs to the grow or no? It's this one. I'll just tell people to type the grow card into Google Maps. It should come up. Oh, okay.
So that's why we'll guide them. Don't worry, we have enough people, and Sean's helping.
Okay.
All of us will help. Yeah.
What exactly is the plan for that? Because it'll just take
What time
a lot of people will be scrambled.
What time is the first talk at the
1 p.m.
1 p.m.
Yeah.
So if lunch is happening before that, around 12:15, we'll start moving people towards
From the
Grill
canteen
Park.
versus the okay.
Yeah,
Okay.
because we want to start the.
Yeah.
Yeah, but I guess we don't have to attend.
So around 12:30, we'll head and set it up, and then around.
Let's just tell people to meet us outside.
These lights when it starts. I'll be here anyway, but control them using this panel. It's panel you can do that.
Let's get started. Hello, everybody. Thank you so much for joining us for the Symposium on Psychedelic Science, co-organized by the Cortex Club and the Oxford Psychedelic Society. I'm Kenneth.
I'm Andrea, and so I'm the current president of the Oxford Cortex Club, and we're a student society committed to sharing neuroscience knowledge and cutting-edge research with the broad academic community of Oxford. And so we organize regular seminars with world-class researchers from all fields of neuroscience, which are available for all to attend. And we also organize social events throughout the year.
So I'm Kenneth. I'm the president of the Oxford Psychedelic Society. We're a group of researchers, students, artists, everybody really, who's committed to fostering dialogue about psychedelics
both within the sciences and also in art and culture. So a few logistical points for today. We're going to be using two different lecture theaters today. The first is this one, the Blakemore Lecture Theater, and the second one is the LeGros-Clark Lecture Theater, which is in another building. At 12:50, we'll start escorting people from the outside of this building to the LeGros-Clark building. That's during the lunch break. And then presentations will start at LeGros-Clark at 1 p.m. If you have an overflow ticket and if you can't find any space for seating in LeGros-Clark Theater, then please watch the live stream of the symposium, which will be taking place in the Florence Buchanan Lecture Theater. That's on the second floor of this building. And secondly, the symposiums will consist of 4 keynote presentations and 10 lightning talks. The keynotes are 1 hour long and the lightning talks are 15 minutes long. We'll be adhering quite strictly to the schedule today. So if you're a speaker, please make sure your presentation doesn't run over time. I'll give you guys a 10-minute and a 5-minute warning. If you have extra time, you can feel free to take questions from the audience, but otherwise, please save questions for the break periods. We've got break periods from 12 to 1 p.m. and from 3:30 to 4:30 p.m. All that info is on the program, which you can get up here. The third point is that we'll be serving food all day in the canteen, which is upstairs on the first floor of this building. To access the canteen, just go through the back doors of the lecture theater over there. Same for the Florence Buchanan Lecture Theater. All other lecture theaters all other rooms you need to access in this building, just access through those doors at the end. And yes, we'll be serving food all day there and also lunch there from noon to 1 p.m. And the last point is that we meant to sell merchandise for the symposium, but it sadly did not arrive in time. So we're selling the symposium t-shirts on the merch website for the Oxford Psychedelic Society, which you can access through that QR code. Great. Without further ado, we'll get started a little bit early with Amir Inamdar, who's going to be talking about progressing psychedelics to therapeutics. Amir is the Chief Medical Officer at Cybin, which is a for-profit psychedelics company that is also sponsoring this conference. Amir, go ahead.
All right, good morning, everyone. I think I will have to shout. I will do my best to shout. If you can't hear me at the back, just raise your... Okay. So as Ken said, my name is Amir Inamdar. As way of introduction, I am a psychiatrist and a physician by training. I've been working in the pharmaceutical industry for about 20 years now, doing drug discovery and development. That's basically taking novel medicines from candidates through to preclinical development and into clinical trials. In case you're wondering what a chief medical officer does in a pharmaceutical company, my job in Cybin is to do exactly what I've been doing, which is taking medicines, novel medicines, candidates into clinical development and progressing them into drugs that can be registered and prescribed. Cybin is a small company. It's a small company focusing entirely on psychedelics. And the mission is to bring novel psychedelic medications into the clinic so that it can benefit people who need them most. Here are my disclosures. In addition to Cybin, I do hold shares in other pharmaceutical companies. Today, what I'll try to do is divide my talk in 4 different sections. I'll first talk about what therapeutics are, what medicines are, and why we need to develop psychedelics as therapeutics. I'll take you through the process of drug development. What does it actually take to bring a novel molecule into the clinic from when it is first identified? How do you identify them and take them through the process of development? What would it mean for a psychedelic? Does this process of drug development actually change for a psychedelic? I'll spend a bit more time on that, and then we'll try and touch on some of the methodological issues with psychedelics and see what next steps are. I'll try to leave some time for questions. Hopefully we'll have some time for questions. If not, try and catch me during the breaks. So as you know, psychedelic drugs are illegal. They are on Schedule 1 of the drug, according to the drugs regulation. This Schedule 1 basically means these drugs are considered to be of high risk of abuse or misuse or a high risk of harm. They are considered not to have any medical value, which basically means even if we think that these drugs may be useful for people with the mental health conditions, a healthcare practitioner, doctor cannot really prescribe them to patients. If you want to do research, you need a license from the Home Office for a controlled substance. And that is not an easy process. It takes a long time. Just the processing of the application takes about 16 weeks. It involves a lot of paperwork, is expensive, and generally cumbersome. If you then want to do research, you will need, as you know, approval from ethics committees in institutions. And often people or researchers end up in situations where the ethics committee will not accept your application unless you have a Home Office approval or a Schedule 1 license. And the Home Office will not accept your application if the ethics committee has not approved the study. So it's a typical catch-22 situation there. Now, if you want to do clinical trials internationally, and you want to import substances or import medications, drugs across borders, it's an even more tedious process. You need import-export licenses, which are expensive, which involve, again, a lot of legal paperwork, and these are time-limited. So it's often an absolute waste of time, which is very nicely captured in the statement there from one of the researchers who said that even after everything was in place, it probably would take them up to 2 years to start a clinical trial. So I'm not going to talk about descheduling or rescheduling of psychedelic drugs. I think that's a topic slightly different for a different time. But what I'd like to speak about is how, by developing psychedelics as therapeutics, as medications that are approved, could be one way of moving from that problem and making these drugs available to people who need them more. So what's a therapeutic? Simply put, a therapeutic is anything that benefits, is a therapy, is a medicine. And these medicines or drugs, before they can be actually marketed or prescribed as therapeutics, they need to go through a rigorous process of testing, and they go through a process which ends up in what is called in the US a new drug application, or the NDA. In Europe and in UK, it's called as a marketing authorization application. And that application is based on a number of studies. And the intention of the study is simple. Primarily, these studies seek to establish whether the drug that you are wanting approval for, whether it is safe. Safety in clinical trials is the fundamental objective of any research program. The second thing that they focus on is, are the claims about efficacy that are being made for this drug, are they valid? Are they based on data? Are they supported by the research that you've done? And do the benefits outweigh the risk? Now, there is always an element of risk with any medication, with any drug. Even paracetamol has a risk. Aspirin has a risk of bleeding. But what we do in drug discovery and what is important for medications is that the benefit from that drug should always outweigh the risk. And that is a fundamental thing that regulators will look at when they assess your application for a new drug. Part of the process also involves looking at the claims you've made on a label. You say this drug is effective in treating schizophrenia. Have you submitted enough data to justify those claims? Then the final important step is looking at the process of manufacturing and control that you have employed. Does that ensure quality, purity, and potency of the drug as you claim it has? And once the drug has gone through this process of preclinical and clinical development and is approved as a new drug, only then it can be prescribed to people who need it most. So what does this process of drug development and discovery entail? Now, fundamental to this process of drug development is the concept of lock and key that was proposed by Emil Fischer in 1890. And this is where everything starts from. You probably already understand this concept, which considers receptors as binding sites for drugs in the lock and key model. And then you've got to find something that fits exactly in terms of shape and size in those receptors. And once this combination occurs, it starts a downstream process which can either amplify what happens downstream, it can either inhibit that process, or it can deactivate. And that's where all drug discovery starts from identifying a site where the drug can act. Now, to start that process, you identify a target, which is the site of action of the drug. And this then proceeds through a number of different steps, identifying and validating the target to finding a hit or a lead chemical or a chemical structure that you can further develop into a medicine, fine-tuning a series of chemicals to get desirable properties in those chemicals. Once you fine-tune this and optimize this lead, then starts the process of establishing safety first in non-clinical setting, in animals. You establish safety and pharmacology. Only then can you move into clinical studies in humans. So starting with the first step, which is target ID and validation, we always and only develop drugs for an unmet need. So we don't develop medications where problems don't exist. So you have to have an identifiable disease, you have an unmet need in the disease, and for that unmet need, you identify a target. This target is usually based on a biological rationale. You can employ things like models, like genetic models, like knockout mice, transgenic mice. You may know from biology that certain genes are upregulated or downregulated in a disease process, and therefore you identify the target where you could modify their function. These targets also have to be disease specific. You can't really have a target that actually is everywhere in the body. That is too non-specific, and you cannot really develop a drug for it. Finally, one consideration is the target druggable? What that simply means is, can you actually develop a drug for that target? Can you find a molecule that fits that receptor? If you cannot, then basically you cannot develop a chemical or a biological agent to treat that condition. So once you've identified a target and validated the target, then you need some sort of a chemical starting point with some sort of hint of activity. This is the process which is called as developing a lead. Now, nature is a very good source of leads. When organisms evolve, they develop defenses. These defenses are chemical defenses often, and these chemicals go on to become good sources of drugs. There are many examples of these, like morphine, penicillin, aspirin, which are all leads that were derived from natural sources. There are other processes that you can employ to identify these leads. One technique that is very commonly employed in the industry is high throughput screening, where on a daily basis, thousands of compounds are screened in an automatic way in machines. And there's a process that involves looking at structure and activity relationships. We know that certain types of structures will have certain types of activities. And this process is used to generate lead molecules from which new drugs can be made. Nowadays, of course, we can use in silico screening, so virtual models in computers that can help us identify lead molecules to take through into development. And then there's this class of me too or copycat drugs. So if there's already an antidepressant, develop a new antidepressant which is similar in structure to the antidepressant, a painkiller which is similar in structure to the existing painkiller. By and large, though, most of the leads we identify are by chance. Almost 70% of the leads we identify, which go on to become drugs, are identified by chance. A very good example is sildenafil, or something more commonly known as Viagra. Now, this drug went through preclinical testing. It was being developed for the treatment of a heart condition to dilate the blood vessels of the heart. So it's a phosphodiesterase 5 inhibitor. It dilates blood vessels. And something very interesting happened during clinical trials. When nurses went to examine male patients, they found that these patients were lying on their stomachs. This was a mystery until one astute nurse figured out that actually the men were embarrassed because they were having erections. The drug was doing its job. It was dilating the blood vessels, but just not dilating the blood vessels in the heart. And this went on to become a blockbuster drug. A very good example of how chance or serendipity leads to development of some very successful and very effective medications. Now for psychedelics, part of the job is done in the sense that there are naturally occurring leads, whether that is psilocin or psilocybin from the psilocybin mushroom, or it is DMT from ayahuasca, or it's the toad venom. These leads are already there, so you get to skip a few steps in the development of these medications. Once you've identified a lead, you need to fine-tune it. And this is where chemists or medicinal chemists come in. And they look at what can you actually do to fine-tune or improve the properties of the drug. These are minor changes that need to be made to, for example, the shape of the carbon skeleton, the scaffolds. Can you do something to that? Can you change the lipophilicity or the water solubility of these drugs? Now, lipophilicity is a very important property of a drug that you would use to treat psychiatric conditions, simply because it needs to cross the blood-brain barrier. It needs to cross membranes in cells and get into the cells. And you can do that by changing certain chemical groups like methylene groups in these drugs. You can do other things, make the molecule more rigid or more flexible by changing the degree of unsaturation, remove a ring, make it better fit into the receptor. Loads of things that you can do, but essentially what this process is doing is improving the potency and selectivity of the drug. And by doing that, you generate a series of analogs. And ultimately, once you finish this process, what you have is a drug candidate. Once you have a drug candidate, the next step is to start testing. Now, testing occurs first in a non-clinical setting, not in patients, in animals, in vitro and in vivo models, and only then does it progress to the clinic or into humans. So the fundamental question that you are asking, or two fundamental questions you're asking in the non-clinical setting is, one, does it work? Does it have pharmacological actions? And two, whether it is safe. Does it have any toxicity that prevents it from being dosed to human subjects? But before we talk about what that process is, let's spend a minute on how this is regulated. Now, the clinical trial industry is one of the most regulated industries in the world. The only industry that is more regulated than clinical trials is the aviation industry. And it's for good reason, because we are dealing with human lives, we are dealing with people, and safety is paramount. There was a time when different countries, different regions in the world had different regulations. It was difficult to apply data generated in one part of the world to another. You had to do separate studies in different countries and separate applications. But this all changed with the International Conference on Harmonization. So since its inception in the 1990s, this group brought together the regulators and the scientific community and the pharmaceutical industry to develop guidelines and agree on the process that needs to be followed for development of drugs. These are largely grouped into 4 categories. There are guidelines on how a drug should be manufactured, what are the minimum standards for purity, stability, etc. Then there are guidelines on efficacy. You may or may not have heard of good clinical practice. So this is a set of guidelines that governs the conduct of research into humans. And this is within the efficacy section of ICH guidelines. The next group is safety guidelines. These are non-clinical testing strategies that must be employed before a drug can be taken into the clinic. And then there's a miscellaneous group at the end, which focuses on multiple different topics. So in this framework, a drug enters into non-clinical testing. And this graphic is an illustration of the amount of non-clinical testing a drug goes through before it can go into the clinic, into patients, healthy volunteers. And even once it's entered into healthy volunteers and into clinical trials, the non-clinical testing process continues. So this starts with broad groups of studies, which include testing for example, genetic toxicity. Does that drug do damage to your chromosome? Does it damage your genes? Safety pharmacology, which I'll speak a bit more on the next slide about. Toxicology, so this is testing into animals in at least 2 different species for a certain duration to make sure that we are happy that if we give the drug into human subjects for that period of time, it does not cause any toxicity. Then there's the question of, does it affect reproduction? Does it have effects on the offspring? And some of you may be aware of a morning sickness drug that was prescribed a long time ago called thalidomide, which resulted in children being born with short limbs or phocomelia, as it is called. And part of the guidelines for doing reproductive toxicity studies have evolved through incidents like that have happened during development of drugs due to insufficient testing that was taking place at that time. And in the background, there are a number of studies that look at absorption, distribution, metabolism, and excretion of the drug that go on throughout the life cycle of clinical development. As a clinician, when I look at the non-clinical studies, I typically focus on the genetic toxicity, but also on a group of studies called as safety pharmacology studies. And these studies are designed fundamentally to look at the effects of the drug on the vital systems. This is the cardiovascular system, the central nervous system, and the respiratory system, and to make sure that there are no adverse effects that can dramatically affect somebody who's taking this drug. The other set of studies that I typically look at and this becomes interesting when you talk about psychedelics, but for other small molecules, these studies are called toxicology studies, and these are usually done in 2 species. One is a rodent species, one is a non-rodent. And these involve repeated dosing of drugs to the animals for a period of time that is determined by the duration of the clinical trial that you want to do. So if you want to do a study in depression for 12 weeks, you want to test the drug in patients for 12 weeks, then you must do a study in animals for 12 weeks. So the duration of the clinical trial is what determines the duration of the non-clinical studies. And one fundamental principle that is employed more and more now in non-clinical research is the 3 Rs, where we try to reduce the use of animals. If we can do work in vitro models, we do work in vitro models. We reduce the amount of animals that are exposed to such studies. We refine our processes and we recycle what we have learned previously. Once we've done the non-clinical set of studies, it is time to enter into the clinic. And that involves a number of different studies before we are even ready to apply for approval. A typical clinical program would start with a first-in-human study. These studies are done in healthy volunteers because we do not want any other confounding factors. These are typically small studies where you give single doses of the drug in an ascending manner to healthy volunteers. Once we establish safety of single doses, then it's time to give multiple doses, because there's no drug, well, there are very few drugs which are only given once. Most of the drugs you have to take for a repeated, for a longer period in a repeated manner. And that's why you need to study what the effects of the drugs are on multiple dosing. Once this is done, we move on to small studies to establish proof of concept. Does the drug work in the indication that we intend to give it for? This process of, or this group of initial studies typically called as early development. This is where most drugs start. Most drugs do not progress beyond this phase, which is often referred to as phase 1, phase 1A, 1B. Once this is done, you're ready to move into your larger pivotal studies, as they're called. They're called pivotal because you use these studies as part of your application to the regulators for approval of a new drug. And generally, you have to show that a drug works in at least two different studies. The thinking there being, you can have a fluke result once, but probably not twice. So you need replicate studies. You need 2 studies to demonstrate that the drug works. Most of our drugs in CNS, in mental health, they need to be taken for a long or an extended period of time. And one needs to show that when they are taken for a long period of time, people don't relapse during their treatment. And these are long relapse or relapse prevention or maintenance studies that one needs to do. As you will see there, long-term safety. Safety will be everywhere in drug development. And as we keep reminding our colleagues and students every time that the first point of focus in clinical trials is to understand safety. Safety comes first. Patient safety, the safety of participants is first. Only then you talk about anything else. So at the end of this process, you generate a huge amount of data. And this data has to be submitted to the regulators as part of an application. In the past, this used to be a pretty cumbersome process. There used to be different applications in different regions, in different countries. But with ICH, everything, as I said, the International Conference on Harmonization, there is a global consensus on how these data should look like, how the applications should look like, and how these should be submitted. And the common technical document is the global standard in terms of how the data from clinical trials, from all of the research and drug development should be submitted to regulators for approval. In the old days, these used to be a bunch of files and papers that were shipped in truckloads to the FDA or the EMA. Now we have something called as the eCTD, which allows us to electronically submit these documents. Essentially, at the end of the whole process, you will have tens of thousands of pages. And that is why when you submit an application to a regulator, they take time. On an average, once you submit an application to, let's say, the FDA, they will take about 6 months to a year to review and approve or reject your application. Submitting an application does not guarantee that there will be an approval. It's a process. There's a dialogue with the regulators, and they are within their rights to reject an application and say the data is not justified and does not support the claims made by the drug. So as you've gathered by now, this is a pretty lengthy process. It is a very time-consuming process, starting right from when you identify a target to when you get the drug into the market. It takes a number of years, on an average about 10 to 12 years. When you consider how much patent life is granted once you register a patent, that's 20 years. So you identify a target, you identify a lead molecule, you patent the molecule, you get 20 years to develop the drug, market the drug, and recoup the cost of development. That's why most medicines, when they come into the market, when they're new, they are expensive because of the amount of money, amount of funds that are spent in developing those drugs. Unfortunately, also, drug development is a very inefficient process. Nowhere else is such a level of inefficiency that you will see. You start with about 5 to 10,000 molecules, and you only end up with one that is approved as a drug. And it takes upwards of 3,000 participants in the entire drug development process to bring a drug to the market when you're ready to submit it for a new drug application or a marketing authorization application. And it's inefficient because it's a very risky process, this business of drug development. As a rule, as you progress through different phases of drug development, the failure rates usually become less and less. They are highest, obviously, in the early phase, as you can see from clinical trial success rates here. In phase 1, the approval rates or the success rates are pretty low. CNS drugs are amongst the lowest, or only second to oncology drugs in terms of their success rates. But basically, it is a process that's not very efficient. You do not often get what you invest into the drugs. For somebody like me who's been working in the industry for about 20 years, I will be lucky if during my career, I am able to bring one molecule from preclinical development, from discovery into the clinic. Most people don't get to do that in their lifetimes in the industry. And in addition to being a risky process, it's a very costly process. And it can cost from anywhere between $500 million to $6 billion to develop one drug that comes into the market. For CNS drugs, for drugs in psychiatry, on an average, it's between $1 to $2 billion to bring it from discovery into the clinic. For psychedelics, it's probably going to be different. So let's move on to looking at how this process applies to psychedelics. How would you develop a drug in this framework? How would you develop a psychedelic in this drug development framework? So I think we're starting with an advantage with the psychedelics because we can kind of skip a few steps. There are naturally occurring leads in psychedelics. As I mentioned earlier, whether these are, this is psilocybin from the psilocybin mushroom, whether this is DMT or this is the toad venom, you already have pretty good leads to take into preclinical development. So the chemical structures there were not labeled on purpose to check whether you're all still awake early in the morning. If anybody can tell me what those structures are. But the one on the left is psilocybin, which is magic mushrooms, the ingredient from magic mushrooms. Unfortunately for psilocybin, it doesn't work when it is ingested. It needs to be converted in the body through an enzymatic process into something called a psilocin, which is the active drug. So what many companies are doing, many drug developers are doing, are starting from psilocin, which is the active part of magic mushrooms, and developing, for example, novel methods of delivery, making synthetic versions of psilocin, or actually taking the active portion of psilocin, which is norpsilocin. Again, we talked about medicinal chemists doing their magic and modifying molecules to make them better. The problem with psilocin is it's highly unstable. So you see mushrooms, you see the discoloration on the mushrooms, they turn slightly darker, sometimes bluish. That's because of the oxidation that happens to psilocin. And that's why it's very unstable. You can't store it for long periods of time. And that's a problem if you want to develop it as a drug. So companies are trying to stabilize psilocin. So instead of developing psilocin, it's psilocin-like or analogs of psilocin. So this is one example of how naturally occurring psychedelics could help us get rid of some of the early stages and actually fast track development of psychedelic medications. Now, we talked about a non-clinical package for non-clinical testing for drugs before they can enter into the clinic. One could ask the question, do you actually need all of that for psychedelics, especially for naturally occurring psychedelics? We know a lot about, let's take the example of psilocybin, which is the active molecule in magic mushrooms. Typically, you would do non-clinical studies before you go into man, before you administer these drugs into man. But we've already got lots of data on these drugs, whether that's in small studies done in the academia or whether it is from underground use or more recently into larger industry-sponsored studies. We know that there is clinical data already out there. So one could argue that we don't really need a full preclinical package before we develop these drugs as medicines that can be approved. But I think it depends. I think it depends on whether it's psilocin or psilocybin itself that you are developing, or it is a molecule which is like psilocin. It is all in what is defined as a new active substance. The EMA guidelines very clearly define what a new active substance is. And this is typically something that has a slightly different molecular structure. Companies are doing that, modifying the molecular structure of psilocin. Whether it's manufactured in a different way, whether it occurs from nature or it's a different manufacturing process, is it an isomer, mixture, derivative, or a salt? And it all depends on whether the substance you're developing will be classified as a novel active substance or not. If it is classified as a novel active substance, unfortunately, it will have to go through the entire process, the entire suite of studies in the preclinical space before this molecule can come into clinical testing. So all of primary pharmacology, secondary pharmacology, safety pharmacology, all of those fancy studies that we saw on a previous slide, they will have to be done. We'll have to look at genetic toxicity. And we'll have to look at toxicology. So this is on the right-hand side of that slide is a table I showed you previously about repeat dose toxicity study in animals before you go into humans. This is a challenge for psychedelics because, as you will note, these studies need to be done with daily dosing. Giving psychedelics every day, or leave alone humans, to animals is not possible. These are very powerful substances. So how do you do a study in animals with a psychedelic a repeat dose study when you cannot give the drug repeatedly. That is a challenge. And that is an ongoing conversation with the regulators as to how the guidelines can be adapted. They need to be adapted. So how do they need to be adapted for the specific requirements of psychedelics? Can we give these drugs, let's say, once in 2 weeks, once a week? Because if you want to do a study in the clinic in depression, for example, this will typically be a study which will be 8 to 12 weeks in duration. As we know from the guidelines, we require studies of at least 8 to 12 weeks in animals to be able to do these studies. But we can't do those studies with repeated dosing. So can we do studies in animals where we give this drug once a week, once in 2 weeks, or once in 4 weeks? Will that be acceptable? How does that fit into the current regulation? This is something that will have to be discussed until the regulations are changed on a case-by-case basis with the regulators in the various countries. When it comes to the clinic, we again saw that schematic of a number of different studies with first in human, multiple dose, proof of concept, the big pivotal studies. And the question then is, do you actually need a traditional drug development plan which goes through all of the stages of clinical development for a drug that is a psychedelic drug? For a class of drug that we know a lot about, there is a lot of clinical information, anecdotal plus open label studies done in the academia. We know that information. Probably not. I think it depends on the question we asked earlier, whether the new psychedelic, whether it is going to be classified as a novel active substance. And if it is, then probably you will have to do the whole host of clinical studies. What would it look like for an analog of psilocybin? You probably will have to do some sort of safety testing in healthy volunteers, maybe in patients, in a small group before moving on to slightly larger studies in hundreds of subjects. But eventually you will have to do a full phase 3 drug development program before these drugs can be approved as part of a new drug application. But doing these clinical trials is not straightforward. There are challenges. There are substantial challenges that one faces in psychiatry in general, in clinical trials in psychiatry in general. One of the fundamental problems in psychiatry is, unlike, let's say, a clinical trial in diabetes, where you can measure the blood sugar level, and the person in whom you're measuring the blood sugar does not have any influence over the blood sugar level. Or blood pressure, for example, if you're measuring blood pressure for somebody, they typically do not have control over what their blood pressure is. This is different in psychiatry because all the assessments in psychiatry are subjective. So you ask people how you are feeling, and it is a subjective report. So how you measure efficacy or improvement in studies, in psychiatry studies, is you use rating scales. For depression, you use something like the Hamilton Depression Rating Scale, where you ask them a series of questions and they give you answers, which is based on very subjective reports, which basically means it will differ from person to person. And therefore, because it is subjective, that assessment is prone to a lot of biases. Biases not only by the person who's reporting, but also by the person, biases on part of the person who's actually listening to the patients and recording their responses to a rating scale, for example, in depression. And this becomes a big problem with psychedelics because one way you address the bias in psychiatry trials, in mental health trials, is by blinding. So gold standard design in clinical trials is something called as a randomized double-blind placebo-controlled study. What that means is you randomly assign people to receive either the placebo, which is an inactive drug, or the active drug. The placebo controls some of the expectancy bias. There can be other controls as well. Neither the patient nor the investigator knows who is getting what. And therefore, you minimize the biases on their part. How do you do this with psychedelics? Because by definition, the psychedelic will induce a non-ordinary state of consciousness. And it's difficult, if not impossible, to mask that state. Can you use a placebo control? Probably not. People have used placebo controls, but one wonders what the efficacy of such a placebo would be in controlling for biases. There are other strategies though, like using active controls. Niacin, for example, niacin is typically given in high doses for treating increased lipids in the blood. And when you give it in high dose, it induces a characteristic flushing kind of response, which can sometimes put people off and make them guess what they are actually receiving. It's not very effective. It's a nasty, nasty drug, actually. And more than a placebo, it is more of a nocebo, which has negative effects on people. The other drug that's used is dextromethorphan, so Benadryl, cough syrup. In high enough doses, it can mimic some of the effects of psychedelics. It's not perfect, but it's getting there. One can use some sort of stimulants, but another strategy that has been recently employed, and in a very large study that was completed by and in the industry recently in depression with a psilocybin analog, used a non-effective dose of psilocybin as a control. Now, again, there are critics of that, and you could argue, well, what really is a non-effective dose? What about microdosing? You think microdoses also probably have effects. So giving a very low dose, is that really a non-effective dose? We don't know. So that's a question that remains to be answered, and I think it will take a while before we can get to the bottom of this. Another way is to play with the expectancy bias. When we randomize people, we consent people, we tell them that you have an X% chance of getting the drug or placebo. And one way to play with their expectations is to tell some people, oh, you've got a 75% chance of getting the drugs. Tell other group that you've got a 25% chance of getting the drug. And in a way, you try to throw them off and try and reduce the expectancy bias. Whether that's effective or not remains to be seen, because experiences with psychedelics are going to be profound. And sooner or later, there will be unmasking or unblinding. And as soon as a person realizes that they are getting a psychedelic, their whole expectation changes and their response to the drug changes. And at times is artificially inflated. So one more strategy that can be employed is blinded assessments and independent assessments. We do this very routinely in studies of depression or schizophrenia, where the person who is assessing for changes in symptoms or efficacy is blinded to the treatment assignment. They don't know whether the patient is getting the drug or not. And they are independent of the study team. So they have nothing to do with the investigator or the sub-investigator or the clinical research staff. So they are free from any of that influence. And often they may do these assessments remotely. And that's one way of minimizing the bias on the part of the investigator rather than the patient. Whether these things will work remains to be seen, but these are certainly certain challenges that are currently a topic of discussion in psychedelic clinical trials. One other issue with the issue as in for measurement with psychedelics is the rapid onset of effects. Usually the effects are almost immediate. People with, let's say, depression who are receiving a psychedelic have an almost immediate response. They immediately feel better. But the instruments that we use to measure this, the rating scales, they are not traditionally designed to measure immediate effects. When you use a rating scale like the Montgomery-Åsberg Depression Rating Scale, which is a standard approved rating scale, the usual time frame for when you ask questions to patients is, how have you, let's say sleep, how has your sleep been in the past 7 days? So you typically ask about 7 days, but really what you want to assess is the rapid onset of those effects in the past hour, in the past few hours. And these instruments are not designed to measure changes over a very short period of time and therefore require adaptation. Any adaptation that you make to a psychometric assessment changes the psychometric properties of the instrument and requires a whole process of validation all over again. Then there's the treatment paradigm, right? Do we give the psychedelic treatments as standalone treatments? Or let's say somebody is having depression and they would more likely than not be on an SSRI, a selective serotonin reuptake inhibitor, or any other antidepressant. Do you stop the drug? Do you stop their ongoing treatment to give them a psychedelic medication? There are some concerns about whether an SSRI could blunt the psychedelic response. It remains to be seen. There are data both for and against this concept. So this is something that will have to eventually be clarified. And that depends also on how psychedelics will be employed as treatments. Would they be one and done, as in one treatment and then people are cured and they don't need to take any more treatments? Or will it be, let's say, every 3 months, every 6 months, every year? And if you can maintain somebody's depression in remission for 3 months, do they actually need an antidepressant during that time? That remains to be seen. That is something that has not been studied in clinical trials. And unless such clinical trials are conducted, we probably won't get to the answer for this. Then there's this whole discussion around psychotherapy and the role of psychotherapy, or a guide, if you like. In almost all the ritualistic practices around the world, there is the role of a shaman, somebody who guides the person through the psychedelic experience. In modern clinical medicine, this is the role of a psychotherapist or a psychologist or a facilitator, as we call it. And this generally takes place in 3 phases. So before the treatment session, preparatory work, which takes place, which helps establish a rapport between the patient and the facilitator, set expectations, what to expect in the session, how to handle situations that arise that can sometimes be quite terrifying. What happens if you have a bad trip? So this is all that's done as part of the prep work. Then there's the medicine session where people actually receive the drug. And through that experience, somebody needs to guide them, be there for them, to comfort them. A bit of hand-holding is required. There are other strategies that may be needed. And what's more important after this is the integration, which is essentially making sense of what people experience during their session on the medicine. Now, does it need to follow, do all treatment sessions need to follow this paradigm? On an average, if you follow this, it takes about 9 to 10 hours of psychotherapist time. Is this enough? Do you need more? Can you do with less? Because there is the question of scalability. There is the question of making this available widely. And if you want a dedicated therapist, well, actually there's usually two. The guidance is for a dyad of facilitators to be present for every individual patient. So how does that impact the resources, and whether that can be scaled to larger groups and made available to people more widely? And that depends on how much you do this. Then there's this whole question of set and setting in psychedelic treatment. Set essentially refers to the mindset of the people before they are undergoing psychedelic treatment. But the setting is also important. You can't be tripping in an airport or in a doctor's waiting room. It has to be a certain environment. And that environment is designed with one fundamental concept, which is to direct a person's attention internally. So all external influences, disturbances, typically have to be minimized. In these sessions, which are medicine sessions, a person will typically have headphones that block out external noise. They will have an eye mask. The idea is for the therapeutic work to be carried out with these psychedelics, the attention has to be directed inwards and there has to be no distraction. But what is the standard? When in different places, there will be different settings. And when you have a very strictly regulated process as clinical trials, where everything is very clearly defined, is there a definition? Is there a requirement? Is there a set of principles for how the setting should be? Again, this is something that is evolving. And then there's the problem of the pilgrims. In clinical trials in psychiatry, there's a growing problem with what we call as professional patients or symptomatic volunteers. This is more of a problem in the US, unfortunately, at this time than in Europe, simply because of the way clinical trials are done in the US. And these are people who hop on from one center to the other. They pretend to have symptoms and they often don't take the drug, and they collect the money. And they are responsible for failure of a large number of clinical trials. This becomes really interesting when it comes to psychedelics. And that's why they're called pilgrims, because it's sacred. The medicine is sacred. And these people, the motivation is different. It may not be money. It may be the experience that they need to derive through the psychedelic. Again, we don't know what the answer is, how to handle the situation. And this is something drug development or development of psychedelics as therapeutics will have to deal with. So what next? I know we're running out of time. We've got maybe about 5 minutes. At the moment, the space with psychedelics is literally mushrooming with companies, no pun intended there. At the time of last reckoning, there were 48 publicly traded companies, there are 36 privately funded companies, and there are 4 electronically traded funds. That's quite a lot in such a small space. What will happen over the next few years, what we think is probably some sort of a consolidation there. There's no money in the market. So some companies will automatically die because they don't have any funds to conduct research. Some large companies might, some small companies may join up to form larger companies. But this is an evolving scenario. What will have to be defined and is a need for the future of developing psychedelics is how these treatments will be scaled, how these treatments will be made available to the wider masses and not the select few, not the wealthy not just the wealthy, not just the people who can afford to go to the psychedelic retreats, but people who are genuinely suffering from mental problems and in whom these treatments can result in dramatic improvements. If you think of the amount of psychotherapy required, 10 hours, the current wait time in the NHS for CBT is roughly about 6 months. So how are you going to prescribe a treatment to somebody when the treatment requires a psychotherapist to give you 10 hours spread over a period of few weeks? So this is something which is actively being discussed and will require a resolution if these treatments are to be scaled. We want insurers, at least in the US, to pay for these treatments. At the moment, psychotherapy is not reimbursed. Only treatment is reimbursed, psychotherapy isn't. But whatever happens, I think we are probably at a stage where there's no looking back. Psychedelics are here to stay. And I say this having been in the industry for over 2 decades. I'm more excited than I've ever been in developing drugs. Partly because I'm a psychiatrist, I've been in the clinic and I've seen these patients and I've seen the suffering, and I think psychedelics can result in changes that we have not seen before with any medications in psychiatry. So whatever happens, there will be a fundamental shift and things will change, and then there is no looking back with psychedelics. I think that's my last slide. I think I have 5 minutes for questions. I did that well.
Thank you for the presentation. There is one thing that is intriguing to me is that you seem to describe the context-dependent variables of the psychedelic experience and the therapeutic potential as things to standardize or to contain, to isolate like, the effect of the substance itself. But clinical experience, and especially in Switzerland, where psychedelic therapy is somewhat legal, kind of shows that psychedelic experience is the context more than it is the substance. And so I was wondering like how, with such a methodological process, how can we determine if what we get as data is not actually saying more about the method and the context in which it is done rather than for the substance itself, and if that doesn't raise concerns about about like let's say the validity of the knowledge that we get from that. And I'll add just a little thing very also we know that psychedelic experience is highly mediated by therapeutic relationship, for example. Certain setting, I get that the more you cut external stimuli, the more you can standardize the thing, but there's nothing in the literature that says that this actually helps significant positive impact as opposed to having mixed stimuli, let's say, and having a constant guide that can navigate that. So I was just wondering there about the scientific relevance of the data that you get regarding the particular nature of the psychedelic experience compared to others.
Yeah, so very interesting question or set of questions. I think many of those answers we don't know yet, simply because research hasn't been done. Is there a particular requirement for the setting? As in, does it need to be standardized? We don't know. Perhaps there will be a requirement based on, from a regulatory point of view, that there are certain minimal elements of that setting that might be required. Would you be able to separate the effects of the drug from the effects of the setting? Do you need to? As long as it is effective, maybe you don't need to separate the effects of the setting from the psychedelic effects per se. But the honest answer is we probably will find out. We don't know because those studies haven't been done. But these are certainly the right questions to ask.
I suppose relatedly to that, I was hoping to speak a bit more to the double-blind randomized control trial as the gold standard, and certainly given your work history. So there's a certain way in which, as developing psychedelic drugs, it doesn't really look good for us to say, hang on, you should change the rules for our drug, it's actually special.
Yeah.
The thinking like a bit further towards the future, like the seeds of the double-blind placebo control trial maybe in 1700s. If we are still about 500 years from now, still developing drugs, I really hope the double-blind randomized control trial is not the gold standard. Right, there will be developments, and so there will be historically contingent forces that shift our ways of thinking about evidence and data in drug development. It seems to me like psychedelics are a very good candidate for doing that, because lots of features of the RCT are obstructing discussed. You can't really do that.
Yep.
Is there a point, say in our lifetimes, when that is to actually be pushing the FDA or it's to say, no, there should be radically different ways of thinking about it?
Yeah. Excellent question. And I think I agree with you. I think the randomized double-blind placebo-controlled design is a bit overrated. If you get large effect sizes from an open label studies, does that mean, are you going to discount those results simply because it's an open label study? I mean, patients who were suffering from, let's say, PTSD for decades and their PTSD is gone, but that was done as part of an open label study. Does that mean those data are less sacred or less pure than a randomized controlled trial? And this is where I think when we talk about the paradigm shift, it's not just in the treatment of mental illnesses with psychedelics, but also in terms of how we do clinical trials with psychedelics in patients with mental illnesses. I think the way we do it will have to change simply because we cannot do it the way we used to do it. There is no way to control it with a placebo, the clinical trial.
Just to build up on your point, actually, there is a precedent where double-blind randomized controlled trials don't necessarily need to be taken for a drug to get market authorization. So, since 2000, there's about 78 drugs that have gone through the process of open-label studies. Most notably, Zolgensma, which is an SMA-related drug, was actually that went in on 16 individuals on an open-label trial design. And so, companies that are interested in developing this space should probably be taking this to the FDA and knowing that there are examples already out there and saying, why don't we try to push for more of a change rather than just designing it to the gold standard, because there is a precedent that can actually be used as well.
Yeah, you're right. With rare diseases, we often see those drugs being approved in a small number with a small number of patients in open label fashion. And there is already engagement of the regulators. As you mentioned, companies are speaking with the FDA. I've been in meetings where we've spoken with the MHRA about doing these kind of clinical trials, and the regulators recognize that these set ideas about doing clinical trials, they will not be applicable to psychedelics, but it will take time.
We don't have time for more questions.
Thank you.
Great. Up next, we have another keynote presentation by Professor Morten Kringelbach, who will be talking about thermodynamics of mind, a novel framework for brain function. So we'll just get him set up real quick and then start with the keynote.
We should just share your screen. The sound is being picked up here.
All right, everybody. You can just get settled. We will now be starting Morten's talk. Take it away.
Yeah, thank you. What a pleasure to be back here. Such a lovely day. I'm going to talk to you about something that I've been working over for quite a while now. In fact, in order to talk about thermodynamics of mind, let me just get this. I need to take you through a long, see there's something wrong with the sound there. Take you through probably about 20 years of research that I've done while I've been here in Oxford on the pleasure system. For some of you, of course, it's not obvious how psychedelics might fit with pleasure, but I think there's a case to be made there. Once I've done that and sort of explained to you how it is that we've come to these ideas, I'll then take you through the idea of what eudaimonia and human flourishing is, how psychedelics can inform that, and in particular, how it is that really in order to understand that, we need to understand about the hierarchy of the mind. Then I'll take you into this novel theory that Gustavo Deco and I have come up with of thermodynamics of mind, show you how we can do whole brain models of the non-equilibrium that your brain is in as we speak. I'll then take you through some of the exciting work that we've done on turbulence of the brain, turbulence in different brain states, and particularly in psychedelics, and again, how one can model that to fully understand what it is that psychedelics are doing to us. We're not quite there yet, but I think these are the kinds of tools that we need. So I came to Oxford in 1998, which of course is a long time ago, probably before many of you were born. And I did my doctorate on the functional neuroanatomy of emotion. And as part of that, I came to realize that emotions are these fantastic, interesting molecules. But of course, if you want to understand the actual mechanics of that, you need to sort of break it down into smaller states. And I became very interested in pleasure and pain and desire and those kind of things. And so through the years, I've been sort of focusing on that, becoming more and more interested in not just what is pleasurable, like food or drugs or sex, but also becoming interested in the kind of things that are meaningful to us, things like psychedelics. Because of course, as you all know, most people, when they take psychedelics, report that this is a highly meaningful experience to them. Things like meditation, which of course is also potentially a highly meaningful state that is perhaps more controllable than what we find with psychedelics. But also things like music, and of course, all three of them work together in very interesting ways that we've looked at over the years. And so I made I wrote a book in 2014, which sort of put all of these things together. But of course, if you were to look at that book, you'll see that what it really is missing is what I'll talk to you today about, namely about these meaningful states. So in about 2006, we formed a group, and I work very closely with the man you see in the middle, Gustavo Deco, on doing a lot of the research you've seen here. But I've also been very fortunate to work with other very gifted individuals, including, as you can see in the top left corner, Peter Wust, who's a professional musician and director of the Music in the Brain Center in Denmark, where I also, in normal times, but these are not normal times, would go about once a month to what has now become the largest center for music in the brain. Right next to him, you find Alan Stein, who is a professor of child and adolescent psychiatry, with whom I've been interested in the social interactions and the meaningfulness of social interaction with babies, as I'll mention to you as well. Then there's Peter Weibrow, who is one of the foremost psychiatrists in the world, for many years, the director of the Semel Institute at UCLA, and probably knows more about affective disorders, and in particular bipolar disorder, than anybody else. And then, of course, the most extraordinary pioneer of them all, Kent Barrage, who is probably the world's expert in pleasure in rats. He's been able to operationalize what happens in rats and take that system apart so that we now have a molecular understanding of why it is that we like things and why we don't like them. And on the route, I've worked with many, as you can see, many excellent scholars. I've been very fortunate that I worked with a lot of women over the years. I'm very keen to promote the research of women because I think being a researcher in this kind of world is difficult, but especially difficult for women, especially if they want to have children as well. And I've also been fortunate, and we have a lot of mascots in the center, in that we have a lot of my former students and current students that actually have had kids while they've been doing this research. And I think that's entirely normal and should be encouraged. So hundreds of people have gone through our scanners. There are not many pleasures that you can think of that I haven't had somebody in a scanner at some point experiencing. At the top left, you can see what happens when people take food, pleasurable food, non-pleasurable food, when they have social interactions, when they take drugs like methamphetamines. These are drug-naive Oxford undergraduates taking methamphetamine for the first time. And of course, what you can see is that the reward system is going crazy because methamphetamine is a very powerful drug. We've also done things like gambling. We've even done sex studies, although I couldn't do them in this country because, of course, we are meant to close our eyes and think of England. So I acquired a Dutch friend, and we did all of that work in Holland. So it's been a wild ride. And at the center of that ride is a region of your brain, which is just over your eyeballs, called the orbital frontal cortex. It's a rather large region, for many years quite mysterious, and specifically it was very mysterious because if you do fMRI scanning in the normal way, you tend to get dropouts in this part of the brain. So it's almost invisible to most scans because you have these large sinuses and there are artifacts from that volume of air that basically destroys the signal in this region. So we spent a long time optimizing sequences so we could actually see what is going on. And what we discovered was a whole new continent of basically finding out how it is that we link rewards and the experience of reward to the experience of having that hedonic experience. And along the way, there were various high points, things that were truly pleasurable. One of the things I've been very excited by and have worked with Alan Stein that I showed you earlier is cuteness. Why is it that babies are so cute, and not just baby, but also kittens and puppies? It turns out that if you use another scanner, which is really exciting, is called an MEG scanner, which at the time when I was actually a postdoc in I think it was 2003 here in this department, we were working with people in Birmingham at the University of Aston, where the only MEG scanner in the country was at the time. And so I had always been interested in this idea that there's something special about the way babies look. Konrad Lorenz, who's a Nobel Prize winner that some of you may know, did his work on imprinting. And he was able to show that if you show yourself to a goose within the first hour after they've been born, they immediately think you're their mother. And so there are these wonderful photos of Konrad Lorenz in the water with a lot of goose looking at him like he's the mother goose. He had the idea, in fact, during the Second World War, he published a book in 1943, where he talked about what he called Kinship schema, this idea that babies basically, the only armor they have is that they're cute. And so there must be something in their brains. Now, if you do these studies with fMRI, you don't see anything in the orbital frontal cortex. So when I did this study with MEG, I was very surprised to find that there was a very quick, fast signature, as you can see in the top row here, to the baby and not to the adult, at the same time as you get activity in the fusiform face area. The fusiform face area, of course, is this area of the brain that if it's damaged, you can no longer see faces. So in other words, it looked at this, there were very fast dynamics in this region of the brain that I had studied for many years in other contexts that also seems to be engaged with something like cuteness. And since then, we've gone on to show that if there's something wrong with that template, if the baby has a cleft lip, then you don't get this fast, but you can, this fast signature in the brain, but of course you can still find them very cute. We've also shown that this is true when you hear baby sounds. We've even shown that, in fact, when you look at puppies and kittens, you actually also get the same response. And then only later do you realize that it's not a baby, but then it's too late. You already go, oh. So that's kind of cute, I think, in many ways. And it's like a Trojan horse, which is the argument we made in this review that we published in 2016. And it tells us, I think, a lot about our humanity. And talking about humanity, I've also worked with this brilliant man, Tibu Aziz, who some of you may know. He's one of the most brilliant neurosurgeons alive. And here he's showing his work to somebody you might recognize as well. And what Tibu does is that he has personally found the target that helps people with Parkinson's. So we now know, because of his work that he did in the early 90s, that you have to stimulate the STN in order to help with the kind of tremors that you find in Parkinsonian, various Parkinsonian disorders. So that's kind of cute, I think. But together with Tibu, he's also had a very large case series looking at what happens when you have to treat chronic pain. Now, many of you will know that the first line sort of treatment for chronic pain would be to give people opiates. Many of you will also know that if you give people opiates, it lasts for maybe 2 or 3 months. And then after that, it doesn't really work with the chronic pain anymore, and you're stuck with the addiction, which of course is unfortunate. Through Tibu's work, he's realized that if you stimulate the pericaudal brain, so it's part very deep in the brainstem or part of the sensory thalamus, you can basically stop a pain in a chronic in a phantom limb that is no longer there. Now, most people have amputations. About 25% of them will develop a chronic pain in that limb. And we use the scanner, the MEG scanner, to show that when you get that when you stimulate in the MEG scanner, and of course we could do this because it was an MEG scanner, so there were no real danger to the patient. We were able to show that when you get that concomitant pleasure or lack of pain, you find again, as you can see in your top right, that there's very significant activity in the orbitofrontal cortex. Again, linking this particular region to the subjective experience of pleasure. Now, interestingly, as I also show you at the bottom right, if you stimulate at 20 hertz here, you get this pain relief or pleasure. But if you turn it up to 90 hertz, which we did initially on table, what you find is that the pain becomes unbearable, suggesting that really pain and pleasure are mirror images. It's the same system that subserves both, which of course is really interesting. And when I told that to a friend of mine, Annie Cattrall, who's a very gifted sculptress, she said, why don't we make an actual sculpture of this? Why don't we take the actual cabling, the actual myelin that goes from this particular nuclei that you stimulate and see where that goes. And so we did that, and we ended up with this amorphous thing that basically are the cabling. That's the cabling you're seeing now that is mediating pain and pleasure in your brain. And so if you go to the museum, you can see this particular thing. But also curiously, and you can see in this article, it looks a lot like the kind of teas that you can put in water, and they sort of make these beautiful flowers. So in general, all of this work can be summarized in something that I call pleasure cycle. Right now, I haven't had my coffee. It was a late morning. And so I'm thinking about how I can get to the coffee. I'm thinking about how I can, it's an interesting thing, isn't it? Why doesn't it change? Something gone wrong? Let me do that again. Should I connect again, or is it shared? There we go. That's coffee for you, right? So I'm now in the wanting phase. I'm in the impetitive phase. I would like to engage with this coffee. Once I have the coffee, I get a lot of pleasure out of that in the consumatory phase. And after that, I can go back to talking to you again. And it turns out that, of course, most of life is like this. We go through these cycles on a daily basis, many times a day. And of course, if we were addicted to something like opiates, we would go through that cycle over and over again, and we would need larger and larger doses to actually reach the phase where we had some pleasure. And so I think that's a potentially an interesting kind of story. And the key thing, and this is where the genius of Ken Barrage comes in, because of course in rats we can do things that we couldn't necessarily do to humans. So through lesioning studies and optogenetics, Ken Barrage has been able to identify the various regions, and I show you the rat brain on the left and the human brain on the right. But of course, on the right is just tentative regions that I have identified with neuroimaging studies. On the left, we have causal evidence. These are the kinds of regions that are, if they are lesioned, so if you take out the nucleus accumbens, for instance, you can no longer feel pleasure, but only for a couple of days, and then the rat comes back to actually feeling pleasure. On the other hand, if you take out the ventral pallidum, VP as it's called in here, if you take that out, the rat will never be able to experience pleasure again. So in other words, the take-home message from this is that we now know what the machine room is doing. We know what is happening in the brain when you are feeling pleasure. what happens when some of this goes wrong. And of course, one of the major hypotheses that immediately comes out of this is that if you have an affective disorder, there's most certainly something wrong with the machine room. And there's certainly something wrong with this. Let me try it again. Here we go. Perfect. Looks like some kind of timeout. And in the meantime, we set up the center in 2016 for pleasure in the brain when it comes to music, which again, I think is a really interesting topic. And those of you who are interested in that should definitely come to and talk to us. And you can see there's quite a few of us now. I won't talk too much about the music today, but I thought I'd just mention it because I find it hugely interesting and of course, potentially extremely meaningful to not just us, but to everybody. So what is the sort of take-home message for 20 years? Well, I think the take-home message is that evolution has built survival machines. We need to be here somehow. And in order to do that, we have these circuits that makes it possible for us to pursue and want things, to actually like them and remember that we like them so that we can now go on and actually survive. So in other words, we have what we call a hierarchical prediction system that relies on reward and pleasure. And really, I think that is the key difference between us and, say, a Turing machine or one of these kind of von Neumann machines that are all around us. We have these systems, these subsystems that basically allows us to solve what in computer science we call NP-hard problems that can be solved in non-polynomial time. And that basically allows us to shortcut. We don't need to have all the information that we need. We are basically able to just use the information at hand and take decisions in the world. And those cycles that I described for you have both short ones, like the one that I'm experiencing for coffee now, but of course, they're also the things that happens over hours in the circadian rhythm, seasonally, and lifespan, and so on. And of course, if something goes wrong with that, that's probably why we have these disorders. But the big question then becomes, how do we then try to actually understand the larger network, not just the machine room, but the rest of it, the reason why things feel meaningful and so on? And that really is a difficult question. At the top here, you see some whole brain imaging of the neuronal activity of the larval zebrafish. And as you can see, there's a lot going on. And from time to time, you see these avalanches where the whole brain just goes. And the question is, how does one actually describe that? Because of course, a lot of the imaging that I showed you before assumes that the brain is just a system in equilibrium, that's a stationary system, and that you can use stationary statistics to describe that. And so in many ways, it often ends up with lots of us going up and showing you these blobs on screens and saying, this is what the brain is doing. But of course, any of you who have seen this movie and will know a little bit about mathematics and physics will know that that's not how you treat complex systems. And so really, we need a new kind of way of engaging with that particular system. And that's really the kind of challenge that Gustavo and I have been faced with over the last probably 15 years. And the way that we've been going about it is that we've taken our lead from Thomas Aquinas. Thomas Aquinas was a medieval philosopher who was very enamored with Aristotle, who is somebody I'm also very enamored with. And he said, quid recipitur mundum recipientis recipitur, meaning that the content is shaped by the container. And so what we've done is that we've actually, in order to really truly understand the system, it's not enough just to observe that there are correlations that might give rise to this globology that I'm telling you about. What you really need to do is to build a model of this. And once you have a model that can reproduce the data that you have, you can then take that model apart and find out which of these parts are actually the ones that are causing those changes. And so we've built a whole program of doing this. And one of the things, for instance, that we can do, and it's doing it again. Let me do it again. There's something, I think this is probably something to do with Max, yes?
Yeah, I was just reading about it.
Let's see. I'm now
You're not
I'm sharing again. Share. Here we go. What was the password?
because your password.
Oh, it's my password. Install the Zoom audio device. Okay. What is it they say about technology? It's a word for something that doesn't quite work yet. Let's see. We're waiting. Should I connect again, maybe?
I will just cancel this and try Skype. Is it good enough?
Okay. Okay, here we go. So I just wanted to show you an example of the kind of things that one can do with these models. But remember, this model is still assuming that the data is stationary. So we're not taking into account the potential non-equilibrium, or not just potential, the actual non-equilibrium that you have in your brains. And so in what we call the awakening paradigm, we were looking to see whether we could take data from people when they were asleep and people were awake and try to actually move one to the other. And in this paper in PNAS, you can see that we managed to do that. And later on today, Jakub Vohryzek, a former PhD of mine and now a postdoc with Gustavo, who's sitting here in the front row, will tell you about how we've applied exactly the same kind of techniques to LSD data to understand what it is, to psilocybin data for depression, for treatment-resistant depression, and shown how it is that we can, in that way, find out about what it is that makes some people respond and some people non-respond. In this particular case, we were just able to show that basically by manipulating the model, we could find out what part of the brain we needed to stimulate to wake somebody up from sleep and vice versa. So I think there's a lot of potential here, of course. Now we just did it as a proof of concept, but imagine you could do this for many other things. And I think what Jakub is going to show you is a first step in that direction that actually not only will tell us about how it is that we can change things, but also give us a full understanding of what it is that's going on underneath the hood. So then all of this kind of happened, and then pandemic happened, and then we thought we'd better set up a center for eudaimonia and human flourishing. And that we've done. Many of you should come and visit us. It's an interdisciplinary center based at Stoke House and at Lineker College. It's supported by the Carlsberg Foundation and the Pettit Foundation. And it's really an interdisciplinary place because, of course, if one really wants to understand something as important as eudaimonia and human flourishing, one can't just look at the brain. You have to understand the philosophy, the anthropology, to name a few of the disciplines that one can engage with. The problem, of course, scientifically is that eudaimonia is really difficult to get. Unlike if I put you in a scanner and gave you some very good coffee or some very good chocolate, you would probably feel pleasure, but it's very difficult to find the kind of states that I could reliably induce in people by putting them in my scanner. But as I write up here, I think there are at least 3 avenues that are proving very promising. One is music, which I won't really talk about today. One is meditation, which I will talk about today. And the other one, of course, is the topic of my talk, namely psychedelics. But in order to actually really understand that, we now have to understand what is a brain state. How does one actually measure a brain state? What is the stability of that, and how do you go between states? And many of you will know this particular hypothesis by Carhart Harris and Friston. Some of you may know them, and you will know that the photos that I put on them don't really look like them in real life. But I think they're great photos, and I think that's what matters. But basically, they have this leading hypothesis saying that psychedelics work by modifying the brain's hierarchical processing in a dose-dependent manner. There's a lot of words there. So the big question, of course, is what do we actually mean by hierarchical processing? And of course, what is a dose-dependent manner? And so those are the kinds of things that we need to unfold. So in terms of the hierarchy of brain processing, there are many different ways one can go about it. You could think about it in the sense that you see at the top left of thinking about how things go from the sensory to the higher areas. You could look at gradiental hierarchies like Margulis, or you could look at the free energy, which of course is what Carl has been doing for the longest time, and looking at these free energy landscapes. And of course, once you've done that, you can then do the whole brain modeling that I just told you about. And for those of you who can't see Jakub like I can, this is a photo of him. So what are the kinds of measures then that allows us to establish the hierarchy? Well, one of them is what we called normalized directed transfer entropy, which is Granger causality done right. It's a way of finding out how different time series are basically causally interfering with other time series. But one could look at things like functional harmonics, as in the work of Selin Atasoy and what Andrea Luppi will tell you about. Namely, this idea that there are these harmonic states that one can then use to establish a hierarchy of brain function. One could also look at turbulence, and I'll tell you more about turbulence, but basically turbulence is a universal principle that governs a lot of things from the smallest of atoms to the galaxies. And of course, it also governs the brain, but only recently did somebody have the idea to actually look at that. And then, because we could really then start to look at the thermodynamics of mind and look at Tenet. So this is where the talk gets a little bit more hairy in a moment. First, though, I just want to give the idea of an actual hierarchy to you. So what you see in different colors here are the colors of the sensory regions of the brain. Red is in the back, you've got the light blue, the dark blue, and then you've got the sort of the taste and the smell in the front. But of course, the key thing when you look at that particular picture, as I show you here, is that here be dragons. There are lots of white parts of that network. There are lots of areas where things come together, where they're not just processing, say, visual stimuli, but actually higher regions. And of course, this is really where we want to go dragon hunting, right? We want to find out what are the top of the hierarchy. And so again, I showed this to Annie, and she couldn't help herself but actually make a set of sculptures that basically shows you the 5 senses and the activity in the brain of the 5 senses, but without the brain. So this is basically what vision looks like in terms of just blobology. But, you know, just thinking about the scientific principles here, a very influential paper by Marcel Mesrilam from 1998 has these concentric rings that we're missing, but let's see if we can. Let's see if we can do this again. It's terrible sitting online watching this, yes. See? Are we
I think that's why.
Great.
Now it should be. Yeah, so it's a bit sensitive. So is it? We can ask now the online audience.
Is it okay online? To all our listeners in Russia. So, yeah, I'm sorry about that. What I want to say, is it still not working or is it?
It isn't. Here it's picking up.
Yeah. Here it's.
Why is it not sharing sound?
Should I?
Wait, well, maybe we can try using yours.
Is that? That could be an experiment. Okay.
That's because you have.
Should I turn that off?
Alright.
Okay.
It's because of the noise cancellation, isn't there? So I think we have to use the
Okay.
mute these.
Okay. Are we good online or not good online? Can anybody see online whether we're good?
Okay.
Fantastic. So apologies again. You missed some great sound effects. I can still hear myself. That's pretty good. Oh, it's this one. Whoa. Okay, back to this. So on your screen, hopefully at home and here, you'll see a guy called Erwin Schrödinger, who, of course, many of you will know Schrödinger's cat. And the big question, of course, is whether Schrödinger's cat is dead or alive. But that was the kind of work he did when he was a quantum physicist. Towards the sort of, when he got a bit older and towards the end of his life, he got incited about probably a more interesting problem, at least to me, namely the question of what is life. And this one just died again. So let's try it again. There must be some Russians here somewhere, right? I mean, that can only be the start video. Share screen. Start video. Share screen. Can you see? No, we don't see anything. Stop share. Share screen. Should I try logging in and out?
I think it's an issue apparently with the new Mac operating system and Zoom.
Okay.
Some computers are having it.
Okay, here we go. Let's just block out for this.
Yeah,
Just,
I can link you again.
yeah.
Now they're seeing this.
Now I see. Okay, I just need to change the right. Oh, magic. It's a bit like Tom Jones, isn't it? Who's going to be my little pussycat tonight? No. So what I was going to say about Erwin Schrödinger is that, of course, he, as you may have heard online and here already, he worked a lot on quantum physics, but he then got very interested in thermodynamics. And he got particularly interested in this idea that really the avoidance of decay is really what thermodynamics is all about. It's basically about trying to make non-equilibrium interactions with a complex environment that means that you can survive. And of course, the brain is at the heart of this. So what do I mean by that? Well, really what it is that we want to break what is called a detail balance. In a system that is in equilibrium, everything is just running smoothly. But of course, what we want to do is not make it run smoothly so that we can actually explore the underlying landscape and explore the possibility of changing something in the world. So for some of you will have seen this movie called Tenet, which is by Christopher Nolan. And it's a curious world in which things go both forward and backwards. In a moment, you see this car, it's a disconcerting experience because suddenly things happen in this world that doesn't happen in our normal world. And this is why he's looking a bit disconcerted, because as he's now looking at the car, he knows that there are funny kind of things happening. Like, for instance, how is it that this particular car, which is blocking their actual, the movement actually then goes backwards in time and basically comes back and do something else. And then, of course, the video stopped. Let me do it again. Backwards in time. We'll try again.
Share screen settings on Zoom.
Yeah.
So apparently this might be able to use. So share screen. Yeah. And then is there a use TCP connection for screen sharing? Is that? Let's see if this solves.
Okay. I think we have a solution. Maybe. Stop share. Share again.
You try sharing this, like the actual.
Yeah. No.
So no. Try
Do this.
sharing that. Yeah.
Okay. Okay, you missed the good bit. Whenever you saw the car going backward, right? So I'll just show you this, which hopefully you will be able to see online. At the top left, you'll see a glass being shattered by a bullet, and you're in no doubt that this is something that is going forward in the world. On the bottom left, on the other hand, you will see a glass coming back together after it's been shattered. And all I've done is I've reversed the actual sequence of pictures. But of course, you immediately know that what happens in the bottom left can't really happen in the world. On the other hand, if you look at a reversible process, and that's what I'm showing you on the right, you'll be very hard pressed to tell me which one is the original one, the one that can happen in the real world, and the one that can't happen in the real world. Because of course, it's a reversible system. It's not entirely reversible because if I left it running for long enough, eventually because of kinetic energy, it would stop. But at the moment, I could be fooling you and in fact showing you the other thing you wouldn't know. And why is it that you wouldn't know? What is it that is happening? Of course, if to understand that, we need to try to share the screen again. Share screen. Do you see anything? No. Share screen. No. Stop sharing. Share screen. 9. Oh, here we go.
Whenever there's an animation or a video.
Yeah, then it basically just breaks. Okay, when that, so much for animation, yes. So the reason why this works is because of entropy, and it's specifically because of the thermodynamics. This is Rudolf Clausius, Sadi Carnot, and Arthur Eddington. Arthur Eddington is probably the one who is most known in terms of his idea of the arrow of time. This is a key concept in thermodynamics, the idea that there is a sort of a set of events that basically will change over time. And again, you know, I won't bore you with the details because you can read all about that in our paper, but basically you have these non-reversible and reversible processes that basically is all about how the entropy production is changing over time. So work is being done, and then that basically means that you have a change in the entropy. Now, there's a lot of beautiful work that has been done on that, specifically the Clausius inequality, and Jasinski is another person, Christopher Jasinski has done a lot of work on this in microscopic systems. But we had the idea, Gustavo and I, that we could use this on brain activities. We could take the forward trajectories of the kind of signals that we get, and then we could reverse it. And then we could start to look at to what extent these signals in the brain are reversible or not. And in order to really make this particular decision, and remember when I showed you the balls going back and forth, it was very difficult to see. So in order to find out whether we could classify that in the best possible way, we turned to deep learning. Some of you will know a lot about deep learning. Some of you will never have heard about it, but most of you would have used things like Google Translate. And you will know that if you use these very powerful neural networks, you can basically classify and do various clever things. they're very useful technology. And here we use this particular useful technology to find out whether we could classify these different things. I don't think there was an animation there, right? Let's try it again. Could it be edurome that is unstable? Can't be, no? Let's try it again. Okay, we're back in business. So anyway, we use this method. Long story short, you do this, you do deep learning on this, you find out what is forward and what is backward, and you can basically classify all of the same tasks that I showed you before. And you can show that when you're resting, you're much closer to equilibrium than when you're doing various tasks. And depending on the task, and this is what I show you here, up here, you can see how it is that you become more and more irreversible. And the most irreversible task in these 1,000 people is basically the one where you have over almost completely irreversible in the social task. And again, we can show the math of this shows that you're breaking the detailed balance. Again, you know, I'm just flashing you images and asking you to believe that that is actually what is there, because we are kind of running out of time. But the key idea, and now I can see I again lost connection. I'll try again. Here we go. So you think it's the edurome? It must be, huh? So when you do that, you would then can ask yourself, what are the networks that are responsible for this level of reversibility? What is the most stable network? The network that is present when you're both doing something like rest and doing different tasks. And for those of you who are familiar with brain imaging, you will know that one of the key networks that Robin Carhart-Harris and others have found have been involved in psychedelics is the default mode network. And some of you will not be surprised to find that if you look at the most stable network over time, and this is the one that there's an animation, so it may stop, but it didn't. This is the region of the brain that is basically, a lot of people think that that is essentially the network that is involved in governing the kinds of activities that you see in rest. But here, importantly, it's also present when you do tasks. And in fact, it's sort of the underlying network. And so if you look at the regions that are being driven by the environment, and remember, this is how you go out of thermodynamics, is that you actually are, you're being driven by the environment, you've been driven by a task so that you actually go away from equilibrium. And you then look at what is common to all of those compared to the ones that are not being driven, that are stable over time. So in other words, if you want to solve a task, you need to have a stable network that is then being able to be influenced by the environment, but also being able to continue doing what it is that you're doing, namely solving a task. And again, if you do the intersection of that, you get something that is very close to what I showed you earlier with the other method that assumed that it was all stationary. And again, we now have another way of showing what it is that is happening in the brain. I'm just a bit conscious because of these interruptions that we are running a bit short, so I'm just going to jump to the next slide. So we get to the meat of the funny things. Here we go. So I won't be able to show you the whole brain modeling. So of the non-stage, but that's not so important. I think it's more important that we talk about what it is that turbulence, how it is that turbulence, and do we see that now? I just need to do that. Yes. So in the remaining 10 minutes, I'll tell you what it is that is so important about this work for psychedelics. So a lot of you will be familiar with turbulence. Turbulence is another system which is in non-equilibrium, very much in non-equilibrium. If you sat on a plane, you will know that when you hit turbulence, everything, you have to be strapped in because otherwise you might hit the ceiling of the actual plane. It's again, like I told you, it's a completely normal phenomenon that governs a lot of things. And what it particularly does, it basically makes sure that you can have energy dispersing over large areas. So if you look at something like, for instance, and again, this doesn't work. It's great. Let me just Yeah. Yeah. Okay. Let's see how long this is going to last. So too many equations for the last remaining 5 minutes. What I wanted to show you is what happens when you then try to model this turbulence that happens in the brain. And the way we do this is not like what you would see if you were sitting in a plane, which of course is where you have fluids or atmospheres moving in terms of the turbulence. But imagine instead if you thought about how the brain is communicating, namely through oscillations. It turns out that you can describe the oscillations that happens in the brain through something called, basically through oscillators. And Kuramoto described how it is that these oscillators are communicating together. And when you then apply that to human brain imaging, you can find that this is just your brain resting, the video that I'm showing you. And you can almost see the kind of vertices in the video I couldn't get to work before, namely this idea that you have these large vortices that become smaller vortices, and eventually they have energy evaporation. And so in other words, you have a way of transferring energy in a scale-independent manner, which of course is what is really important when it comes to the human brain, because you may or may not know this, but your brain are extremely slow. So from one neuron to the other, it takes about 40 milliseconds. Now remember earlier on, I told you that for consciousness, it takes about 200 milliseconds from when you see something to you become aware of it. And you can show that with various experiments, and you can look at the brain activity. You can show that after 200 milliseconds, you get this dispersion of activity. Now, the big question is, how does that happen? Because if it takes 40 milliseconds to get from one neuron to the other, you don't have a lot of time to actually do all of the computations that you need in 200 milliseconds. If it was just a linear process, you would only have 5 of them, right? But of course, the brain is a network, and the spreading of information happens through this particular turbulent dynamics that I've shown. So in other words, this particular framework tells us something about how it is that activity is spreading over time. And the way it can spread is partly through that, but also partly through, and again, we'll skip over a lot of things, through these long-range connections that you have in your brain. So one of the reasons, for instance, that when I showed you the babies earlier, that you basically could see that there was activity in the frontal part of the brain almost at the same time as you got it in the back. The reason why that activity could go that far was because there is basically a connection from the visual cortex to the prefrontal cortex, a long myelinated fiber that basically can send that information. But that doesn't explain why it is that once you then become aware of that, how that becomes distributed to the rest of the brain through this global workspace. And I think the answer, and again, if you're interested, I can tell you more about it, but I think the answer truly is that you have these turbulent dynamics. That's the way information is spreading. And now the big question then becomes, how is that then changing in different brain states? And so this broke again, so I'll just unbreak it, maybe. Maybe not. Now we go. Good. Okay. Blah, blah, blah. So if you use this math, if you use this principle to look at what happens in different states of the brain, you can then start to look at how it is that information is being driven very differently in this turbulence when you take LSD, when you take psilocybin, or when you are in different states. And these particular different states that we've investigated with this paradigm is meditation. So what we had was we had meditators in the scanner, and we could then look at to what extent that the turbulence, so the non-equilibrium system, is changing in meditation compared to rest. We could look at how it changes in deep sleep, which is the middle column that you see on the top. And we could look at how it changed in disorders of consciousness, whether they were minimally conscious or whether they were unresponsive wakeful syndromes. And what you see here is basically the kinds of patterns, the kinds of oscillations that happens on a particular scale. And again, the details of this is a bit convoluted, But when it comes to these different vortices that I talk about, they will have a scale. And if you look at a particular scale of this, you can see how it is that the information is spreading. And so what I then show at the very bottom is probably what is most understandable about this figure, namely essentially a spider plot of how it is that these maps that I show you in the middle, how it is that they can be organized and shown as a spider plot in terms of what larger scale networks that they're changing. And what is very clear is that they're very different. So meditation is very different from sleep, which is very different from two different kinds of coma. So in other words, this method is very precise and very good at actually finding out what it is that is different about these states in terms of the turbulence. And we shouldn't be surprised that there's a lot less turbulence when it comes to the disorders of consciousness than, for instance, in the awake state. And that's really what we're showing here. Now, the question then becomes, what happens if we take it to the opposite? So if you like, we have the awake state here, where we are sort of at some level, and then we have the sleep, we have the anesthesia, we have perhaps the meditation somewhere in between, and then we give people psychedelics. Will they be more like these states, or will they be in a different place? And the nice thing about this is that by using this particular paradigm, we can show that the information cascade of turbulence across different scales is also changing. And again, the bottom line is that things like LSD and psilocybin, there's a reason why they feel very different, in that the way that they change the turbulent landscape is different from each other. But if you compare them to the other ones, and now I'm comparing all of the 5 different states. So we have meditation at the top, we have sleep, we have various forms of comatose patients, and then we have the LSD and the psilocybin. What you find is that they have very distinct signatures, meaning that, in other words, information is transferred in a very different way. And the reason why they're transferred in a different way, and this is the final part before we can take some questions, is of course because they act differently on the brain. They're changing the functional landscape of the brain. So the functional landscape of the brain, of course, is dependent on the structural connectome that I showed you earlier. And things are moving within the structural connectome. But then, of course, we have a potential for changing that very briefly by changing the neurotransmitters. So neurotransmitters basically work locally in the brain to change whether you can travel from one part of the brain to another, from one neuron to another. And what I'm showing you here are the receptor maps of the serotonergic receptors. And of course, the one that I've highlighted is the 5-HT2A. And we know that if we block the 5-HT2A, we basically, if we give people psychedelics, the actual subjective experience of that is much reduced. And the reason why that is because basically by taking psychedelics, you are foremost changing the 5-HT2A distribution. So you're changing the landscape in which this turbulence can happen in the brain. And you can do a modeling of that, and you can show exactly how that happens. You can show exactly what it is about this particular landscape that is changing. The big question, once you have a model, of course, and this is where we're going now, is could then one start to think about how it is that you could perhaps develop new psychedelics? Could you make psychedelics that basically would change the landscape in a different way and yet give rise either to no subjective experience and still be potentially effective for things like treatment-resistant depression? Or could you have some where you actually attenuate the actual response? So I think there's a lot to take in, and I'm sorry for the technical problems. Perhaps we should take a few questions. This is my friend. Everybody should have a friend in Barcelona. And this is my favorite animal. Thank you.
Time for questions.
Are all of these responses universal across all humans? Meaning, when you do meditation, psychedelics, music, are you biologically or designed to have certain built emotions, or are there some other
It's a very interesting question. I think it's safe to say that our connectomes are more similar across cultures than different. It's clear that there are different paths to both pleasures and to flourishing in different cultures. And that's exactly why the center is interested in that. It doesn't mean that the ultimate state is not the same. I think I have not seen any evidence suggest that not all people can feel pleasure, that not all people can feel desire, that not all people can feel meaningfulness. But the route to that particular state is different for different cultures, which is why let's go back to psychedelics, we do work with people in Maastricht on ayahuasca. And of course, what we do is we work with the Santo Daime Church. And what they do is that they, as you know, they play music, and then instead of the sacrament, they take ayahuasca. And that for them is highly meaningful and is also dose dependent. They will take different kinds of doses depending on what it is that they're used to. It doesn't mean that that state of meaningfulness is very different from the kind of meaningfulness that other people say other psychedelics get to. In fact, all my data suggests that that depends on that machine room that I told you about in the beginning, and that machine room basically is similar or present in all of us. Of course, except if there's something wrong with that machine room, in which case things like depression is the almost certain outcome of that. Thank you.
Great. Now we will have a lightning talk by Mirima Sabanovich about finding long-term effects of psychedelic drugs on cognitive and anatomical plasticity in a mouse model. I apologize for all the technical difficulties. All the further presentations are going to be streamed directly from the conference desktop, so we don't anticipate any further issues with the live stream. Thank you very much.
I know that statistically the worst time to give a talk is right before lunch.
If you speak there,
Perfect. Okay. But I hope if you're thinking about sandwiches, you're not alone. But if you've been looking forward to seeing a world premiere, some new exciting data, hopefully this makes it worthwhile for you. So my name is Mirima Sabanovich, and I'm a final year PhD student here at University of Oxford, and I've been developing a project for the past few years, looking at long-term effects of psychedelic drugs on higher levels of plasticity, specifically anatomy and behavior. I don't think that this audience needs a lot of reminding why we find psychedelics a fascinating area of research. But for me, it was just thinking about the goal of any therapy. We want something to induce a fast and lasting change in behavior. And the traditional pharmacological therapies that we have for mental disorders remain not as effective as we would want them to be. They require weeks or even months to start showing effects. If they do at all, they require you to keep taking your happy pill in order to feel those effects, and are associated with a wealth of different side effects. And even if you are able to stop taking the drug, you're most likely to go through a withdrawal syndrome. On the other hand, the past decade and a half has seen a very publicized research into psychedelic-assisted psychotherapy that proves that with just a single dose or a few doses of a psychedelic, you can have fast and lasting remission from very different disorders across very different symptomatologies. And when administered in controlled settings, they are very safe and non-addictive. So of course, we're going to start wondering, well, how come they get to be so special? And one of the original theories, things got messed up in this computer, so I apologize, things look a bit weird. But one of the original theories that have been developed by Robin Carhart-Harris and David Nutt was that psychedelic-assisted therapy opens up this window of enhanced plasticity and environmental sensitivity such that significant therapeutic work can be done and you can have lasting mood and behavioral changes. And this process does not just occur in diseased populations, but in healthy individuals as well. Plasticity, which we define as capacity for change at the level of the brain, can be considered from just a single neuron to whole brain regions, to functional networks between brain regions, which we just heard about now, and finally at the level of cognition and behavior. We have growing evidence that there is a wealth of cellular plasticity effects happening due to psychedelics. So within just, most notably in that first week after drug treatment, we have a rapid growth of new dendritic branches, new spines. These spines do form functional synapses such that there is a consequence to them, both amplitude and frequency of spontaneous and miniature excitatory currents of neurons are increased in that same time frame. And these changes can persist up to 1 month after just a single dose of the drug. So naturally, we can predict that there might be a snowball effect, that these cellular plasticity effects will result in correlated simultaneous facilitation of higher level plasticity changes as well. And that's where we come in to try and see a proof of principle working in a healthy mouse model. Can a single moderate dose, and here we're using a synthetic drug called DOI, can we see evidence of this environmental sensitivity that should follow enhanced plasticity? Can we see enhanced cognitive flexibility? And also, can we find evidence of regional volume changes in the brain, at least at the level of brain matter, which should be, again, followed by these cellular effects? And for this, we use a triangulation of different methods of behavior, pharmacology, and imaging. Jumping right into our experiment on environmental sensitivity, what we did is that we habituated one subset of animals to a recording chamber prior to drug injection. So to them, their environment was familiar, while the other animals only saw that their recording chamber on the day of injection. So to them, it was a novel environment. And we gave them that single dose of DOI. And in the first 30 minutes after the drug, we were first looking at these psychedelic-like responses in mice, which are called head twitches. So it's just a rotational movement of the head. And these are part of the normal repertoire for the animals. So they are present at a low baseline level in the vehicle control animals, but are dramatically increased in the presence of DOI. However, more so in those animals injected in a novel environment. We also looked at exploration effects. So this graph shows you the total kind of distance traveled in those same 30 minutes after injection. And you can see that there's no drug-related effects in the animals injected in a novel environment. However, for those injected in a familiar environment, there was a significant reduction in the total amount of distance traveled. We saw the same pattern of results looking at exploration of novel objects. So we just placed two novel objects in the recording chamber for about 15 minutes and see if, an animal even cared while they were on the drug. And so the same pattern as with locomotion, there was no drug-related difference in the novel environment, but a significant reduction in the exploration of novel objects in the familiar environment. So together, what this data showed us is that novelty enhanced sensitivity to the psychedelic effects, but familiarity enhanced sensitivity to exploration effects of the drugs. This was an important validation that we can find evidence of environmental sensitivity in animals as well as in humans. Next, we looked at cognitive flexibility. And for this, we used a two-step decision-making task that, as the name suggests, requires the mouse to first initiate the task at a central poke. They're doing this in an operant chamber. And then it has to make a choice between going left or going right. And depending on that choice, one of the top or bottom states is going to light up and deliver a water reward. These are water-deprived animals, so they're very keen to find that water. And the caveat is that there is a probabilistic structure to this task. So the water reward is present at a higher 80% chance only at one of these top or bottom pokes. And the transition between that step one, going left and right, and step two, going top or bottom, is also probabilistic, such that, for example, in one state of the task, left choice will most commonly take you to the up state 80% of the time, and the reverse being true for a right choice. So if you were an animal and wanted to get water, the correct choice would be to pick left, because that will most commonly take you to the up state, and that's where you will most often find the water. The further complication of the task is that these probabilities can change. So the first thing that can change is the reward probabilities. Instead of being more rewarded at the top poke, you will now be more rewarded at the down poke. And these are actually learned by the animals throughout training and throughout testing. They are surprisingly very good at these, and a superstar mouse can do about 20 reward reversals in 90 minutes. So they are very good at adapting to them, and that's the first thing that we're going to look at first at baseline, after the mice have been fully trained up on the task without any manipulation at this point. This graph shows you the fraction of choices the animal is making to the pre-reversal correct side. So before a reversal happens in the gray shaded area, you can see that the animal is picking the correct side almost all of the time. It has learned what's the right way to go. But after a reward reversal happens, it will switch away slowly and start picking the opposite side more often. So even though the legend here says that this is a comparison between two treatment groups, keep in mind they haven't actually received the drug at this point. So this is just to show that animals are consistent across each other before we give the drug. However, when we do give the drug in that first week after, there is a small but statistically reliable difference such that the DOI-treated animals, the shape of their adaptation curve has changed. And we have confirmed this analysis with also utilizing a double exponential model fitted to these curves and then comparing the time constants. So what this means is that basically the shape of the curve has changed such that the DOI-treated animals are ever so slightly quicker in the first 5 to 7 trials. So keep in mind, we are very close to ceiling here. As I said, animals are very good at this task. But we do have some inclination to see that they've become slightly better at adapting to these reversals. More interesting thing happens after a reversal in transition probabilities. So instead of right taking us to top, the right will now take us to a bottom poke. And you can imagine that this is a much bigger shift in task structure and is much harder for the animals to adapt to, especially because these are novel. So they haven't actually experienced these reversals until my omnipotent hand does it to them. So we initiate a single transition reversal that 1 week post-drug, and then monitor how the animals relearn the task in the next 2 weeks. And to look at adaptability in these reversals, we utilize a logistic regression model of state switch behavior. And I'm not going into any detail of this math, so do not worry if you're not familiar with logistic regression models. The only thing you need to know about this graph is that it shows you a regression coefficient for something that we call an interaction between a reward and transition. So if the value of this regression coefficient is strongly positive, it means that the animal has built an internal model of the task such that it's not just simply repeating rewarded trials. It actually knows that it should repeat trials only if they were preceded by a common transition, because those are the ones that are going to give it reward most often in the future, versus those that have been rare transitions. And indeed, that's how the animals solve the task. They have a strong positive value on this coefficient. However, after a transition reversal occurs, the sign of this coefficient is opposite, just because the animal seems to be following rare transitions. But that's because what now is rare used to be common, and the animal is learning that the change has happened. And as they do that over time, they will go back to having a strongly positive value. However, the DOI-treated animals seem to do this significantly quicker than the vehicle-treated animals. So they were better at switching away, flipping their internal model. So what this data set has showed us is that DOI made animals quicker to relearn rules following both novel and learned reversals. And I'm not going into any detail of this data now, but we do have some inclination about how come they get to be quicker. And we think it's because they've started incorporating learning, not just from rewards, but reward emissions, something that mice don't normally do. So I'm happy to talk about that later if anyone's interested. But this does not necessarily impact the amount of learning. So in terms of accuracy, number of rewards, these treatment groups have actually been comparable. In the last few minutes, I just want to talk about the exciting data we have on the structural changes across the brain, because as you will know, these are very limited when it comes to animals. So in this first study, we took out the brains the next day after drug treatment and did an ex vivo MRI study, which was kind of explorative, unbiased across the whole brain. And we did see several areas that had significant volume increases. So they were all volume increases, which we would predict from those cellular plasticity changes. And they were mostly in sensory areas, such as the auditory and somatosensory cortex association areas. And the strongest changes we've seen were in the primary visual cortex. So we know that 24 hours after, there are increased volume of sensory areas. We're currently analyzing a study that is on brains that were taken 3 weeks after DOI, and we do not seem to find any volume differences there. So it's either that these changes have normalized by this point in time, or they have diminished to the point where we're no longer sensitive with whole brain MRI to find them. Nonetheless, our data as a whole shows us that a single moderate dose of DOI can have long-lasting effects on both brain and behavior. These are likely very time-dependent, very task-dependent, also probably dose and drug-dependent, but I'm sure our next speaker will actually be talking about that a lot more. So for me, thank you very much for your attention, and these are other people that have made this work possible.
Time for 1 or 2 questions.
If you just a little bit more about these two terms, you said you had a theory about what was underlying the changes.
Yes, so the data that I shown at the graph about the regression coefficient that looks at transition outcome interaction, there the regression coefficient involves both rewards and reward emissions. But we know that there is asymmetry in learning such that mice just pay attention to rewards and don't care about reward emissions. So when we built a different model where we actually split that regression coefficient to code separately for rewards and reward emissions, what we found is that it is a non-significant predictor for choice behavior in vehicle-treated animals and for DOI-treated animals at baseline and throughout the task. But after this transition reversal, in those last few sessions where we do see that they're becoming quicker at relearning, we also see that reward emission predictor becomes significant. So they've actually started incorporating learning from reward emissions too, such that they're switching away from reward emissions that were preceded by a rare transition. So they've actually have built a richer model of the task compared to the vehicle animals. Does that help answer? Yep.
I was just thinking ahead to one of the talks later on, psychedelic sociality. What would you anticipate would happen if what was interesting in terms of novelty or familiarity in the environment was actually like a non-engagement or like a sense of occasionation?
So what we think happens with the novelty familiarity is that in a familiar cage, the animal was able to not have that split attention to having to explore the cage, having to figure out what's happening around them, and was actually able to focus on the internal. We have no data to support this, so this is just a hypothesis. And sadly, the social studies on how do animals or humans respond to psychedelics in a social environment are very limited. But there will probably be a difference in terms of if you were with a novel cage mate or with a familiar cage mate. I think aggression might be a troubling variable to have to control in those cases. But yeah, I hope that kind of answers the question. Thank you very much. Do I need to do anything?
Oh, yeah, no, go ahead. Thank you, everybody. We're now going to hear from Meg Spriggs, who's going to be giving a lightning talk about indexing neuroplasticity in neural research. After Meg's talk ends at 12, we're going to be having lunch in the canteen upstairs. So a reminder to access the canteen through the back doors of this lecture theater. And then we'll be shifting to LeGros Clark Theater for all the subsequent presentations. And we'll be convening outside of this building, right outside the lobby at 12:50 to bring people to that theater. Thank you.
If you want to make
No, that's cool. All right. Okay, thank you very much. And thank you for the talks this morning. They've lined up what I'm going to talk about really nicely. Today, I'm Dr. Meg Spriggs. I'm a postdoc at the Center for Psychedelic Research at Imperial. And today I'm going to give a little brief insight into how I think we can use electrophysiological markers of neuroplasticity as a novel approach for psychedelics in humans. We just heard a fantastic talk about how rodent models are being used to demonstrate plasticity mechanisms in relation to psychedelics. And this underlies our kind of understanding of how we can have these single high-dose psychedelic experiences that can lead to these long-lasting changes. However, one of the things that I think is currently missing from our sort of bridging between these animal models and these long-term therapeutic changes is how do we measure these plasticity processes in humans? How do we measure them during the psychedelic experience and after the psychedelic experience in the human brain? If we can better measure these things in humans, then we can better develop our psychedelic-assisted therapy processes and tailor it to different drugs and to different indications, so we can really harness this capacity. And also, being able to measure plasticity processes in the human brain allows us to better sort of get to grips with this plasticity process between the acute hyperplastic state that psychedelics induce to the short-term plasticity window that we just heard about, where therapeutic intervention can happen, and then into the long-term sort of changes. So there's a bit of a step, or we haven't yet really explored that step between those short-term therapeutic interventions and how that translates to the long-term therapeutic benefits that we see months after a psychedelic experience. And today, I hope that I can kind of convince you that EEG provides us with this unique window for exploring these plasticity mechanisms. And for those of you who are unfamiliar, EEG is the process whereby we put electrodes onto the scalp and we measure the brain's electrical activity. It's a very old neuroimaging technique. It's often forgotten about as it's surpassed by its more technologically advanced cousins, MRI and PET and things. But I hope that today I can convince you that EEG is still a very relevant tool for us to be using in neuroscience. And we've already heard from Morten this morning, about the sort of predictive processing models of brain function. So I don't need to spend too long on my little sort of toy animation of demonstrating this. But basically the idea being that, our brain builds models of the environment. And these models of the environment are shaped by our experience and they shape our ongoing experience. So we hold these prior beliefs about the world and they sort of are held in these higher order cortical areas and they trickle down the processing hierarchy and shape our experience of information as it comes in to the brain as we move around the world. So what's actually sent up that cortical processing hierarchy is the error between what we predict and what we perceive. And then that error signal gets sort of used by our brain to update our models of the environment. And I'd say there's sort of three key steps in this process. The first is error detection, which is that error signal that I was talking about. The next is our ability to use that information to update our models of the environment, which can happen in the short term, perceptual learning. But then there's this process of moving that into long-term model calibration. So transferring that short-term learning into long-term therapeutic change. And we have these two really unique EEG indices that potentially allow us to start unpacking this. So the first is what's called the roving mismatch negativity. And this is where we present participants with sequences of tones. And every so often that sequence changes tone frequency. Now, every time the tone frequency changes, the brain gives off this error response, this surprise response, this, I wasn't expecting that response. And that looks a little bit like this. So that big blue negative going ERP or event-related component is referred to as the deviant response. That's the mismatch response, and that's your brain's surprise response. Now, over subsequent repetitions of the same tone, that surprise response returns to a baseline or standard response. And that's that process of model updating. So we've got these two processes that can be measured with the roving mismatch negativity. Then we have the process whereby that short-term learning needs to be translated into long-term learning. And here we have the visual long-term potentiation paradigm. So long-term potentiation is the process, the cellular process, whereby it's the idea that cells that fire together wire together, and how we lay down memory formation within the brain. Using the visual long-term potentiation paradigm, what we do is we present participants with visual stimuli, in this case, vertical and horizontal sign gratings, both before and after what's referred to as a high-frequency visual tetanus. And that tetanus is high-frequency stimulation. And this is based on what they do in rodent models where they use electrical high-frequency stimulation to induce the process of LTP. We're doing it with visual stimulation. And typically what we see following that high-frequency visual tetanus is an enhancement of the N1 and P2 components of the visually evoked response over the occipital cortex. And this is what we believe is representative of an enhancement of synaptic efficacy, thus indicating that the process of sort of LTP has started. So here we've got these sort of these two paradigms to look at these three steps, or the three steps that I've outlined. So error detection with the mismatch negativity, the roving mismatch negativity with model updating, so repetition suppression, and in that long-term calibration. So what evidence do we have for this already? I'm going to talk through two sets of results that start to show this in the acute and post-acute states in psychedelics. So this is some work that Chris Timmerman and I analyzed a few years ago now, where we looked at data collected from the mismatch negativity in the acute LSD state. So 20 participants were presented with the mismatched negativity in 2 conditions. The first was placebo and the second was LSD. So in the placebo condition, we did see the classic mismatched negativity response, which is that the solid blue line compared to a standard response, which is the dotted blue line. In the LSD condition, however, there was a reduction in the magnitude of the MMN, but an increase in the magnitude of the standard response. So what this demonstrates is there's less of a distinction between what would usually induce a surprise response and what's usually considered standard. So there's less of an error signal, and this corresponds really nicely with what we understand about the acute psychedelic state, where people are more open and less rigid, and sort of opening up that window for the capacity to experience things in a new way. And then we have this very new data looking at the post-acute phase using the visual LTP paradigm with psilocybin for the first time. So in this study, we have data from 16 participants who were given a low dose or a high dose of psilocybin. And approximately 5 hours after psilocybin administration, so this is post-acute, in the high dose condition, there was a greater potentiation of the N1 component of the visually evoked potential. So this demonstrates that using the LTP paradigm, we found evidence for an increased capacity to undergo the beginnings of that that transitional process from experience-dependent learning to long-term change. So that's just 2 examples of how we can start to use these paradigms. There's lots of open questions remaining around this, and I think that there's a long way to go in understanding this plasticity process. But I hope that I've kind of demonstrated how we can use EEG to start looking at that process from the acute hyperplastic state to short-term plasticity to long-term therapeutic change, and start to lift that black box that we've got over plasticity mechanisms in the human brain. And I'll just finish on this quote here. I hope that I've convinced you today that EEG is not only a brain imaging tool for the poor, but it's actually the ultimate brain imaging tool for those who are interested in temporal dynamics of large-scale brain networks in real-life situations. And on top of that, I would add possibly one of our unique tools for understanding plasticity as well. So just a huge thanks to these people here, particularly those in the red boxes, as they contributed massively to the work that I've presented today. And yeah, I've left lots of time for questions, so shoot.
Time for 2 questions. Hi, so I'm wondering if you could speak a little bit to whether you're aware of any evidence, or whether it's something that you're working on about kind of more direct evidence linking to the metrics mentioned of the mismatched negativity and the visual evoked potentials to something like the brain driving factor, or some like kind of standard markers of neurogenesis or synaptic plasticity. So is there kind of any validation of that?
Yeah, that's a good question. So in terms of specifically with BDNF, we do have some data where we looked at the BDNF val net polymorphism, and we found that LTP was greater in visual LTP was greater in those who carried the who were val-vals, so people who have higher synthesis of BDNF, and that was also related to memory performance. The visual LTP paradigm and the mismatched negativity have also been studied in rodents where so in the example of the visual mismatched negativity paradigm, visual LTP paradigm, where things like NMDA agonists and antagonists have been used, so we know it's related to like glutamatergic functioning. So there is some translational evidence there as well. Yeah. Does that kind of answer your question? So there is, we have looked at like visual LTP and memory performance, but not any specific cognitive tasks. So that's a really, that I can think of. That is something that we will be exploring though. But yeah, that's an interesting question. Yeah. Yeah. So in the psilocybin data, these were people who had not taken a psychedelic before. Bad experiences. So when they come in for our studies, whether they have bad experiences, we have people who have more challenging experiences, but it's, you know, in the psilocybin work, they're always in a therapeutic environment. So we sort of hold that experience with them. Great, sandwich time?
Great, so yeah, we'll be having lunch now. Sam, wave your hand, Sam. Will be up there to direct people towards the canteen.
Basically dies as basically stories that yeah, basically scared. Yeah, so I actually, I don't want to steal you, but
And then if we can just log in, click on the YouTube link and see whether it's still live. You sent it to me. You sent it to me on yeah. So we are live.
Okay, nice. Great.
Cool. Oh, we have a lot of questions on this, which we haven't. I can't really address this. So, okay, so you've already downloaded all of them, have you? Okay, so they will be in the downloads folder, and you want to have this.
And does this also charge your laptop or not?
Doesn't seem like it, but
yeah,
I think I do have.
we should keep it just because we're going to be using this until 6 p.m.
True. Yeah.
If you, I would charge her.
Yeah, please do. Yeah, you can connect it to you. Does it have a? Yeah, it does have a power. Ah, great. So it is. Nice. Is the audio going?
Okay, yeah. Hi. What was it? No, this is the smaller one. Oh, okay. Last one.
Okay, so let's quickly.
Let's go. Yep.
These more.
You might
put them in the room. Yeah, here we go.
Yeah, still free to chill out, like Yep.
Yeah. I think the word I was looking for is terrifying, but that's separate.
Terrifying from a COVID perspective. You have to basically put a dead corpse right there. And then
Well, I can pretend to be Zoom frozen. You know, I'm sure it's gonna go well, right?
So Andrea.
Wait, this is not live, right? Is it?
It is live.
The people online are seeing me being saying
But there aren't
stupid
that
things.
many. Yeah.
Okay, all right. Smile.
I don't think there are that many people online at the moment because that one is when
Alright.
it started. So here's your presentation. So let's just enter. I don't want this. So let's enter the presenter mode.
Is it being recorded as well as being live?
Yes, but it's gonna be cut.
Okay,
It's gonna be, yeah, don't worry. No, I will only put the. So here's your presentation. So the one thing that I want to make sure is that what people see is actually your presentation here and not the presented link. I'm just gonna check on this.
I'm gonna do my stuff.
Yeah, no problem.
I think so, yes. Well, I was telling you about the things, but I think they know. And the book, I want to know about the.
So this is where you got your presentation? Then
which one?
Both.
You want the converter?
I mean, I got here the. You can put the USB one here. Yeah.
Will I be able? Is this the view that I will have?
Yes.
Okay, great. Because in that case, we can
they're not
copy.
seeing.
No, they're seeing that.
Okay, excellent. Sorry, you finish, and then I'll
just
Which
one
slide is
thing
your video on?
Sorry, that's the cursor. How does?
You can just.
Oh, okay. I'm not used to this fancy technology.
Is it this one?
So this, yeah, and then it should go to the next, and the question is whether it's gonna. Is this for showing next? Okay, yeah. So that looks good. I don't know if you're gonna be able to hear the sound.
Oh, the sound of the video.
Yeah, video comes with sound as well. That's pretty. It's just vibrations. There's nobody talking.
Okay, so online they will now here. That's a good question.
okay, yeah. I always go to this Kindle school lectures.
So just maybe try playing the video.
Try again. It's not. Yeah, we're missing the sound.
Speakers and now how to access them is the question.
And now spoilers are being had.
I am selecting the audio.
How will the rest find this place?
I think this kind of is
there's going to be
taking the others.
I want my audience.
Well, I don't want to. You can keep the audience. How about that?
I think it's going to be an Andrea afternoon because there's a lot of Andrea in my talk. So.
Oh. Yeah. Delighted. Terrified. You can choose.
Yeah.
I think it's
By the way, if you want to work with this coaching guy, don't. How will the rest find this place?
I think this kind of is
there's going to be
taking the others.
wait.
When you start pressing. I don't want to. You can keep the audience. How about that?
I think it's going to be an Andrea afternoon because there's a lot of.
Yeah, I'm vaguely afraid of that. I think this kind of is
there's going to be
taking the others. When you start pressing. I don't want to. You can keep the audience. How about that?
I think it's going to be an Andrea afternoon because there's a lot of.
Yeah, I'm vaguely afraid of that. Remember your life.
Oh, am I?
Yep.
Crazy. Maybe I should do an interpretation.
Yeah, you can. You can get people to clap and sing.
I love this. It's so extraordinary. I didn't think that things like this existed.
Yeah, I really like this when I was an.
You came here for.
Yeah, I remember freestyling in the
This is the PDN kind of department.
Exactly, yeah, department. Like this is physiology, nothing in genetics. They always lump together a lot of things. These kind of departments.
Yeah, I think unfortunately I can't get
No
the
sound?
audio to work.
Okay.
Are you staying, Andrea? Are you going back tomorrow?
I'm going back tomorrow. Tomorrow morning.
In the meantime, we'll find yours.
It can work from there too, whatever.
Is it the PDF or?
No, the reason I asked is because I've got the notes on the PowerPoint, so
No
it's
problem.
better.
I'll just.
All right.
Okay. It's quite substantial.
I see. Maybe that's why. Let me copy if it allows it.
I mean, we can leave it there too, it's fine.
But just in case if there's some malfunction, I'll be able to find it here easily. Okay, so you will. Your name again is
Emmanuel.
Okay, so you're presenting right now.
Yeah,
Okay,
yeah.
fantastic. Perfect. So I'll actually open yours up so that we have that. This I don't need. This we want now. Okay. I'll start it for you.
Shall we check this?
Yeah, we can check it right now. So do you want to just go through them, or do you want to?
They're fine. I was just looking at that. There are some notes in. Oh, yeah, they are.
So they will appear when we do the slideshow and we do. We can start. This is what you see, and there is what we.
That's the most important for me. I'm useless with keeping time.
And here.
Yeah, and that's important. The next slide is really important.
So maybe this is annoying for you. So we
it doesn't matter so much. It's fine. Yeah, it's fine. It's just seeing what comes next.
Yep. And so the people online will be seeing whatever.
They will see that, or they will see this.
They will see that, but they will see you through the camera. So
Okay.
This is, yeah.
Okay.
I'm just gonna put this. So
they will not see the notes.
No, the people online shouldn't be able. Or
that's silly. What spelling mistakes?
Yeah, I'm pretty sure they won't be able because I'm sharing the second.
That's fine. That's fine.
So
yeah.
In the beginning, there we go.
And oops. What is it that you did?
What are you looking for?
Oh, I was trying to take this out.
Oh, take it out. Yeah.
Because you're working from.
Yeah, now it's on the.
Great. I like how we make computers smaller and everything, and then we have to keep all this.
All these adapters.
I have a bag full of adapters. Of course, there's quite a few people.
Yeah.
Oh, yeah.
Yeah, okay. So this is what's being seen online.
That's fantastic. Really.
Okay, I think we should be can we do them maybe a bit different?
So the only thing I can do is I can either select those slides or I can select these slides. Currently I can select these ones so that they don't.
Okay. Let's have them on. Yeah, that's better. Otherwise, so maybe start like 3 minutes late just to give people time to settle in. Okay.
I don't think I will take an hour. So, I will I will stop this.
Sure, you can close that.
So I know, yeah.
And yeah, just to change slides.
Yeah, okay. Oh, is that the reset? Gosh, a large audience. I hope they got that one.
What's your pointer?
Yes.
Let's have a look. We can get that.
Oh yeah. That's fantastic. Yeah.
There's another speaker who may want to use a USB stick.
They can give it to me now.
Yeah, I'm just trying to look for him. I don't know where he is. Carl Smith is the guy.
Not Andrea.
Not Andrea.
We did his.
The other one is there.
Oh, there's Carl. Carl, we try the USB thing. Yeah. We'll be starting in a few minutes.
Plug the USB stick in there.
Yeah.
And then. Actually, we can take it there, and then I'll just take it there.
No, it's not the final one. I just wanted to test to see if the sound works and the formatting's
okay.
okay.
I see.
If that's okay.
Okay.
That's the one. Should be.
So unfortunately, sound I can't get it through.
Okay. Oh, I need sound.
You need it.
Yeah, it's really important.
Tried it.
I really need sound. It's really important.
Laptop audio.
Otherwise, I would have brought a speaker. You said it wasn't. Unless we can get a Bluetooth speaker or something.
I was trying earlier.
Yeah, it's really critical to the talk to have sound. Yeah.
Okay. So then we might start a bit later before I get this sorted. And how do I get the sound?
I'll play. Oh,
so try. Just try playing it and let's see.
Okay, it should be playing. So here we go.
Yeah, so I selected the HDMI.
Yeah. So let's just see if it plays anyway through here. Just grab my headphones.
Look, it's set up here for some reason.
Yeah, so it works through here.
Okay, so if we go back to the sound setting.
I mean, otherwise we could just grab some Bluetooth speakers.
If it's crucial.
Yeah, it really
there are
is.
speakers here, but I can't get the sound to go through the HDMI.
Let's try together. So is it something to do with? Have you tried?
So yeah, so I selected laptop audio.
Yeah.
Which is what it tells you to do.
Right.
Control audio is the plus or minus. So
yeah.
I'm sure volume is not on mute.
It's definitely not on mute. It's coming through here. I can see it.
Exactly, it's there. It says.
Do we have anyone else technical?
I see, because I do have somebody with a speaker.
Yeah, that's a good backup if this is going to be an issue. Does that need to be plugged in? possibly. Let's try. Oh, that looks interesting.
It says, I think it's this, right?
So where does this lead? Into here. Let me just try sound again. Let me try the video.
Let me just press play there.
Yeah. Yeah, so it's playing. You can see it there.
See how wonderful Marcelo.
Right, just on PC. It's all right, we got it.
He would have said.
Hey, so we'll keep that plugged in. I guess that's maybe.
That's what it is.
Yeah.
Oh, fantastic.
Oh, that's
Okay,
great. So I'm going to delete this one because I'll just. Yeah, I'm still completing it. So.
It's gone back to laptop now.
Say again.
It's gone back to laptop.
Yeah, no, I just turned it back so it was really loud. But I think we've got it now. So.
Sorry with the name.
Yeah.
Are we all good, Carl?
Yeah.
I'll just okay, and then
And we have a pointer.
The microphone is the
And the pointer is.
But is there a microphone for
yes, the pointer.
Is there a microphone for the room?
For the room? No, but
Yeah,
I think
okay,
everybody can hear it. Yeah. Oh, please, that's. Let me just check that the live stream. I'm just going to check that the live stream is
All right, everybody. We're going to get started again. The first talk in this section is by Emmanuel Stamatakis, and he's going to be talking about the dynamic neural processes underlying psychedelic-induced experiences. Thank you.
Thank you. Is there... Oh, there is microphone. Can everybody hear me? Yeah, good, excellent. Okay, good afternoon, everybody. Thank you for the invitation. Speak up?
Yeah.
Speak up, okay. I'll try. Can you hear me at the back? Excellent, fantastic, thank you. All right, so thank you for the invitation. I want to take a couple of slides to introduce myself, introduce the general context of the work, and then I'm going to talk a little bit about the work we do in unconsciousness, and I'll explain what that means, and sort of try to explain how we got into the psychedelics work from unconsciousness. So for about 10 minutes, I will run the two in parallel, and then we'll go properly into psychedelics. So my name is Emmanuel Stamatakis, as you already know. So let's start with telling you what I'm interested in, and the kind of questions that I deal with mostly revolve around brain injury, especially severe brain injury. So severe brain injury that leads to unconsciousness. So people in coma or what we call minimally conscious state. And we try to understand what happens to the brain of those people and why they cannot wake up. And at the same time, we run experiments with anesthetic drugs like propofol, which are routinely used in operations every day in hospitals. So we make people unconscious. We have people who are unconscious because of an illness. And we look at what happens to brains with psychedelics too. So a couple of very, very basic slides on what I mean when I talk about connectivity. So the tool of my research is an MRI scanner, and with an MRI, especially, sorry, I'm trying to start the timing of this so I know how far I'm going to talk. So what do we do with an MRI scanner? We take scans of roughly how oxygen moves around in the brain. And we put all those scans together. So we designed experiments like this very simple one here. So we have an on and off condition, an on and off condition. So a person in the scanner does this for about 30 seconds, and then they stop doing it for 30 seconds, and then we subtract the two, and we get these colorful pictures that you see here. So these parts of the brain, primary motor areas, motor areas, responded to when we were doing this, this action here. And beyond that, we can ask more complicated questions, such as which parts of the brain communicate with other parts of the brain while we're doing this, while we're doing something, we're engaging with the stimulus. So that's what we call connectivity. And connectivity questions don't have to be one-to-one or one to other parts of the brain. They can also be answered by building matrices like this, where we look simultaneously what happens, how each part of the brain connects to every other part of the brain. So we can build matrices like this. These are correlations, basically, between two time series that we extracted from two different parts of the brain. We can use different atlases to break the to separate parts of the brain. And we then, after we build these matrices, we can calculate properties, and we'll talk a little bit more about these properties and how useful they are a little further down the talk. Now, a lot of the work that has been done with fMRI concerns tasks, putting people in the scanner and asking them to do something. However, 15 years ago, we discovered a little bit more than that, actually. We discovered that even when we asked someone, a volunteer or a patient, to lie in the scanner for about 5 minutes to half an hour, we see these patterns that you see again in the colorful blobs there. I say blobs because Morten said blobology earlier, and this is what he meant. So we see these patterns that we call networks. So in other words, all the areas that you see here co-activate when we are in the scanner doing nothing for 5 minutes. So they fluctuate in activity, they co-fluctuate in activity. And we gave them names because we know these posterior parts of the brain in the occipital cortex have to do process visual stimuli, the same for auditory stimuli, executive function here, and so on and so forth. And we can see, I mean, the reason these things are have become so popular is because they tell us something about brain architecture, how the brain is organized. And they also very, very useful when we have patients that they cannot do tasks. Not every patient you can put in a scanner and say, can you move your, can you make your index finger to touch your thumb? Or can you engage with these regular and irregular verbs and press a button as a response? So resting state networks, which are acquired from resting scans, can convey a lot of information. And a lot of the talk that I'm going to do today has to do with resting state networks. But to try and persuade you a little bit further that they are useful, I'm showing you here somebody again doing this task. And this is, if we ask the question with what we call a connectivity analysis, tell me which parts of the brain relate to M1, primary motor area, while you're doing this task. The reddish areas are what you're going to get as an answer. And we can ask the same question when the person is not doing anything, and the orange areas there is what you're going to get as an answer. So you see there's a huge overlap. The areas engage more when you're actually doing the task than when you're not doing the task, but there's still the same network is there. And to persuade you even further, well, not we, but Cole and Bassett put together 7 tasks. And so here we see a matrix that's been constructed from putting 7 tasks together and 56 minutes of resting state. So you can see they're not identical, but they're very, very similar. In fact, they correlate by 0.9%. Now, when this whole discussion about resting network started, there was one network that we had not encountered before. And this was called, then the name was given the default mode network. And we had not seen this network before. And we noted that it is very, very prominent in this resting condition. So of course the big question was, what does it do? Does it do these parts of the brain that you see down here and up here, are they important for thinking what you're going to shop from the supermarket after you finish with your scanning? Or are they important for planning your future? Are they important for thinking about your past? All of the above, it seems, but some people suggested that that's the only thing they do. That's the only thing they do. That's, you have half of your brain which you use to basically ruminate and plan and such stuff. And we published a lot of work to show that is not the case. How can you interpret, how can you be conscious if you don't have all the information that you have learned so far? If you have all your knowledge disappears, can you be conscious? So, I mean, we kind of published a few papers, but this picture is a good illustration of how the DMN, which is, so the default mode network you can see down here, and each of these strands represents how a part of the brain relates to another part of the brain. And here you have a working memory task that gets increasingly more difficult, so very, very difficult at the end here, where you have to remember, you see single characters on the screen and you have to remember what 3 characters back, whether 3 characters back is the same as the one you're seeing now. And you can see that the more difficult the task becomes, the more you get these strands of the DMN relating to these other frontal parietal areas that are essential for the task. And we took this a little bit further, and we showed that the posterior part of the DMN here, and this is work that Diane Liu did, the posterior part of the DMN here is very important for integrating, for bringing information together. And all of this, if you believe in models such as predictive coding, you understand the usefulness of the default mode network and how it facilitates integrating information. And Diane showed that actually the posterior part of the DMN here is important in deciding whether you want to access more information from the environment or whether you want to access stored information in order to interpret what's happening from moment to moment. And this relationship changes depending on how familiar you are with your environment. Now, the other thing that I quickly skipped is a German psychiatrist called Georg Northoff is interested very much in the self and the idea of the self and how is that represented in the brain. So I'll give you an example of an experiment that can involve showing the person in the scanner a picture of a toothbrush, and you can ask them, think you're doing something with this toothbrush, and think your best friend is doing something with this toothbrush, and think the queen is doing something with the toothbrush. So you have degrees of separation. Now, the closer you get to yourself, the more these medial areas that are marked here with these dots and colorful shapes are involved. So each of these, the experiment I gave is an example, but each of these is a different experiment that activates those medial areas that heavily overlap with this default mode network when you do tasks that relate to yourself. The other reason why we got quite interested in the default mode network is because it has been written, there are numerous papers now that talk about involvement in neuropsychiatric and neurodegenerative diseases. So you have autism, hyperactive medial frontal cortex, prefrontal cortex. You have amyloid plaque deposition overlapping heavily with where the default mode network is. You have implications in schizophrenia. You have the default mode network being hyper-connected when you have depression. You have the default mode network being connected in lower than normal in PTSD. And both PTSD and depression are really important to us from a traumatic brain injury perspective, because a lot of those patients suffer from those conditions after after their injury. So Ioannis Papas did an experiment where he used a model where a hidden Markov model, in fact, and this allowed you to split the brain into states. You can define them any which way you want. In this case, he used the principal component analysis, and he found that these two states relate to depression score. Now, these two states are not identical to the default mode network. However, they relate heavily to areas that we call default mode network. And what he found is that going along with this idea of rumination in depression and the inability to get out of that state, he found that in terms of probability, you switch between those two states most frequently, and it's kind of very, very difficult to get out of that. And these big arrows, these thick arrows signify that it's more likely that you will stay in here forever and ever and ever and ever, and it's more difficult to get out of this situation. The other reason that I was interested in the default mode network is that it has been shown that the more unconscious you become, the more disconnected your default mode network is. So whether you look here at the posterior part of the DMN or the frontal part of the DMN or the parietal, inferior parietal areas of the DMN, you see that as you go from healthy controls, I can't read that actually, can you read that up there? Okay, so you go minimally conscious, a vegetative state, and then you have coma patients. You see in whichever part of the DMN we look, it becomes increasingly more disconnected as you become more unconscious. And theories, now trying to put this with theories of consciousness, such as those proposed by Giulio Tononi, who says that for the brain to be conscious, it's really important to be able to put information together, so integrate information, but also keep information apart, so segregate information. Information that comes from all the sensory cortices have to be kept apart up to a stage where we need to be integrated and construct concepts, if you want. To this end, Andrea, who will speak about another piece of research later, looked at data from people who have received anesthesia and sedation in this case, actually the same drug, propofol, and he looked at people who have disorders of consciousness, so they can't communicate fully, and he did that with an index, by using an index of connectivity. So he looked at every single voxel in the brain and to compute how connected this voxel was to any other voxel. And he also looked at diversity, a proxy for how segregated different voxels in the brain are. And then he overlapped the two, and he found this area in the posterior DMN, in the posterior cingulate, and and precuneus areas. It spans both of these areas. And then he did the same for patients in disorders of consciousness, and he put the two together, and the overlap between everything was in the posterior DMN, telling us that posterior DMN is important for consciousness. Now, if you remember earlier, I talked about constructing more complex relationships than one-to-one relationships, and making up these matrices and calculating properties. And one of those properties that has been discussed a lot in the last 10 years in the context of brain organization is small worldness. Now, small worldness means an arrangement like this, so densely connected with each other's neighbors. So each of these could be us in a social network, or could be a part of the brain and its relationship in terms of activity with the rest of the brain. So here we have each of those areas, nodes, are connected strongly on a local level, and but they also collected on a global level with other areas. And the balance between this integration and segregation is called small worldness. It has to do with efficiency of organization, this balance between local and global connectivity. Now, the important thing is that we know the levels of organization, this balance, we know what the value for this balance is in a conscious brain, and we know we can test whether in unconscious people, it goes up or goes down. And Andrea went one step further than this and calculated, given the theories of consciousness and the importance they give to integration and segregation, Andrea calculated two states. They calculated states in the brain that are very integrated, so instances, if you want, out of 5 minutes, that the brain does very highly integrative work, and when it does highly, when it's important, segregation is important. And he found that in unconsciousness, what is really important, the small wellness is reduced when at the periods of integration. So when you're trying to put information together, or in the parts of the brain that information is put together, small wellness decreases with anesthesia when you're unconscious. And I've given you some evidence now about the importance of the posterior medial areas in the brain in terms of consciousness. Now, at the same time that we were doing this work on consciousness, the team at Imperial was doing a lot of interesting work on what psychedelic substances like LSD and psilocybin do to your brain. Well, how they change function and functionality in the brain. And what they found at the same time, and the reason I went all this long introduction about the DMN, is that integrity in the posterior, especially in the posterior parts of the DMN, you can see it here specifically, is reduced with LSD and changes with psilocybin. So we have in the same part of the brain changes that we found that is important for consciousness. And what happens is it changes in the opposite direction from the one that we showed for unconsciousness. So furthermore, remember the picture with lots of dots in the midline of the brain, self-related processing. Furthermore, there is a relationship between the integrity of this posterior part of the default network and the ratings of ego dissolution that people describe after they've taken LSD or psilocybin. Oh, here's a picture that I want. So you can see that in this part of the brain, we have coincidence between LSD, psilocybin, propofol, which is an anesthetic agent, and people with disorders of consciousness. Now, Robin Carhart-Harris at Imperial and all the team there also came up with this idea that has now moved on a little bit, expanded rather, that in order to measure and understand what consciousness is, we need to look at entropy and complexity in the brain. We have already started doing this work in unconsciousness, in patients and healthy people. So we thought, the measurements we're doing were down here in reduced consciousness in a case of, in a system that is subcritical. So we thought, well, what we can do is test our methods initially and see whether we'll get values that are up here. So the way we got into psychedelic research is initially trying to confirm that our methods worked, and these models led us to correct predictions. So here then we have a continuum where we have a critical zone that the brain works in normal everyday kind of way, and then we have higher entropy where we have things like psychedelic states, we have REM sleep, we have psychosis, and we have low entropy where we have anesthesia, we have unconsciousness, we have either psychiatric conditions like OCD and addiction. And Ioannis again was the first person he looked at complexity of connectivity in the brain. And this was never published, but it was kind of the first result from the lab. So I quite like showing it. So he showed that when you look at the complexity of connections, you start with disorders of consciousness down here. So very, very small complexity. We have sedation, where you've given propofol. You have awake, healthy people, and you have psychedelics up here. So you see an increase in complexity, confirming the theoretical models that existing. And of course, we did this with more, in a slightly different way. Theories from physics suggest and have shown that the more near criticality a system is, in this case the brain, the more fractal it becomes. So Thomas suggested, why don't we look at fractal dimension in these brains of these patients who have disorders of consciousness? And the difference here is these people appear to be awake, but they don't really communicate, minimally conscious, and we have also vegetative state patients down here. So you can see as you go from healthy awake people to minimally conscious to vegetative state, the fractal dimension of the networks, the brain networks constructed in the way I talked about earlier, decreases. So the important thing here for us who study brain injury and disorders of consciousness is that this measure seems to be able to differentiate between minimally conscious and vegetative state, which can be quite useful in terms of making clinical decisions. Now, Thomas then went on to do the same thing with people who have taken psychedelics. So here's a brain of an awake person who has taken no drugs whatsoever, whether anesthetic or psychedelics. And here's how connected the brain of a person who has taken LSD. So you can see those matrices I was talking about earlier up here. You can see that this is a lot more complex than this. And if you try to quantify it, I should say here that all of our data on psychedelics have been shared by the Imperial group. When you try to quantify this, you can see that you get a very significant increase in fractal dimension in the brains of people that have taken LSD, and the same with psilocybin, although not as strong. It's still significant, but not as strong. So we have some sort of increased criticality on the brains of people who have taken psychedelics. And if you look, and this was looking at the whole network, and this is when you look at time series from individual parts of the brain, and then you average them, you get exactly the same result, except the psilocybin result is not significant. When we look at complexity, and in this case, Lempel-Zip complexity, at this stage, this kind of measures were used with EEG data, which is richer in terms of temporal resolution. You see that, again, the complexity of brain function increases when you take psychedelics and decreases when you take sedative or anesthetic agents. Now, Andrea came along again and said, I want to extend a little bit the work that I've done on anesthetics. So this time on segregation and integration. So it's a complex slide, I know, but I will direct you to the areas in the red rectangle. So in the first instance, what Andrea looked at was to see how the structural makeup of the brain differs from the functional connectivity in the brain. So you have the scaffolding, you have the white matter, you have the axons that connect different parts of the brain. So you're asking the question, if you construct a network, a matrix like the ones I've showed you, how does it differ from the functional one? So if you put the two together, the data there in terms of anesthesia and unconsciousness was that if you are unconscious, the two look very similar. So you don't have any richness in the functional connectome, as they're called, these matrices. So you have a functional connectome that's very, very similar to the structural connectome when you're unconscious. But again, exactly the opposite thing happens when you have taken psychedelics. So the psychedelics seem to increase the complexity in the brain, and this with very many different ways of showing it. The additional thing that Andrea found was that although in the context of consciousness, we saw that the stage that the integrated state, if you want, in the brain is more relevant. However, in the case of psychedelics, it's where you see all these differences in the segregated states. So there isn't as much, the more important phase in this kind of two phases, it seems to be the segregated part rather than the integrated part, which we saw changes for propofol and people in disorders of consciousness. So we tested with yet another method the complexity. The results remained as they were. So we see increased complexity with psychedelics, and we also showed decreased functional correlations. Now, from the resting state networks that I showed you earlier, one of the big take-home messages was that the brain has this, is permanently, especially at low frequencies, in this anticorrelated state. So if you're resting, if you're urged by a researcher to lie in the scanner and think of nothing in particular, you have the default mode network that is strongly anticorrelated with its executive frontoparietal networks. And we found that when you take propofol to be anesthetized, or when you are a patient with disorders of consciousness, these anticorrelations are reduced. And this time we saw exactly the same thing with LSD. So the anticorrelations, this kind of tension in the brain seems to be reduced when you take LSD. And we calculated small worldness here again, a property of these graphs, and found that it relates the difference in small wellness between a state of being awake and when fully awake without having taken any drugs, and when you take LSD, relates to all these things that people report after they've taken psychedelic drugs. So things like increase in complex imagery, ego dissolution that we already discussed earlier, and this blissful feeling. Now, this work was done, as I said, in a broader context of understanding consciousness and consciousness, understanding whether the methods we developed work in the way we expected them. But at the same time, of course, psychedelics have been enjoying so much attention now because we see them as agents that can help people with psychiatric problems which are sometimes resistant to drugs. And there is, as you can see here, there is increase in the number of registered clinical trials that are happening. And that's the kind of direction that we went after we developed all these measures. So in this particular case, we wanted to bridge data from the brainstem with all these cortical networks. This is all in the context of work that we did, again, with unconsciousness, and found out that dopamine and the dopaminergic system is really, really important when it seems to be disconnecting when you are unconscious. So we try to do the same thing, and as I said, we focused on a part of the brain called the brainstem that you can see here. And Lenny, who's also here in the audience, did this work and looked at two nuclei, two strongly serotonergic nuclei in the brainstem called the raphe nuclei, the medial and the dorsal raphe. And what he found was that when you look at the resting state connectivity of this raphe nuclei, which are major sources of serotonin in the brain, you can see that they relate to that same posterior area of the PMN, the default mode network that we discussed earlier. So how does this relationship go? The more, so we see that if we take it a step further and say, how does this area that we found connect to the rest of the default mode network? we find out that the more connected the median raphe nucleus is to this posterior part of the DMN in the precuneus PCC, the more this area relates to the rest of the DMN. So we see some kind of serotonergic modulation of the DMN by the median raphe in the brainstem. The same was noted for the dorsal raphe, but not to the same extent. So now we go to the really, really interesting and important study that was published last year in the, if I'm not mistaken, 2021 in the New England Journal of Medicine, which showed that if you compare psilocybin treatment with escitalopram treatment, which is a really commonly used SSRI, what you find is that psilocybin may work slightly better. And there was no imaging data that was published in this original study. And we asked the people at Imperial who collected the data if we can use the data and use the same kind of technique that we used to do the discovery about the dopaminergic system in unconsciousness. And we tested all possible nuclei that we have ROIs for in the brainstem, so various neurotransmitter systems. And what we found was that the greater the median raphe, which is shown here in green, connectivity, pre-treatment, so the greater the connectivity, the co-fluctuation between these two areas before treatment, the smaller the depression score is. So if you look at this, so now we're talking about some kind of diagnostic marker of people with depression. So we're saying that the more connected these areas are, so the more connected they are down here, the least depressed the people in this population are, okay? So then Lenny took it one step further and showed that the functional connectivity, so the relationship in terms of activity between these two areas correlates with the difference in depression score. So the more impaired the connectivity before the trial, before you took the drug, the greater the strength of the treatment response. So we're going from diagnosis to prognosis and perhaps treatment monitoring, because the third relationship that we found was that the strength of improvement in QIDS, which is the depression score assessment, was associated with higher median raphe precuneus connectivity. Excuse me. So a little bit of a summary then. A ZZ escaped there somehow. That is the wise part of the summary. So activity, what we find then is that activity in the brain on psychedelics is more complex than normal. We found that LSD makes activity in the brain a lot richer than it was, and it makes it a lot more different than the scaffolding, the matrix that we form by just looking at the scaffolding in the brain, the white matter connections. We found that increased small wellness, a graph property, predicts LSD-induced ego dissolution. And we found that LSD is important for segregated states rather than integrated states. And it has the more effect in those states, that is. And we found that the results in a major depressive disorder cohort provide empirical evidence in favor of psychedelic-induced serotonergic system reset for therapeutic effects. But I mean, take that with the last one, especially with a pinch of salt. A lot more work needs to be done on that front. But thank you for your attention, and thank you to all the people who were involved in this work. Sorry, which one is that? Was it in the propofol or? Oh yeah. Maybe they went as awake as I described. No, I'm sorry, I don't. But yeah, how tight everything is here, and then yeah, that person down there. Maybe they maybe they taken something before they before they came to be scanned. We yeah. I mean, we don't have genetic data on those people, but what we have maps of gene expression from atlases that have been published, and we're looking at that now. But so maybe, I mean, yeah, maybe we're not in a position to provide conclusions about individuals, but we're getting in that direction. Yeah. Sorry, I know I didn't answer your question, but.
Thanks for that. It's been a while since I've had to worry about neurobasis depression. Can you speak a bit more to this remedial method of connectivity with biomarker?
Okay, yeah, of course.
Is it just being presented as sort of definitive phenomenon, or is there evidence for mechanistic?
Well, I mean, yeah. So there is a connection in the literature between this default mode network and depression in general. So although I don't accept this idea of half the brain switching on and switching off when you're thinking and when you're executing a task, I think, well, as a complex kind of spaghetti picture that I showed is that all parts of the brain are involved in all parts of that. But there is a kind of very broad way of thinking that says the more you internalize, the more you focus on the internal, the more depressed you are. Okay, so and that's what that hidden Markov model showed. And I mean, our research and that particular piece of work is not the only one that shows this connection with the DMN and depression. There's a lot, and there is work on this particular group of people where they were depressed following traumatic brain injury, but this kind of pattern is shown in people with clinical depression who haven't had traumatic brain injury. So, I mean, the most common drugs that you give people with depression are SSRIs, right? So we thought, well, and psychedelic drugs are serotonergic drugs, of course, and we thought, of all these tiny nuclei in the brainstem, where can we look? And we looked at all of them, the ones we have ROIs for. There is a group in Harvard that has published a very comprehensive set of brainstem nuclei that relate to arousal systems, actually. And the arousal comes from our angle of unconsciousness. That's why we're interested in those. So we tested all of those, and we thought it was interesting, but the only one we saw this kind of response was the RAFI, especially the median RAFI, which is heavily serotonergic. Is it an epiphenomenon? I mean, we don't know. Our data can talk to that yet. One thing that I can say is that we've replicated it with another data set. So at least it shows us, neither are published yet, but they're written up, so hopefully soon. So hopefully what it shows us is that it's not just a cortical phenomenon, right? And these modulatory relationships from the brainstem are important too. So that's as much as we can say at the moment. Although I'm sure Lenny's looking at me. Is there anything to add?
No, not necessarily. I mean, it's at the core, the idea of what depression might be is one unknown hypothesis, like Matthew said, right? This is serotonin. So I think for a very long time, we thought about depression drugs as things that, like you had today in the morning, have a locking key principle drug, binds the receptor, from there the effect is typically been considered in very cortical terms. Instead, looking at the actual source of serotonin in the brain will give us an idea
idea of why psychedelics sometimes just require 1 or 2 dosages to bring about quite significant therapeutic effect. It seems like this particular relationship, actually modulating the big scheme of the cortex, might be how this comes out. So what you saw in the beginning is basically when someone takes the drug, there's a sort of knockout effect, the connectivity disappears. After they've been treated, it becomes a lot stronger than it was before. And for that reason, there's this idea that potentially it is in a sort of hypo-serotonergic state beforehand. Taking psychedelics, powerfully serotonergic drug, might reset how the serotonin system functions.
Is there anything that's like natural brain solution? Is there anything to suggest that is
Depression?
Yeah.
Well, unfortunately, the brain stimuli for serotonin have basically been ignored for a very long time, other than in animal research, which unfortunately we don't have many sort of behavioral reports. But it's definitely a very, very important question. I agree.
I mean, again, this was research that was sort of, we got into trying to validate what we've done in unconsciousness. I mean, is this the MRI scanners that we use vary in terms of strength of magnetic field and a 3 Tesla, which is it's a common kind of research scanner, MRI scanner. You can't see those nuclei. So there's a lot of people that say, well, this is all rubbish because you can't see the nuclei. But however, I don't accept that if I test all these other nuclei and I don't see any effect, and I see the serotonergic effect for the serotonergic drug, and the same we see an upregulation. If we test this, the ventral tegmental area with the dopaminergic drug, we see it upregulated, and again with the same part of the cortex, posterior default mode network. And it's not a coincidence that you have, this upregulation of dopamine of the default mode network, and you have all this connection with consciousness and psychedelic drugs, especially posterior parts of the DMN. All these things cannot have just happened, right? Sorry, I kind of went a bit fast on that part. So we're talking about 2 kinds of connections and essentially 2 types of MRI imaging. One that is very sensitive to how the brain is actually physically connected, white matter bundles, as we call them. So you can do some complex, actually that particular technique started here in Oxford. And you see how different parts of the brain, so you parcellate the brain to whatever resolution you want, to 5 parts, to 20 parts, to 1,000 parts, and you construct a matrix with the strength of the structural connectivity, of the strength of these bundles, how many lines go through the central point if you want. It's an example of calculating this, if you connect these 2 areas. So that's how you construct the structure, the scaffolding. So the thing that allows you to connect different brain areas. Now, functional connections don't have to be direct from point A to point B. They can be via other points, right? So you can calculate the richness of the structure of the functional connection. Structural connection is rigid. What you see is what you get. That's it. It doesn't change unless you have disease that destroys some of this connection. So what we're comparing here, what we see here is if you're unconscious, the functional connectome and the structural connectome become very similar to each other. So there is no richness in function, right? When you've taken psychedelics, you see they move further apart. They're more different, these structural and functional connectomes. And less correlated. Yeah? Yeah? When you use correlations, there are a lot of other techniques you can use to find the same thing. You get an increase in small wellness as well. Yeah. I forgot to say, a lot of these papers are available here if you want to explore it a little bit more. I'm sorry, I can't. Ah, I see, I see. Well, there is another bunch of evidence that I didn't talk about, but there is, so if we try and localize in the brain, first of all, this is not research that we have done, but if we try and localize in the brain, well, if you want social parts of the brain, the parts of the brain that help us interact with our environment, that allow me to answer these questions now, are heavily overlapping with what we call default mode network. So special areas in the inferior parietal cortex, in the medial prefrontal cortex, all seem to be important for us socializing. There are people in Harvard that believe, there's one particular researcher I have in mind called Rebecca Sachs, who believes the parietal part is more important. There are other people who give more importance to the frontal aspect of the default mode network in terms of being able to socialize. Now, somebody noticed this fact, that those areas overlapped with DMN areas. And I presume at some stage or other, this was a very, very early study. There are a lot more studies since then, which I'm afraid I haven't followed. But they said, let's check this and see whether there is any kind of correspondence. And that's what they found. The frontal aspect of the default mode network, in between the two hemispheres, if you think just about here, was differently connected in people who had autism than their healthy control, age-matched and demographically matched healthy controls. So that's what that slide was said. There's been a lot more research on that one since then, which I haven't followed, I'm afraid, but there's a lot more research out there on that topic.
So we talked a lot about kind of the connection between the research in anesthesia and kind of the source of consciousness and that in psychedelics.
Yeah.
And I was wondering if you could comment on this kind of more recent idea that we may actually use psychedelics directly on people with source of consciousness, kind of bring them back to baseline. There's been a couple of papers on that. Though there seems to be obvious ethical issues. So I'm wondering kind of what your thoughts on that are, whether you have any plans.
Well, it's quite interesting in that you see this continuum, so you think, well, how can I push this person who's down here up here, And I think Andrea has a good anecdote about that, because it was your paper, right? And we got a reviewer saying, you're unethical to suggest, we haven't even suggested it, right? We didn't make a mention of that at all, but they suggested, you're unethical to suggest that you should give people with disorders of course the psychedelic. There is discussion going on. We're not involved in this, as far as I know, but there is discussion about that. And when I said, I didn't show a slide, but I mentioned earlier that we found a strong dopaminergic dysfunction in people with disorders of consciousness. I'm not arrogant enough to suggest that, brain injury is so heterogeneous. I'm not arrogant enough to suggest that that's the only thing that goes wrong in these people with severe injuries, right? Ideally, and one way to go forward with this is to build maybe connectomes for each of the brainstem nuclei, maybe connectomes that are informed by the PET maps that Morten talked about earlier today, and bring or the small wellness or all that, and build a picture of what's wrong with each individual, and then decide on what treatment. Maybe more than one kind of treatment.
Just very briefly about the role of the relationship to people with no effect. I was wondering if anybody would want to perform that.
Yeah. How long do we have? So, the posterior, both of these regions, the medial prefrontal cortex and the posterior parietal cortex, medial, are disproportionately connected to the rest of the brain. And there are areas that evolutionarily have expanded disproportionately more than any areas of the brain. The precuneus and posterior cingulate cortices have strong connections with the hippocampus. So in a way, and please don't quote these exact words that I'm about to utter, but I think, and we've seen all this evidence for integration of information. So I think they are, in a context of a predictive coding model, they provide the priors, right? What we know, what we have experienced. And I think then there are people who believe that there is some kind of comparison in the posterior part of the DMN. I think it's more the kind of comparative mechanisms that sort of update your priors happen more at the frontal cortex. But so you can have this kind of process where you have priors retrieved, and then you have evaluated, updated, priors retrieved again. So you can see how I'm thinking about this. There is a paper actually that is under review at the moment, but we have a preprint in our website, not this one, by Diane. And again, that shows that when you do a binocular rivalry task, the one where you're looking at two stimuli that are different color, and the stimulus doesn't change, however, you only perceive one color, right? And Diane showed in this other paper that uses both MRI and EEG data that were collected at the same time, that you see a signature of the change in the perception of whether you're next going to see red or green, or which stimulus, well, not which color, but the fact that a change is coming up in perception in the posterior part of the DMN. So before this happens, before you press the button to say, I've seen this, my perception change, you see a little signature of that in the posterior part of the deformal network. So all those bits and pieces make me kind of mention the DMN in the context of this predictive coding models. Okay, thank you.
Excellent. Thank you very much, Emmanuel. We're next going to hear a talk from Andrea Luppi. It's going to be a lightning talk, mapping a landscape of consciousness and the dynamics of the human brain. What was that? Oh, that's okay.
Yeah, the sound works now. Yeah.
If I seem to be running out of time,
I'll give you 10 minutes and 5 minutes. Okay. Yeah.
One thing, Andrea, do you prefer the lights up? The lights up. Okay.
All right, let's get started.
Just the outcome.
Just use the arrows, and I think the video starts automatically. So
okay. All right. Hi, everyone. It's a pleasure to be here. So thank you to the organizers for having me. And thank you all for being here after lunch. So I'm Andrea Luppi. And as my title suggests, I am interested in mapping a landscape of consciousness. Now, some people might think that that is almost a paradoxical exercise, because how can you map consciousness? Consciousness is ever fleeting, like the flights and perchings of a bird, in the words of William James. So that is akin to wanting to map the clouds. But I am here to argue that we can do away with the magic, and we have seen some of that already. And I'm going to try to convince you that we can do better than magic. But doing away with magic does not mean doing away with mystery. And the mystery I'm going to talk about here is one that has been touched upon already. And that is, our brain has a fixed structure. Across a few minutes, the structure of the brain is not going to change. And yet, we can do all sorts of things, and we can think all sorts of thoughts. So how does that variety of mental states, of brain states, arise from the same fixed structure of the brain? That is the mystery that I'm going to try and shed a bit of light on today. And of course, talking about mapping structure and function might sound like a very abstract problem. But actually, that is something that we are all very familiar with, or at least all of those among us who have to commute every day to work. Because you can think of the structure of a city as the set of roads that connect different parts of it. And of course, those are fixed. And we know how fixed they can be and how slow it can be to change the layout of a city. And yet, we also know that the traffic can be very variable, and that if you choose wisely the point in time when you commute, your commute can be very different. So the presence of a road between two cities does not necessarily mean that there is going to be a lot of traffic between them. There can be, but that doesn't need to be. So that is a case where function and structure can vary their relationship. And of course, that is one way that we can look at it in the brain. So if you take different parts of the brain, different regions as your cities, then you can think of the white matter connections between them that Emmanuel was describing earlier as the roads between them. And then naturally, the interrelationships between the activities of these regions become your measure of traffic. Now you're wondering, we've just been looking at pretty pictures of sand over a metal plate. I've been talking about traffic. I've been talking about brains. And you're here because you want to listen about psychedelics. Now, how do those things relate to each other? Now, the point that I'm trying to make here is that we can bring all of those together, and the answer is going to be through math. But bear with me. So the reason I'm showing this here now is that I'm hoping you notice that the patterns of sand as that metal plate was made to vibrate were not random. They were pretty, but they were also systematically related to the frequency of the sound that you were hearing, which was the frequency at which the metal plate was vibrating. Why do we care? We care because that is a case where there is a fixed structure, a metal plate, and different functional configurations taking place over it. And that's starting to sound like the problem we wanted to investigate earlier. And it turns out that this is not only very well understood mathematically, it is also very widely applicable. Because this is the same principle that allows humans to make music by taking the bodies of different instruments and making them vibrate at different frequencies. And interestingly enough, if instead of a metal plate or the body of a guitar, you take the structural connectivity of the human brain, what we call the human connectome, then the kind of pictures that you can get are going to look like this. And I'm hoping that you realize the pretty similarity between these sets of pictures, which is not random, but rather is driven by the same kind of math taking place in both instances. And just delving a little bit more into these connectome harmonics, which is so called because these are harmonic modes, the name of the math, pertaining to the human connectome. You can see here that what they are is large-scale whole brain patterns of activation that each of which comes with a specific spatial frequency. And that will come into play later. So mathematically, this operation, which we call connectome harmonic decomposition, is in principle the same as the Fourier transform. Now, the Fourier transform takes a signal from the time domain and turns it, re-represents it into the domain of temporal frequencies. Connectome harmonic decomposition does the same thing, but in the spatial domain. So it takes a signal from the spatial domain, in this case, a map of regional activations, and turns it into a signal into the domain of spatial frequencies. Now, I am not a mathematician by training, And for those of us who are not native speakers of math, such as myself, then it's helpful, I find, to think in terms of smoothies instead. Now, you can see here these smoothies may have the same ingredients, but from their color, you might infer that the proportion to which each of those ingredients comes into play is rather different. And that is exactly what we do. The same thing, but for brain activity. And when I say we, what I really mean is Selen Atasoy and her collaborators, many of whom are in this room here, who developed this connectome harmonic decomposition framework and applied it to psychedelics. And I did promise that psychedelics were going to come into play at some point, so here they are. Now, what they showed is that these connectome harmonics can produce a signature of the psychedelic state, specifically distinguishing between LSD in terms of the brain activity transformed into harmonics that it generates, and a placebo. And here you can see that signature. And in particular, you will notice that there is a reduction in the contribution of low frequency harmonics and an increase in the contribution of the high frequency harmonics. And the same researchers were also able to show that this signature is not just specific to LSD, but rather to other serotonergic psychedelics as well, namely psilocybin and more recently DMT as well, which all act on the serotonin 2A receptor. And what we were wondering is this signature specific to serotonin 2A receptor agonism, or is it specific to a psychedelic state of mind? And to address that question, what we did was looking at an atypical psychedelic, which is a subanesthetic dose of ketamine. Ketamine acts very differently on the brain than serotonergic psychedelics do, because it is an NMDA receptor antagonist. It doesn't act directly on the serotonin system. Nevertheless, what we found is that just as it creates in the mind psychedelic-like experiences, so it creates in the brain a connectome harmonic signature with those typical increases in the high frequency connectome harmonics, although differences exist as well. But I am in a cognition and consciousness group, so part of our research interests are in looking at loss of consciousness. And so how can we look at that? Of course, it is by administering anesthesia to healthy volunteers and asking whether the same or a different signature will be observed. And strikingly, what we find is that once you give propofol to people, their connectome harmonic signature does not simply change. It changes in the opposite direction as it does when they are under the effect of psychedelics. That is, increased proportion of low frequency harmonics and a decreased proportion of the high frequency harmonics, exactly the opposite. And I should add that this signature is observed when people lose responsiveness, not just when they have a mild dose of propofol in their bloodstream that makes them drowsy but still responsive. And once the person regains consciousness, then that signature goes back to being the usual one. But we tried to go even one step further because we wanted to see that this is not just specific to one particular way of inducing loss of consciousness, but rather we wanted to see whether it can be claimed to be a generalizable signature of loss of consciousness to core. And the way we did that was comparing two groups of patients with a disorder of consciousness. This is what you see below it, but the important part is that the purple image is not obtained by comparing patients versus healthy controls. It's obtained by comparing two groups of patients. All of them are clinically unresponsive from the behavioral point of view. If the clinician asks them, squeeze my hand, look at me, they will not be able to do that. However, a few of these patients, about 8 of them out of 20, were able to demonstrate in an MRI scanner that they could hear what people were telling them because they could, upon command, imagine playing tennis or imagine navigating around their homes, and the corresponding brain regions, which are very well localized, would show up, just like they would in me or in yourself. So we call these covertly conscious patients, and we compare them with patients who cannot do that. So for all intents and purposes, as best as clinical science can determine, are actually unconscious. And when comparing those two groups of patients, this same signature is what you see, with in particular, a great reduction in the proportion of high-frequency harmonics. And so to wrap up, what we can see is that we have one particular signature of the psychedelic state that is obtained from the connectome harmonics, but is not specific to any individual psychedelic substance. Rather, it captures the psychedelic mental state itself. And we see that the opposite signature is obtained when people lose consciousness, whether as a result of anesthesia or as a result of a disorder of consciousness. And this signature is obliterated when people come back and regain their consciousness. So it is specific to consciousness and its loss. And of course, now the question is, if we have the beginnings of a landscape, how are we going to read this map? And one way that we know how to do, because of the mathematics that underpins connectome harmonics, is in terms of structural coupling. So you can think of the low frequency harmonics as indicating that function is tightly coupled to structure. Because remember, harmonics are called connectome harmonics because they come from the connectome, which is the structural wiring of the human brain. And on the other hand, the high frequency harmonics are those that are increasingly decoupled from the structure of the brain. And so what this means is that just as other studies already have indicated, and some of which are own, loss of consciousness corresponds to increased structure-function coupling, and the psychedelic state corresponds to decreased structure-function coupling. But of course, the beauty of this is that you don't have just one number for each person, rather you have an entire repertoire of connectome harmonics, from the most coupled to the least coupled. And that means that what we can get is quantify the richness and the diversity of these harmonics that each person exhibits, and obtain what is a rather striking continuum from loss of consciousness on the one hand to the psychedelic state induced by different substances on the other, which is not present when you compare, for instance, test and retest scans of the same people as they are awake, and is not there when you compare people before they lose consciousness, even if they have a level of drug, of propofol, already in their bloodstream. So it really is specific to loss of consciousness and the psychedelic state at the two ends. And our hope, of course, is that using this framework, we can start to build this landscape of consciousness, this atlas of the clouds of consciousness, if you will, that I've been talking about. Because if we can do that, then maybe with some of the technology and developments that Jakub, for instance, is going to be talking about later, we can hope to move people from a state where they shouldn't be to a better state. And of course, all of this work was done in synergy with very many people. And I am delighted that you're getting to hear from many of them here today. And I wish to express my sincere gratitude to all of them, because it is the combination of all of these people's work that has made possible what you've just heard from me. And of course, I also want to thank my funders, Gates Cambridge, who make it possible for me to be a PhD student. And I'll be happy to take any questions if you have them.
All right, great. We've got another lightning talk from Carl Smith, this time about exploring the phenomenology of the M-hole.
So it should come up in a second.
There we go.
There it is.
Yeah, so the audio was working as you saw.
Oh yeah.
So it's on PC. Was it too loud when?
I think it was. Let's just test it again.
So Carl, I'll give you a 10-minute and a 5-minute warning.
Okay, cool. It's only just one audio thing now, so yeah, totally. I can do it here, actually. Sounds good. Mystical architecture. All right.
All right, let's get started.
Hi, everyone. Okay, I'm going to talk to you about something you might not have heard too much about, which is the analogues of ketamine. So I'm Hayden Smith. I'm also I co-founded the Psychedelic Society, where we're looking at how to create altered states without taking drugs, so mainly using technology. And I'm also co-founder of the London Experimental Psychonautics Club. So the abstract, MXE and other dissociatives are very powerful tools in the exploration of consciousness because of their unique spectrum of properties. And today we're going to explore some of the definitions and the history of MXE specifically, the phenomenology of the whole, and also the afterglow of the whole, and some of the application areas, so therapy, treatment-resistant depression, which I think is actually much more powerful as a tool than ketamine, which I think has got lots of problems. Nootropics, pain management, augmenting breath work, addiction recovery, time management. Then we're going to look at some of the protocols, safety management, and the training techniques. So MXE existed from 2010 to 2014 legally in this country and around the world. If you look up the definition of dissociative, a dissociative is a hallucinogen. But MXE differs very much from ketamine and PCP, as they are developed as general anesthetics. MXE was actually designed for recreational use. It was actually designed by somebody who had phantom limb syndrome. He lost his arm and using ketamine all the time to try and take that pain away, and actually was destroying his bladder. So he invented MXE. So it's got a unique action. It's like 3 drugs in one. So you've got this opiate effect where it eliminates anxiety. You've got this very stimulating MDMA rush, and you've also got the disassociation. So that combination is extremely powerful for therapy, and I'll talk a little bit about why that is. So it's originally developed through the use of intelligent drug design. It was very, very popular, one of the most talked about drugs ever. A lot of heroin addicts were using MXE to get off heroin, very effective, almost as effective as Ibogaine, but limited data still exists around its pharmacological properties, metabolism, and the toxicity. So highly advised, if you're ever lucky enough to get hold of any, to use the harm reduction practices, which I'm also going to mention. So yeah, it's a rare example of a drug being used, being banned absolutely across the world without an existing medical use. So the big and dandies on blue light are a really good resource for exploring the history. As I said, massive amount of discussion around the substance. And this is actually a really interesting book, multidisciplinary MXE analysis. So in terms of the folklore, I mean, it has gone into folklore because so many people just really, it changed their lives. Some interesting, this guy, Jeremy Carl, who was actually an ordained minister, he created a church around MXE. There are various churches that have sprung up across mainly America. The Church of Inner Light was actually developed for DPT, which is a DMT analog. So yeah, it's very different. It's a very different mechanism for getting to a unity state. So it provides. So it's like you dissolve yourself and everything else comes in. So unlike 5-MeO DMT, where you just become everything instantly, you're kind of doing it in the reverse, but it's extremely powerful nevertheless. So this is some of the stuff coming from the minister, a molecular sacrament, he called it. So you can look at that in the recording. But this is where it starts to get interesting. So there's all sorts of analogues of ketamine. And there's a whole class of drugs within the dissociatives class. So you've got down here at the bottom the stimulants of the dissociatives, things like 3-MeO-PCE and 3-MeO-PCP. Those are your stimulants, far superior to things like cocaine. And I think, you know, if these things were more known about, which is partly why I'm here, people would get off these toxic drugs, which have a very toxic, you know, blood on your hands kind of scenario, politically especially. And then you've got the opiates of the dissociative class, things like 3-HO-PCP, 3-HO-PCE. And then above, you've got the disassociatives of the disassociatives. So things like OPCE, which is currently the best MXC alternative. And then again, they're almost different drugs within the same drug, accessible via different routes of administration. So if you were to drink OPCE as opposed to snort it, you'd have a very wildly different time. It's almost like a different drug. So an alcohol analogy, you can say beer at the top, ketamine is your beer, and then the wine is the MXC and the OPCE, and then at the bottom your spirits are the 3-MeOs, so the much more powerful substances. And again, the protocols for each of those differ widely because of this different strengths. So phenomenology, there's the K-hole. I don't know how many of you have been in a K-hole, but the phenomenology is quite distinct. So people talk about out-of-body experiences being separated from your body, melting into your surroundings, teleported to other places. Some people enjoy K-holes, some people relate them to near-death experiences. There's a really interesting Vice article where people specifically talk about how K-holes have changed their life. And then you've got DXM. So DXM is actually the first drug my mother gave me at 5. It's a cough medicine, and you may have all been drugged by your parents so they could party. So DXM, there's plateaus. Again, there are whole classes of drugs within the distinct DXM plateaus. So at the first level and second levels, you've got sort of typical ecstasy-like effects. And then in the third plateau, and this is all to do with strength of the dose that you've taken, you're experiencing ketamine-like effects. And in the fourth plateau, you're experiencing PCP-like effects and deliriums. So let's just hear a bit of a testimony of an M-hole.
I'm in, and my mind took over. I was then given a bizarre cosmic tour through the nether regions of my mind. Mystical architecture floating on clouds, strange rooms that I'd never seen before, intense size and space distortions like nothing I'd ever felt before. One minute the entire universe felt like it had been stuffed into a small corner. The next, I was suspended above an infinite void. It's like you're bathing in a warm, radiant, existential glow. Everything can be placed into perspective. You get it when you're in the hole. And the very fact that you exist is reason for a jubilant, joyous celebration of the soul. The whole helps me see how wonderful my sober life is. So what do I do now? Is it okay to go into the hole or not? What are the dangers? Emotionally, intellectually, physically? I now know how to enter a state of godlike, ecstatic, euphoric bliss anytime I want. I mean, isn't this completely absurd? What does a human being do when he's given this ability? How can I look at life in the same way after experiencing this? I just feel like something is different now. I'm ultimately nervous, excited, content, confused, amazed, enlightened, perplexed, scared, hopeful. Enter the dissociative world that we're in. If you want to go to heaven, experience nirvana, feel ultimate cosmic bliss, and achieve a rapturous state of being, you can know what it's like to be God. But how will you live with that knowledge?
Okay, some more in terms of the phenomenology. So it's actually fascinating in terms of its extreme euphoria, because it's a dissociative, and you would associate dissociatives with not such extreme euphoria. But as I mentioned, it's a very different pathway for reaching unity. But some of these drugs are like the keys to the kingdom, and I think it's fascinating that these are man-made substances, but yet have profound entheogenic effects. And instead of a come down, which is why I think these could replace things like cocaine and MDMA, is that you actually have an afterglow. So one dose of MXC will leave you with an afterglow of a month long, which means you don't really need to take it more than once a month. So following strict protocols is extremely important. And they're amazing tools for ramping up your imagination and practicing letting go, which I think is also massively important for the psychedelics. People report massively increased synchronicity, and again, things just making sense in your life and being able to meta program. It also shows the switchboard under which the workings of your depressive mind work, and empowers you to see beyond it. And some of the application areas. So this ability to do very serious self-therapy, specific combinations allow you to sit inside your backstage. So because you don't have any anxiety, you're literally able to have this 360 panoramic view, and you can see all the stuff you're lying to yourself about, but you're able to just sit with it and not judge yourself, which I think is incredibly powerful for therapy. And also for meta-programming, because you have the will. Unlike ketamine, you're very stimulated. So the first thing that happens is you just go up to all those people you've got a blockage with, and you just get shit done. So yeah, it's the true power is the ability to create lasting change. So altered traits. And I think the big problem with ketamine is that it's extremely addictive and you have to take it very often. And I think that's going to cause a huge problem. These clinics that are springing up everywhere. So there's more money going into ketamine research than all the other psychedelics combined. And I think it's absolutely the wrong way to go. So nootropics, some of these drugs, especially 3-MeO-PCE, incredible nootropic effects. You'll take one dose of that, and then within one day you'll have more insight, more ideation than in your previous 12 months. So incredibly powerful tools, very, very much like you have to take them very, very seriously and be very wary of dosage and redosage. And also, as I mentioned, lots of people, when MXC was on the scene, got off heroin because it was just not as good. Time management, and there's a serious time distortion effects. OPCE incredibly slows time down to such a factor that you could argue that things like transhumanism, which is all about life extension at all costs, is just not necessary, because we can go a hyperhumanist route and actually just slow down the perception of time rather than actual time. So just to finish off, application areas. One of the things that you can do on dissociatives is take a lot of pain. So whether it's a very intense massage that you have to deal with, or standing on a bed of nails, Shakti mat, highly recommended, because you can really sort out your body while standing on a Shakti mat. And normally you can stand on there for about 5 minutes, but on a dissociative you can stand on there for an hour, and you're really seriously helping your body in ways that you wouldn't normally be able to stand. And breath work, you can do very much longer breath holds on dissociatives than you would be able to normally. And also there are effects in lucid dreams that are very similar to what you're experiencing in things like the M-hole. So enhancing your lucid dreaming abilities, crossover between those. So protocols and safety management, hugely important, as I mentioned, but the combination of something like THC with CBD and MXC creates something as powerful as ayahuasca. And I just think that these things should be given the clinical study that they deserve, because they, the kinds of transformative effects that you're experiencing are just mind-blowing. It's also important to remember these are man-made substances, so to take the antidote at the same time as taking the drug. So green tea, very well known for protecting the bladder, and also any anti-inflammatory drugs, things like turmeric, ginger, etc. All taken at the same time really helps some of the toxicity issues. And the training techniques, talking to Chris Timmerman a lot about this, the psychedelic trainings that you can do. I think there's a whole other series of techniques that you can apply to dissociatives. And working with the tolerance. I mean, I think the thing that protects you from dissociatives is the tolerance rapidly increases, so you just don't get any effects if you continue to abuse them. So that's sort of baked in, which is really interesting, and I really think that's a good safety mechanism. One of the interesting areas is your ability to bring anything back from the whole. Training in that sense, how do you bring your insights back? And as I mentioned, MXC is the modern day Quaalude. You cannot get it for love or money, but yeah, I think we should be clinically studying it and getting it remanufactured so people can experience the wonders of it. Thank you very much.
Thank you, Carl. This next talk is our last talk in the second lightning session on psychedelics and consciousness. And it's from Benjamin Bryant. Am I pronouncing that correctly? Yeah. Psychedelic compound 5-MeO-DMT induces an altered awake state in mice. I think he actually had a new presentation. Yeah. It's all right. Just correcting a few typos, really.
Brilliant. Do you have videos in your presentation? Do you have any videos?
No, I don't.
I'm going to turn down the volume so there's less noise.
Maybe like a 10-minute and a 5-minute.
Yeah, all right, cool. Okay, everyone, hi. So I'm Benjamin, and can you all hear me correctly? Good. Online as well, I suppose. So hi, I'm Benjamin, and today I'll be talking about how psychedelic compound 5-MeO-DMT induced an altered wake state in mice. So to give a little bit of background to my research, I am not really interested in how psychedelics work. I'm really interested in vigilant state. So I'll be briefly introducing to you guys what is a vigilant state, but what I'm doing really is just to use psychedelic to try to give a little nudge into brain state and see what it does to try to understand, well, basically, yeah, wake and sleep. So what are vigilant states? Well, yeah, as I said, it's just a fancy name for wake and sleep. And we roughly all know what this is, really. Wake is like when you're just moving around or paying attention to a lecture. Sleep is, well, you can also be in a lecture theater for that. And it's basically when you are immobile, probably. But most importantly, the definition of sleep would be a reversible state of immobility and also low responsiveness to external stimuli. So now what happened in the brain during these states? Do we have markers? Do we have specific things that happened in the brain? Well, to look at that, we can simply plug an electroencephalogram machine and an electromyogram machine to measure respectively the brain activity and the muscle activity. So during wake, this is roughly what happens. So you have these little squiggly lines that shows that the EEG signals are composed of waves of low amplitude and high activity. But also what's really important about wake is you have quite an active EMG. Well, that's normal, the animal is just moving around. And these are two main markers of wake. Now, if we have a little look at sleep, well, it's a little bit more complex, right? We have two substates. You have one here in blue called non-rapid eye movement sleep, non-REM sleep, and in green, REM sleep, rapid eye movement sleep. So non-REM sleep is mostly composed of what we call here, and you can see that in the EEG curve, prominent large slow waves of high amplitudes. And as I said, sleep, state of immobility, so no muscle tone. REM sleep, however, is a little bit more complex and a little bit more surprising. We're quite astonished when we decided to pay attention in it in the 1950s. Basically, the brain looks like the wake state, really. It's just little squibbles of low amplitude. But it's mostly composed of a wave we call the theta wave. So no EMG again. This is sleep, so immobility. However, we can see some twitches as well. So if you have dogs and pets, this is the moment where they just start moving around during their sleep. Lowest responsiveness to external stimuli. This is the lowest stage of sleep. We notice rapid eye movement, hence the name. And also, this is the state of sleep where humans mostly report vivid imagery. So I'm telling you these signals, these EEG signals are defined by waves. So what do we mean by that? Well, if we do a little bit of mathematics, we can actually divide the signals in frequency and see how much they are expressed in the EEG. And we have this kind of figures. So here, each colors represent a vigilant state. So you have in red, you have wake, the wake signal, blue non-REM signal, and green REM signals. And I can plot, if I can have the pointer, I can actually show you better. Well, should be there. Yep. You can see here, for example, each of the frequencies are plotted, and here on the y-axis, it's how much they define the EEG signals. So you can see here in the blue line during non-REM sleep, in the frontal derivation of the electroencephalogram, there is a high proportion of slow wave, which is what you see probably here. And during REM sleep, actually, it's in the theta range where you have a high bump. So that means that these signals are mostly, these states are mostly defined by the expression of these waves. So we have state-specific markers, state-specific EEG markers. Now, how can we just disturb this vigilance state a little bit? Well, this is the reason why I'm here. Let's use psychedelic to do that. So I am using 5-methoxy-DMT that comes from these little guys over here, very nice chap, Sonoran desert toads, mostly. So what do they do to humans? Well, when given 5-methoxy-DMT, humans report high changes in mystical experience, in mood, positive mood, or in the perception of time and space. And actually, so in this plot, you have the 5-methoxy here in the blue line, and a low dose of 5-methoxy is actually comparable to a high dose of psilocin in this study. So there is something happening there, right? The human wake experience is changed by psychedelic according to these reports. So what does it mean in terms of brains, and what does it mean in terms of vigilance states? And that's something I'm actually looking at. So my main question is, what is the impact of psychedelics on vigilance states? So I'll just divide the question a little bit more. So by that I mean, can 5-methoxy alter the overall sleep architecture? So we'll be looking at if 5-methoxy is wake promoting, or if it delays sleep onset, for example. Or also, are wakefulness from REM and REM sleep expressed in the same way after the administration of 5-methoxy? And another layer of question is, does 5-methoxy change state-specific signals? So we'll just be looking at the slow wave activity I've been talking about, and the theta activity as well. All right, so first, let's focus on sleep architecture. So I plotted here in blue non-REM episode sleep onset, non-REM sleep episode onset, sorry, and in green REM sleep episode onset. So in broader term, that means when the animal falls asleep or when it enters REM sleep. So what we can see here in the vehicle condition, where I just inject my animal with saline, compared to the condition where I inject actual psychedelics to my animals, there is actually no difference to be found between the non-REM sleep onset. So that means globally the animals will fall asleep at roughly the same time, but there was a difference in when the animal entered REM sleep. So what all that means, quite simply, is that even though non-REM sleep onset is untouched, REM sleep is suppressed by 5-methoxy, which is a very classical response to psychedelic. But what about long-term effect? Is it just something that happens on the spot at the moment I do the injection, or is it something that happens over the course of 24 hours? So I plotted the percent of time spent in each vision state, again, red for wake, blue for non-REM, and green for REM. And you can see that over the course of 24 hours, the animals spend roughly the same time between the two conditions. So you have the classic, the animal starts falling asleep at the beginning of my recordings, so the percent of time spent wake decreases and then increases as soon as the active period of the animal starts again. It's mirrored with non-REM, obviously, when the animal sleeps, spends more time in sleep, that's logical. For REM sleep here, I just enhanced it a little bit because they always spend not a lot of time in REM sleep, and there is not a lot of difference to be found out as well. Just not even a percent different, actually. So what does that mean is that there is actually no effect on sleep architecture over the course of 24 hours of the injection. So actually, the effect on sleep by 5-MeO-DMT is an acute suppression of REM sleep. All right. But then what about what happens in the brain? So when I was asking that question, we decided to focus now exclusively on what was happening in the brain, mostly just after the injection, because we see here that there is not a lot of changes actually happening over the course of 24 hours. So this is roughly the figure I was showing you at the beginning of my presentation, right? So you have here the frequencies at the bottom and how much they are represented in the EEG signals. And 30 minutes after the injection in the frontal EEG, you can see here, that's the vehicle condition in brown. There is a little bit of slow wave activity, but not really, and then a little theta bump, and the expression of the frequency decreases. But for 5-MeO-DMT, I observed that slow wave activity actually increases in the frontal derivation. And it's more striking in the occipital EEG as well, where there is a higher proportion of slow wave activity and actually a complete absence of the theta peak I was observing in the vehicle condition. And what's actually more striking is that an hour after the injection, all of these effects are reverted. I observed exactly the same thing between the two conditions. So 30 minutes after the injection, yeah, frontal slow wave activity and occipital slow wave activity increases, but theta activity decreases, and there was no difference 60 minutes after the injection. So what I'm showing here is that 5-methoxy transiently increases slow wave activity and decreases theta activity. So I'll wrap all of that up in a brief summary. So all of my results show that 5-methoxy doesn't change the quantity of sleep. It doesn't really alter sleep architecture. 5-methoxy's effects stop within the hour following the injection. So it's quite an acute effect. It's not long lasting at all. 5-methoxy increased slow wave activity during wakefulness because the state I was analyzing before, the first half hour was roughly always in wakefulness. And it decreased theta activity during wakefulness as well. But as I mentioned at the beginning of the presentation, slow wave activity and theta activity are two state-specific markers. We define non-REM as being a state with marked slow wave activity. And this is what I observed during wake here, actually. So my broad conclusion over this data set is that 5-methoxy injection induced a transient hybrid state of vigilance in mice composed of markers specific to non-REM sleep, increased slow wave activity, combined with markers of wakefulness, which is a motor activity, because in all of my videos of the recordings, the animals was clearly moving and doing stuff, which is the main definition of wakefulness. So what I'm claiming here is that actually psychedelics such as 5-methoxy change the quality of vigilance states rather than the quantity. So, well, thank you very much for your attention. I'd like to thank my group, obviously, and my sponsors.
Thank you very much. We're now moving on to our third lightning session, modeling the brain's response to psychedelic treatments. And our first talk is by Marco Fabush, brain monitoring of for optimizing ketamine treatments of depression.
Great. Well, thanks for inviting me. It's a great joy to be here. And thanks to Kenneth for pronouncing my surname correctly. It doesn't happen very often. Sorry? Do you have any videos? Not with audio. No, that should be good. Okay. So I'll be telling you about our work here in Oxford on ketamine treatments of depression. And this talk actually builds on very well on some of the previous speakers, which I'm very happy about. So first of all, we've heard a little bit about ketamine, but let me kind of introduce the phenomenology of it a little bit better. Hopefully you can see that should be repeating. Yeah, so I mean, ketamine, as we've heard about, is a dissociative. And I mean, it's not particularly easy to try and convey kind of what happens during such a trip, but I've tried to include a little bit of a demonstration of that. So some of the kind of, I guess, easy to explain effects are things like auditory and visual distortions. Those can be quite positive, those can be quite negative, depending on your experience. Closed eye hallucinations, especially kind of geometric patterns. But some of the most important effects are the kind of internal ones. So things like an altered sense of self. If you go all the way, we have the K-hole, which is similar to the M-hole in some ways that was described previously. And yeah, and you get kind of a changed meaning of percepts and all kinds of strange kind of dissociative phenomena. And the other thing that's also changes is kind of your perception of time, which is hence that Salvador Dali painting in the corner. So why does this happen, right? That's the kind of first question I'll attempt to answer. So this is a relatively complex figure. I'm not going to go into all of the cellular neurophysiology because I'm not a biologist. But I want to talk about it a little bit because, as we've talked about, as some of the previous speakers have mentioned, ketamine is interesting because it's not traditional. serotonergic psychedelic. It in fact acts on the NMDA receptors. And very roughly, we think that one of the main mechanisms of action that it has is that it blocks inhibitory GABA interneurons. So that's those ones, the ones in circle. And so basically what that means is if you block the inhibition in the brain, and in fact most of the neurons in your brain are inhibitory, there's a lot more excitatory effects. And so in particular, the most prominent excitatory neurotransmitter is glutamate. So you get this kind of glutamate burst, so to speak. And that's thought on the kind of molecular cellular mechanism to lead eventually to things like long-term potentiation, which can lead to things like increased neuroplasticity and kind of synaptogenesis and all these kind of good things that come afterwards. So in general, basically, it makes the brain easier to excite. But whilst that's kind of relatively well understood on the cellular level, how exactly does that explain why we have, wavy hallucinations and things like that? And for that, we have to move to a slightly different level of explanation. So whilst keeping this one in mind. So the first thing I want to talk about is how it affects the brain oscillations. So previous speakers have mentioned that using things like the EEG, the electroencephalogram, you can measure the electrical activity of the brain. And this comes in kind of different frequencies. And that's what you can see on the figure there. So on the left is kind of just when you're awake, there's a very prominent alpha rhythm around 10 Hertz. And the kind of black line is where the ketamine is introduced. And you can see that as the kind of more and more ketamine gets into your system, you can see that alpha is a little bit disrupted. You get more of that kind of lower wave activity. But what's really prominent here is in that corner is higher frequency activity. I can probably point here. Which we call gamma activity, something around 40 Hertz. And that's thought, that gamma is thought to represent kind of the underlying neuronal spiking pretty well. So hence kind of, increased excitement, increased gamma. And finally, how does that kind of manifest on the kind of mesoscopic or the kind of the whole brain network level, which is some of the stuff more similar to what Andrea and speakers like him have been talking about. And what happens there is, again, we get into these networks that have been mentioned many a time. So, for example, it affects the default mode network, which is thought to be, again, the kind of result of why your sense of self feels very different. And the particularly important region for ketamine seems to be the SGACC, part of the anterior cingulate cortex, which is connected to a bunch of regions of the brain, but in particular, it's associated with processing affect. So, if you experience something, how do you feel about it? Is this a positive, negative experience? So that kind of general emotional processing is affected by ketamine. Now, I'm not going to talk about the anesthetic effects of ketamine. In fact, it turns out ketamine originally was used and still is used as an anesthetic. And in very, very high doses, it makes you basically unconscious. And so it does have other effects, like, for example, it also seems to act on the spinal cord. So there's a lot of kind of pain relief that you can get from ketamine. But here I'm more interested in the psychedelic effects. So now, we've talked a little bit about what ketamine does. If some ketamine spawned in front of you and somehow got into your body, what would you experience? And we've also talked about why that may be. But why I'm interested in it, in part, is that, as again was mentioned before, it seems to provide rapid improvements for people with especially treatment-resistant depression. So we're working in partnership with the Oxford Ketamine Clinic, and you see some of these patients, and so treatment-resistant means they've tried at least 2 antidepressants and they haven't worked for them. So they have depression kind of chronically for a very long time, and they take ketamine, and kind of a few hours later you see significant improvements, which is really fascinating. However, as one of the previous speakers mentioned, one of the big issues is that you fairly quickly develop tolerance, and that means that very often these patients need higher and higher doses, and they become dependent on the substance, and that's one of the last things that we want. And so one of the issues is that at the moment, the treatment is still relatively new, and we don't really know where the kind of optimal dose is. And in fact, there is mixed literature so far. Some people hypothesize that the effect of dissociation, some of this kind of psychological effects, may actually mediate the antidepressant effect. Now, this is very much an open question. There's about 10 papers on it out there. including 2 reviews, and basically everyone concludes, maybe. You know, if you are in the yes camp, you can make very strong theoretic arguments for why that should be the case. If you're in the no camp, you can also make very strong theoretic arguments for why that shouldn't be the case. So as far as I'm concerned, it's a relatively open question. I'm very happy to kind of not answer that here. But the point is, if some of the psychological effects of ketamine are associated with it, it would ideally be helpful if we had some objective markers of getting to that, because especially if you have a patient who's never taken ketamine, they don't know the level of dissociation that they're at, you know? And so that's the thing that we're trying to work on, which is, can we find brain-based measures of dissociation in particular and ketamine experience in general that would correlate with the treatment outcome? And so this study is in relatively early stages, so I'll just more kind of talk you through the methodology and a little bit what we've done so far. And so basically, as I mentioned, we're working with the ketamine clinic, which is really good because, you know, they've kind of, I think they processed their like 100th patient last year. So anyway, so they have a fairly high throughput of patients working within the NHS as a private clinic, still not kind of approved for general treatment. And that means that it's fairly easy for us to kind of slot into that pipeline and hopefully get a relatively high number of subjects. And we'll be using a relatively simple but clinically useful measuring device, which is normally used for measuring brain activity during anesthesia. It's called the BIS monitor. It's that thing in the middle. Basically, it's frontal EEG. And on top of that, we'll also be measuring things like blood levels, and so that we can get the ketamine concentration, even things like about the set and setting, so things like ambient noise levels, whether the patient is listening to music, also their kind of heart rate, things like that. And on top of that, we have a barrage of kind of psychometric testing, which includes some kind of standard dissociation scales. However, here's my issue with it. So here's an example of what that kind of scale looks like. You know, you can see some of the questions, like for example, I feel spaced out. Now imagine that you feel like your ego has just died and you have experienced all of the history of the universe in a single moment. Rate that on that scale. And so, yeah, so as you can see, I mean, we are working with some of these scales because, you know, they have been used by other researchers and it's important for our research to kind of be in that context. But we're still thinking about how to best do this, because one of the reasons why there may be such conflicting literature on this is that these scales are very far from perfect. In fact, a lot of them have been developed for PTSD rather than for ketamine. And so basically, that may mean that they're not best applied. And yeah, and it may also vary, kind of like if you get the same patient and they now have a ketamine experience, so they know more about dissociation, maybe the ratings change. So we are working on kind of optimizing some of these scales. One thing that we're thinking about is, for example, and if there's any kind of more objective measures in the sense of, like, I don't know, the time perception may be more quantifiable than something as elusive as how dissociated are you. But yeah, and in the kind of last few minutes, I'll just very, very briefly show you something. So this is not actually from our data. This is from an open data set that I found online from some other researchers. But it's it's very similar to the kind of data that we will be collecting. So it's basically frontal EEG during a ketamine treatment of a young patient with depression, and this time using a commercial device called Muse. And just to give you an example of kind of what this looks like. So on top we have the resting frontal EEG activity. You can see a lot of that alpha. That's the kind of main oscillatory rhythm you see there. And ketamine is relatively similar, except you notice these kind of little kind of smaller spikes. It's a bit noisier, and that's in fact the gamma activity. And we can look at that kind of on the frequency axis. So it's just some quick analysis I did in preparation for this talk. And you can compare the two states, and you can see that in fact you do find very significant differences, especially in the gamma power. But there's also other interesting things. For example, it's very clearly also the case that something called the Lempel-Zit complexity, which was mentioned earlier, is also increased in ketamine. And so we're looking to kind of look at all of these kinds of both traditional and new markers to try and see what ketamine does to the brain and how we can monitor it. One thing that I'm particularly excited is about something called empirical mode decomposition, where we can look at the fact that these waves are actually non-sinusoidal. Everyone talks about waves, but in fact, there's a very big difference between this wave and this wave, right? And it turns out that quite often the brain waves are closer to this than that. And the Fourier transform is pretty bad at capturing that, whereas some of these new techniques can actually elucidate that. So that's everything I wanted to tell you so far. Thanks to all of my funders, colleagues, and yourself for listening. And I think I may even have like a minute or two for a question if anyone has something. Yeah, so we have one question. Do we have any questions? Oh yeah, please go. So you mentioned Yes, that's really exciting. So this is in very late stages, and I didn't have time to include it in the presentation, but there is a tool that was developed, I think, by the Qualia Institute, where, so there are tools where a lot of these drugs produce traces. So if you move your hand, you kind of see the background of that hand. And there are tools, like computationally, where it kind of draws you dots, and you ask to match the kind of frequency of the dots with what you see. And so that's for traces in particular, and we are thinking whether there is something like that, but for time perception. We don't have the answer yet, but if there was, that would be really cool. Oh, cool. Yeah, come to the pub and tell me. Anyway.
Thank you very much, Marco. We are now moving on to the second talk in this session, Whole Brain Modeling for Psychedelic Treatment in Depression by Jakub Vohryzek. Take it away.
Is it being recorded if I'm here?
Yes, yeah, exactly.
Do you have any videos and audio? No, it should be fine.
Yeah. So let's get me started. Thank you for having me. Again, it's a pleasure to be in person here with all of you. Yeah, I would like to share some of the work we've been doing on whole brain models and in their use in psychedelic treatment for depression. And of course, as many speakers have said before, it's always a joint collaborative effort. And so so it's been done with colleagues and friends here in Oxford, but also at Pompeu Fabra in Barcelona, and with colleagues at the Imperial College London at the Psychedelics Center. So our motivation was to see whether we can, unlike Marcos, that was focusing more on experimental designs and doing the analysis, we wanted to see more how mechanistically we can kind of look and see whether we can predict certain treatment influences in various brain states. Here it will be for treatment-resistant depression. And just kind of in general, when we talk of treatment, it can be various different methods, such as deep brain stimulation, transcranial magnetic stimulation, which are external magnetic or electric, we call it perturbations, manipulations of the complex system of the brain. But also it can be more, let's say, simple sensorial inputs that can also propagate through the hierarchies of the brain and induce changes. Or it can be, of course, pharmacology, which is the case with psychedelics, psilocybin, for example, and we know that they are very good at mediating through the serotonergic receptors and neuroreceptors. And for example, in the context of the deep brain stimulation, rather than relying always on the animal models or neurosurgical empiricism, we would hope that at some point we can have an in silico, a virtual brain, if you may, that we could perturb, we could try and see whether we can find an optimal incision point that can then alleviate certain conditions. And in our context, we looked at our goal was to promote a transition in the treatment-resistant depression trial. This was a data set from Imperial College London. It's the pilot study of open feasibility study where 15 treatment-resistant depressive patients with treatment-resistant depression were collected. I think exactly you mentioned that there were two antidepressant treatments didn't work for them, and these were scanned before and after psilocybin treatment of low 10 milligram oral dose and high 25 milligrams dose, I believe. And then we have 1 day fMRI scan. So this is this resting state that Emmanuel was talking about. fMRI, we call it brain state, spatial temporal dynamics of the brain. And what's interesting is
course, we hope that we see 15 responders to this treatment. But in our context, it was very nice to see that we can have 5 weeks after the treatment, 6 responders and 9 non-responders. So that gave us an opportunity to see and explore mechanistically whether we can say something about what is about the treatment that works for some patients and doesn't for other patients. So I've mentioned mechanistic and brain states a lot. So I guess these are the 2 ingredients that we need to put together in order to arrive to a representation of this virtual in silico brain. The quantitative description of brain state was nicely mentioned by Andrea, where it's one way to look at it, where we can go through the connectome harmonic decomposition, for example. But of course, there are various methods. I'll be speaking slightly about a slightly different aspect. And then the unifying mechanistic model is a way to arrive to a virtual brain or a representation of a brain that then we can use and then perturb bit by bit to see how that can promote a certain transition between different brain states. So when talking about brain states, I guess the traditional representation would be we have these scans, and then we apply atlases and predefined atlases that allow us to obtain a regional time series of different brain regions. And then, as Emmanuel and many other speakers have discussed, then you can do a correlative or a statistical measure that tells you how much different brain regions talk to each other. And that's what we would call a static functional connectivity. It's a very nice measure because it's very intuitive and gives us a very rich description of a brain state quantitatively. I would like to motivate that we need to perhaps go a bit more dynamic, because here what we essentially do, we're collapsing time. So we're basically saying, a snapshot of a brain is basically a frozen description of its functional network. Whereas what we can do, and in this method, I'll just briefly talk you through it. We do something what's called a signal, which we turn the signal into, and then that allows us to have a phase information, as you can see in the polar coordinate, polar plot, and that allows us to obtain an information phase relationship between different brain regions. But unlike in this static point of view, now we can have every time point in time. And this is really cool, because what it means is that every snapshot in time, we have a pattern of connectivity that evolves in time. So what we can do with this is then ask ourselves, well, are there some underlying substates that kind of reoccur in time, that they come and go? And that's what we did through a clustering method. And what you can see is that the temporal trajectory arrives to kind of goes towards a certain centroid, which would be the dominant representative substate of this brain state, and then kind of veers off and goes to another one. So we could think of them as metastable substates, that they are not something that the brain falls into, but rather kind of approximates and then kind of goes to a different state. And then we can summarize them through this, what we call probabilistic metastable substates, which is a way to say that on average, there's a probability of a certain substate being active in the recording. And so we take this as a dynamical measure of what the brain state is. And so we applied this to the data set, where we had the responders and non-responders before the treatment, as I mentioned, and after the treatment. And as you can see on the left, for the static measures, there are no differences, or we couldn't parse differences between the patients. Whereas in the dynamical sense, there were differences when looking at this metastable substates between pre and before and after for the responders to the treatment, which wasn't the case for the non-responders. And then we saw differences between the responders and non-responders after the treatment, which at the baseline wasn't the case. And that gave us a motivation to go further and try to see whether we can model it, and then see whether we can say something about this kind of mechanistic, causal, treatment-based approach with the modeling. And that's the second ingredient, which by this point I hope you've heard at least once from Morten. Maybe, I think at least so you kind of have a sense of what the different ingredients are when we're talking about having a somewhat unifying principle that allows us to build a virtual brain, if you may. And so for that, we need the anatomical structure, which is basically the white matter tracts of the brain. At every node of this network, you can describe mathematically a regional representation of the dynamics. Because we have some expectation that the dynamics will look on the regional level in a certain way, and that's what we do. We use a Hopf model that is a simple bifurcation-based model that allows us to have a flexible representation of the different dynamics that one can see, different dynamical regimes that one can see in the brain. Kind of more noisy-based, more oscillatory, driven by the intrinsic frequencies. Somewhere in between, at the bifurcation point, is this fluctuating, somewhat metastable state that has been shown to represent very well the resting state activity of the brain. And then again, the last part, this is important, is to fit this representation of the brain to the quantitative description of the choice of the way we want to describe the brain state. So in our case, it's the probability method for both substates, as you can see there. And so this is exactly what we did. And as you can see visually, so what I'm showing you here is, so here you have the responders before and non-responders before the treatment. And when we fit these models to these descriptions, we get a pretty good fit to these conditions, both in the responders and non-responders. And the way we do it, we fit this global coupling parameter that seems to be the one that affects or can represent, can tune well dynamics. And this is exciting because what we have now is basically a in silico representation of the preconditions of the patients. And now we can kind of go and perturb individually different brain states with different intensities, both for the here for the responders and for the non-responders, and see how the dynamics evolve on the spatial temporal kind of spatial temporal sense, and then compare their PMS descriptions to the healthy state, which is here 5 minutes, thank you, which is here represented by the responders to the treatment, because for whatever reason in their brain or in their description, spatial temporal dynamical description, they seem to respond to the psychedelic treatment. And this is what we did. And that's what I'm showing you here, where you have the brain regions that we individually kind of perturb and see how they unfold. And then we on the y-axis, on the x-axis, you see this simulation type and intensity. On the negative, you will be simulating it with the noisy dynamical regime. This is also the fact that this is also to be said that the model is fixed, and all we are changing is the parameters, the local individual parameters of the model, every brain region. And what this essentially allows us to do is to find an optimal region, as you can see here in red, where we can see that on average, the regions give us a very good description of moving from the responders before the treatments to the healthy state, which is interesting because then we can take this region and compare it to the non-responders and see whether in the difference of within these regions, whether we can see some differences that would give us an idea of what is it about the non-responders that doesn't work. And so that's what I'm showing you here, when you can rank order the brain regions that seem to work in responders to the treatment, but that's not the case for the non-responder. We see some posterior regions around the temporal parietal junction, and also quite highly ranked, there are some limbic regions such as the amygdala or parahippocampus. So that's very interesting. That kind of warrants further investigation. And lastly, what we did in the remaining of time was to also, because we know that both the psychedelics are serotonergic agonists or serotonergic neuroreceptor agonists, but at the same also the depression has been hypothesized to also be, as we heard before, related to the serotonergic release. So we just wanted to see whether there is something about these regions that can be reflected in the density maps of these of these receptors. And what we found very interesting that they seem to be correlated only with the 2A and 1A receptors, which one the one known to be the 2A very much the psychedelic experience being mediated through these receptors, but also the 1A seems to be implicated in the what Robin Carhart calls the passive coping mechanism in depression. And so yeah, now just to conclude, yeah, so it's just an idea of using these in silico models as a new way for psychedelic treatment. You know, to different explanation of brain state, you would have a different model. Markov, for example, has a nice mathematical model that tries to link connectome harmonics as described by Andrea. So having a different type of model to this description of the brain, which is also very exciting. And yeah, and so if we can use these models, maybe we can have a more mechanistic evidence of what type of regions might be driving, I guess in this context, psychedelic treatment, which I think there is a lot of space to further explore, because these models can be enhanced by further more biologically relevant parameters that can give us more insight on the influences of the psychedelics to the depressive patient, and then yeah, so then hopefully we can extend it to other neuropsychiatric disorders. So thank you very much.
Just on time. Yes, just on time. All right, so now we're in the last talk before the second break. So just a note for the break, you're welcome to go back into the Sherrington building. Just please do make sure that you wear a mask whenever you're moving about the buildings. We've just gotten a few emails from the department reminding us and encouraging us to please wear masks indoors. Great, and without further ado, we've got Rayan Zafar, who's going to be talking about harnessing scientific discovery with clinical equipoise, strengths and pitfalls of psychedelic neuroimaging.
Good afternoon, everyone. My name is Rayan Zafar, and I'm a PhD student at the Center for Psychedelic Research and Neuropsychopharmacology at Imperial. And today's talk, I'll be talking to you about harnessing scientific discovery with clinical equipoise and some strengths and pitfalls of psychedelic neuroimaging. So neuropsychopharmacology is a study which describes neurons, which are the fundamental cellular units of the brain and the central nervous system, the psyche, which describes the totality of the human mind, the unconscious and the conscious, and pharmacology, which looks at the interaction between chemicals and biochemical properties associated with disorder in humans. It's an extension of a discipline of psychopharmacology, which specifically looks at how drugs interact with the mind, but neuropsychopharmacology particularly wants to know how and why these changes happen. At Imperial, what we conduct are clinical trials in OCD, depression, addiction, anorexia nervosa, and chronic pain. But on top of that, we also want to understand the mechanism of action. So we look at neuroimaging to understand why the treatment outcomes occur. Within these constructs and these studies, there's sometimes a difficulty between the therapeutic intent and the neuroimaging paradigms, where therapeutic intent is not the primary objective. However, there is a predominant ethical view that all clinical trials should have therapeutic intent. But what this actually fundamentally boils down to in our trials is something called the therapeutic misconception. And that describes when a research subject fails to appreciate the distinction between the imperatives of clinical research and of ordinary treatment that is received in clinical care in society. And therefore, these individuals inaccurately attribute therapeutic intent to research practices. Over the course of this talk, I want you to consider the different ethics involved, specifically in neuroimaging in clinical trials, and some of the risks and benefits that these may entail for participants in the context of psychedelic neuroimaging. So, first of all, in order to understand neuropathology, we have to understand disorder in the body. And there's several ways that we can earmark disorder or neuropathology. And One of the ways that we can do this is by looking at neurochemicals. So we've heard today about serotonergic deficiencies, which may be underlying depression, but then we also, in addiction, look at dopaminergic deficiencies. So using specific radio tracers and comparing between healthy individuals and individuals with addiction, we can quantify receptors in the brain. So dopamine receptors are a target. And what we found from imaging the dopamine receptors in the brain of individuals with cocaine addiction versus healthy controls is that they have fewer receptors in the brain. But the number of receptors in the brain doesn't really tell us much about the function of dopamine. It just says that there are fewer receptors. And so in order to understand and to quantify how much dopamine we have in the brain, there's certain drugs that we can give to people to stimulate dopamine release. And unsurprisingly, these are stimulants. And so what we give to participants inside the scanner is amphetamine drugs. And so this is a powerful stimulant which causes dopamine release in the brain. But in order to quantify in addicts, specifically cocaine addicts, how much dopamine releasing capacity they have, we have to also give them these stimulant drugs. And what we found from these studies is that as well as having lower receptors, these cocaine addicts also have lower dopamine releasing capacity. Using similar sorts of techniques, we can also look at different endophenotypes of addiction. So we can look at individuals who've got opioid or heroin addiction. And what we've done in this study over here is we looked at the differences in dopamine release in the brain to heroin to understand whether dopamine was the contributing driver towards heroin's reinforcement effects. And what we found that it wasn't. So dopamine doesn't seem to be particularly important in the reinforcement effects of heroin, whereas the stimulant addictions it does. But what we did see when we asked these patients how they felt in the scanner is that they particularly had increases in urging and craving, and also felt more increase in intensity levels in terms of withdrawal, and also felt high. So one of the fundamental questions or findings that we found is that cocaine and heroin addiction are fundamentally different neurophenotypes. Dopamine seems to be driving cocaine addiction and not heroin addiction. But there were also some important clinical considerations from these early findings. Essentially, what we've had to do is give stimulants to cocaine addicts and heroin to heroin addicts. And although this gives us an insight into the brain, what we could be doing is perhaps facilitating a further entrenchment or an exacerbation of their condition, or perhaps giving them the possibility of relapse. But these early studies have fundamentally allowed us to develop other paradigms which don't involve pharmacological stimulation. And this specific behavioral paradigm we're developing at Imperial is a roulette task in the scanner. What we do and what we've weighted this to do is to give you more reward than losses. And so we call it reward-enriched roulette task. And with fMRI, we've been able to see that it activates reward pathways in the brain. Why this is specifically important is because using simultaneous fMRI and PET, we can actually look at the changes in dopamine for the first time in the human brain in response to behaviorally induced tasks. And so you might think, what's this got to do with psychedelics? So we hope to be able to understand whether psilocybin can restore these dopaminergic deficiencies that are observed in individuals with addiction, and whether we can actually see a restore of dopamine. So this will be the first molecular account for the restore of dopaminergic deficits in addiction as a result of psilocybin when the results of this study are published. Another powerful way that we can look into the brain is through cue reactivity. So cue reactivity describes a process where we provide individuals with addiction images of things related to their addiction or the environment that they're in. And what we see more generally across this meta-analysis is that several networks in the brain robustly activate. And this is seen particularly in the salience networks and also executive networks too. What we're interested in at Imperial is understanding how this translates into behavioral addictions. And so we're piloting this new fMRI paradigm where we look at individuals' brain responses to gambling cues versus social cues, food and nature cues, which are considered to be rewarding and naturally rewarding stimuli. Recently, we featured in a BBC documentary with an ex-football player called Paul Merson. So Paul Merson played for Arsenal and also for England, and he's been a gambling addict for most of his life. He's lost over £7 million due to his gambling addiction. And he, at the time of this study, had been 6 months abstinent. So we looked inside Paul's brain to see how his reward systems were reacted to some of these control cues. And so in response to food, we saw no activation in the reward system. Equally to social cues and nature cues, we saw no activation. But in this N=1 study in fMRI, well, we're doing a larger study, but in one patient, what we saw was robust activation following 6 months of abstinence in the brain regions related to reward and salience. And this is powerful, but more importantly, what we asked Paul after the scanner was how he felt. And he said he felt like he wanted to gamble for the first time in 6 months. He was visibly unsettled. And he said that it was scary that something like 10 minutes of watching random clips, even if only a few of them being gambling ones, could precipitate relapse in him. But what this powerfully shows is that there is aberrant salience processing in addiction. So as I mentioned, Q exposure derailed the therapeutic process in individual, but in relation to psychedelics and treatment response, if we use these paradigms after somebody's taken a psychedelic, so after the therapy has been involved, what we will be able to show is whether psilocybin can restore some of these functional impairments of the brain, but we're not necessarily sure whether they can necessarily be therapeutic or safe for individuals with considerations to relapse. So what I've discussed over here is how we can look at the clinical effects of psychedelics in a pre versus post-experimental paradigm, but we can also look at the acute effects of psilocybin on the brain. So that's under the influence of psychedelics within a scanner. So in this particular study, they wanted to understand the effects of psychedelics on social exclusion. So social exclusion is prevalent across many psychiatric disorders. And increased reactivity to this could actually lead to greater pathology. So they wanted to understand whether psilocybin can modulate negative social isolation. And so they developed this fMRI paradigm where a ball is passed between players, and essentially you, the participant under psilocybin, don't get past the ball, and you are disconnected from the internet. So you're frustrated, isolated, essentially bullied, whilst under the influence of magic mushrooms. So what they found was there were brain regions specifically associated with this processing, and surprisingly or unsurprisingly, psilocybin reduced the feelings of negative isolation, and this was correlated with feelings of unity. So whilst this is powerful and it can show for the first time the relationship between psychedelics acutely and social isolation, essentially, it's not the nicest thing to make you feel socially excluded whilst you're tripping. And it could facilitate, some pretty negative experiences. And actually, another study found that experimental settings involving neuroimaging strongly predicts unpleasant and anxious reactions to psilocybin. So perhaps acute measures of psilocybin aren't the best way to model psilocybin more generally. So I'm going to finish with telling you some results which were unpublished, which are quite exciting from our New England Journal of Medicine trial, which was published last year by Robin. So for those of you that don't know, we've done 2 studies at Imperial College. So the first was an open label study in treatment-resistant depression, which is widely regarded as a catalyst for the modern psychedelic renaissance we're witnessing today. But we did a follow-up study where we wanted to compare in major depressive disorder, individuals giving 2 doses of psilocybin versus escitalopram, which is a commonly prescribed SSRI antidepressant. We've also spoken today about the rebus model, which suggests that the brain in pathological states has these very rigid ways of thinking, and neurocomputationally these can be modeled by having free energy landscapes which are very rigid, and cross-cortical communication has been blunted, and that psychedelics can acutely change this by increasing functional connectivity in the brain, and therefore allowing a reset of balance across the board. And so in our first study, using this modularity concept, so modularity describes how well connected the brain is. So high levels of modularity mean that the brain isn't very well connected and it's very independently segregated in terms of its communication. Low modularity means that it's communicating with all brain regions, and psilocybin robustly showed in the first set of our open label study it decreases brain modularity. What we also found is that those individuals which had lower brain modularity, so more cortical connection, had lower scores of BDI, so lower depression scores. So it's a good biomarker for depression. And then we also found that those had the greatest changes in modularity, so the greatest decrease, represented by increased functional connectivity in the brain, had the greatest benefit from psilocybin, so the greatest reductions in BDI. So what we also found in our follow-up study, and this is the unpublished and excited data, is that we followed this up and we found that actually in the psilocybin group, but not the antidepressant group, that modularity was decreased in the brain, and that this decrease was predictive of treatment outcome at 6 weeks. So it was a prognostic and a diagnostic tool, as we've heard before. And actually, these were happening in regions of the brain, such as the executive network. But quite interestingly, and in my line of work with addiction, we were seeing these changes in functional connectivity leading to treatment response in areas of the salience network, which is a reward network, and also regions of the default mode network, which we've spoken a lot about today. So what we fundamentally have found from this research is that psychedelic drugs change the brain in order to lift depression, which SSRIs do not seem to do. So what I've discussed today more broadly is that we can look at neuropathology in the brain, but sometimes this can be at the detriment to patients, and we can give them certain neuroimaging paradigms that can precipitate withdrawal or exacerbate their conditions. However, these functional and PET imaging paradigms are powerful ways to assess the mechanism of action in the brain of psychedelics, which has not been done as extensively up until now. We can also look at the acute impacts of psychedelics in the brain, but we can also understand that perhaps these aren't the best models to model psychedelics from. And then finally, we've been able to prove some computational models which show the power of neuroimaging. And I think neuroimaging more broadly allows us to have confidence in a specific mechanism or confidence in a therapy, because it's great to understand clinical outcome, but as scientists and doctors and even regulators, knowing why and how will fundamentally get this through to the market. So I'm going to finish on a quote from Aldous Huxley, who says, "Orthodoxy is the diehard of the world of thought. It learns not, neither does it forget." And a piece of advice from the director of our center, Professor David Nutt, who said, "All researchers need to make a specific effort to avoid being embalmed into the orthodox consensus." I'd like to thank all of the team at Imperial. Some of them are not on that picture, but they're all instrumental to a lot of the work that we've discussed today. And I'd also like to highlight the Cyprus Clinic, which is our the first NHS psychedelic research clinic in the UK, which is now open, which many of our studies are running out of. If you have any more questions, you can email me. I'm on Twitter, but thanks for listening.
All right, break time.
Yeah. Can you? Okay, cool.
If you want to, if you want to look through your slides now.
Sure. I also just want to edit something in my presentation.
Oh, you did. Okay.
Quickly, so.
Yes, of course.
Cool day. Lots of stuff.
Yeah.
Okay. Yeah.
Sorry.
Sorry.
Is there still? Yes, there is. Yeah, there's still snacks and food in the canteen.
Okay, but I'd like to see beforehand, just so I don't want it to be, it's just easier for me to, Okay.
Nice. Hey. Yeah.
Oh, so are we on the camera? Is it live at the moment? Is it
Okay, nice.
Oh, so it's not.
Fantastic, yeah. That's great.
They can hear everything.
What's our plan for letting people back in?
But then you're saying, okay, so this, you want us to sort of present here so people can see it?
Yeah, exactly.
I see,
So that's
okay.
why if you do use this or this or this, it's a bit better because it forces you to be
Yeah.
to the camera. Do you want to see your presentation?
Yeah, I do actually.
So which one is it?
It's Kieran. Yeah. So what do? I go to, is that presenter
So I'll
mode?
just
No, I want it in the full mode.
The full mode, okay.
Because do the people at home see the slides?
They see whatever's on this, which is
Okay.
so they won't see this presenter. You don't. You would rather not have the presenter mode, or?
I'd rather have the normal slides. Yeah, the way that, because I've not got any notes.
Okay. So let's see. I think it should be
Just so it's like the biggest version possible. Is it not that one down there? No.
I think this one, because for some reason it's defaulting to the.
Okay.
That might be better. Yeah.
Yeah. But is that?
I think it didn't. It didn't like that, did it? So when we do that, it does. How do we make sure that's presentive? We don't want it. I'm not sure why it defaults to that. It typically is a setting.
Okay.
Is use slideshow? Then
Try that.
Yeah, so that gave us the full screen, but then it.
Oh, there we go.
But it, I think it then turned off. Did it not? Yeah, exactly.
Ah, okay. That's annoying.
So what's your goal is to try to have it present but like take it full screen?
Yeah, just because I haven't got great eyesight as well, and some of the stuff I'm reading, so that was
okay.
my intention. But
I see. Maybe there's a way we can just like
make
this. Yeah, like how about that? Is that good?
okay, so yeah, that's all right. Actually.
Okay. All right. Nice.
Yeah. Can I see? It's not that easy to see that. Okay, so I'll just have
You
to
can turn the lights off if you prefer.
That's all. That should be all right. Okay, that's fine. That's cool. Thank you.
Nice, of course.
That's good to.
Yeah, it's a good idea. So cute. I'm going to take a picture, guys. Do you mind being in my photo, or like,
Or turn around.
Or like, I don't particularly
Just
want you to
casually
be in my
walk
photo,
up.
but like.
Mine is empty.
Yeah, there you go.
You
Do you want to be in the photo?
Yeah,
might,
me and Julia.
might look a bit lonely if you're
Yeah,
just the
because me
only
and Julia are
person in-
presenting in a second.
You can do it before and after.
Okay, 3, 2, 1, shrooms.
Yeah, thank you.
That was so smooth.
Is it really beautiful lecture theatre?
Wait, damn, we look so fucking cute. What's going on here?
That's good. It's really good photo.
Yeah.
Oh, but you made a photo. Yeah, you kind of killed it. Well done.
I can give you one.
Yeah, I'm really good.
Put that on my film Twitter.
Did you find others? I don't know where they are. Oh, here's mine, look. But it is in presenter mode. Wait. We're live streaming.
Okay, let's wait.
How do you?
We can do play from start. Okay.
And then, but I want mine to. How do you know how? Yeah. So I try to do use slideshow. Okay, but then so it's fine now. But then when I move off, then it goes like that. But so I think they've said just do it as presenter view.
Yeah.
But how do I do that? I need it to be as big as possible, so I need to be able to see this. Is that as big as?
I think that's as big as it gets.
Okay.
But
That's
otherwise,
fine.
you can just like, I guess, make notes about it, huh? Like on a piece of paper for you to see it. But can you read that?
Yeah,
Do you have
I
your
can.
glasses?
Yeah, I have. Let me see if they work.
But where are the notes?
Such a nerd. So let's just click on that. Okay, I'll just have them down here.
Yeah.
Just in case, yeah.
Okay, but I need to be able to see my goddamn notes.
And
otherwise, I'm so fooked. That wasn't recorded.
And that's no water. You know, I think it is.
I'm so embarrassed.
So when we're speaking, where do we stand?
Like that one guy was just.
Yeah, but then you're not on camera. Oh, I see.
I mean, I'm gonna be there.
Wanna go? Yeah. I guess we can move around. I think in subconsciously in my sleep, so not too much.
I'm just testing the audio a second. Don't worry if I speak to myself. I'm just testing the audio here on the live stream. Test, test. How about now? Can you hear me now? Test.
Don't worry. We're just gonna preview Julia's slides real quick.
Do you have any audio that you would?
I don't need audio.
Okay.
I just need my notes.
Yeah,
Yeah, that's
yeah. I think you'll be able to see that definitely.
Slideshow.
Oh, incredible. Thank you.
If it's in the way, we can either get rid of it.
Perfect.
Or we can put it up there.
Perfect. Thank you so much.
And you can also make this a bit wider if you want.
Ooh, spicy.
Yeah.
Whoa. Okay, cool. I'll play with it later. Thank you, though.
I'm just gonna put the QR codes back.
Thank you.
Of course, yeah.
Do you know where the bathroom is?
Yes.
Thank you very much.
Structure for
In the sense that it's sleep, right? Oh, I think yes, there's been but just like an example, what things are these programs? I know. Oh yeah. That's a good thing. Yeah. I guess when he was taking back therapeutic effect also right? So like I said, I mean, there's like I think that like in terms of the yeah, which I can see that you could imagine it. Yeah, that's true. I guess if you see that kind of point, I don't like it. I don't think people like I don't really, but it's true. Something so in that kind of
left room, and you had a suggestion you would like to improve it with a question, or yes.
Oh, sorry. Can I have those like laser pointer? Cool.
Whenever it's here, it's visible.
What's that?
Whenever it's on this screen, it's visible. And then you can use either this or the space bar.
All right, everybody. So we're in the final stretch of the symposium. We've got our last keynote delivered by Ravensbourne on psychedelic sociality, followed by our last lightning session. Just wanted to make another quick plug for our merch, which sadly didn't arrive in time, but you can order it from the Oxford Psychedelic Society website through this link or on one of the QR codes you find here in the lecture theater. And without further ado, Ravensbourne.
Oh, so I'm Lior. I work in Imperial College for the last 9 years in the Center for Psychedelic Research. Started a lot with neuroscience and then research about therapy. But I've moved in the last 3 years to more look at like social dynamics with psychedelics. Actually, a bit more than 3 years ago, Morten examined my viva, which was fun. Excellent examiner. You'll be happy to hear that I'm having my graduation ceremony only in 2 weeks. So, so the world was on pause for a while, right? So, been 3 years ago. So since then, I had like some tangent in my research and then going to sociality, and I won't have a lot of neuroscience today in the talk. Maybe if I'll have some at the end of the talk, if I have some time, I'll actually show some brain blobs and networks and stuff. So sociality, anybody knows what does it mean? It's a kind of a niche term. A lot of evolutionary psychologists or evolutionary biologists used to like this term. And it's the idea that animals, humans, primates form into groups, right? And that's part of their evolutionary advantage of primates usually, but then humans perfected it, is to form into groups. So all primates do that. And as humans, we manage to perfect that ability and to create really, really, really large group. And this is usually done by our culture, right? So culture is the cycles or the movement between generations of stories, narratives, ideas, and technology. So this is kind of like one of the definitions of culture. And this is one of our probably
what we advanced as human the most is culture and sociality. And there's a lot of things there because culture is also related to learning. And as we already saw, psychedelics change learning, but we'll get there. So a lot of psychedelic research starts like this, like usually clinical talks about psychedelics, they start by saying depression is on the rise, depression is an epidemic, it's all over, psychiatry is in crisis, there's no more good medicines for this, but psychedelics will save us. And maybe they'll help save psychiatry, but what kind of a lot of times these talks miss is that the social political reasons for the decline in mental health, right? So if there's a rise in depression, it's probably something in society that doesn't really function well. Some of the candidates are alienation, loneliness, lack of meaning, cultural poverty, and maybe the problem is not that psychiatry is in crisis, it's that society is in crisis. Right, so a lot of like psychiatrists and people from the mental health background kind of look at it, and this discourse is very familiar within mental health. I would like to say that I'm very much influenced by Bruce Alexander. Bruce Alexander is a psychiatrist from Vancouver who studied addiction, also very much influenced the work of Gabor Mate. And Bruce Alexander is known for his studies of the rat park. So a lot of people, there's famous studies about animals and addictions that animals, if you give them heroin or cocaine, they'll press the lever constantly until they are dying. You know, that's kind of the classic studies of addiction. And Bruce Alexander did the rat park experiments, which show that if you give rats the option to take cocaine in an enriched environment in which they have some like wheels and friends and other mice there, then they'll go to the lever, but much less frequently. So they go once in a while, they get their hit of whatever drug, but they're not getting addicted to that in the same way. And then Bruce Alexander will continue to study the same kind of point about addiction and the social component of addiction. Usually through this book is through historical accounts of addiction and how societies in decline, addiction is rising when societies are in decline. So he gives a lot of examples of how drugs which were part of a society, let's say opium in China, or let's say different substances that were kind of part of the culture and were not addictive, became addictive once the society started changing and dislocating. So he studies Native Americans in Canada, and he saw that the addiction of Native Americans is also related to dislocation from their tradition. What's his claim as well is that capitalism, while usually this phenomena is seen in societies in decline, it's also seen more in capitalism, even when it's flourishing, because capitalism is breaking also the social bonds. So it's like a very social political idea behind the reasons for addiction. And I like to say that like this is part of my motivation as well, and part of my motivation is also my own personal experiences with psychedelics. A lot of times with they're social, they're with friends, they're fun, or they're socially meaningful if it's in a ritual. And I felt that this is not communicated in the discourse of psychedelics. So a lot of times, you know, this is kind of portrayed as the what is psychedelic therapy with eye mask and music, very isolated, speaking about inner healer, personal therapy. But not necessarily very much relating to the relational and social components of psychedelic use. This is very far from how psychedelics were perceived, you know, not so long ago. Joe Cocker and Woodstock, you know, really rocking it, revolutionary, social political change, anti-establishment. So there's another cultural and social component to psychedelic use that sometimes is not in the current discourse of mainstreaming. So the talk will cover and is kind of dedicated to that component, to the social component, and the talk will cover connectedness. It will cover psychedelics and interpersonal relations, psychedelics and groups, psychedelics and culture, and psychedelics and revolutions. If I'll have time, I'll also do some psychedelics, some sociality and neuroscience. But it's important to kind of always keep that in mind when we speak about psychedelics, that there's hard to really say what is inherently psychedelic. Because it's always context dependent, or at least a lot of the research shows that. So when we look at like psychedelic therapy, the long-term outcomes a lot of times are related to the experience. to the state, but that experience is influenced by the environment, by the dose, by the traits of the people, by the pre-state, and all of these lead to certain experience, which leads also to outcomes. So it's really hard to say psychedelics do this or that. And we can say that maybe they enhance the influence of environment, of context, maybe they enhance meaning, something that might be inherent in them, but they're not therapeutically inherent, or they're not even social connectedly inherent, as if I'll show in the talk. It's always in relation to context. So the first kind of zoom in on where we see relationality and connection with psychedelics is actually in the experience itself. And if you look at different components of the psychedelic experience, you can actually see that and realize that there is a component of connection in them. So in the mystical type experience, or the unitive experience, or the kind of ego dissolution, but the positive kind of ego dissolution, this merging of self and other, there's a component of connection. So when people speak about the unitive experience, they speak about interconnectedness, about feeling connected to some, maybe humanity, the world, or maybe just the people around them. The experience of awe also have this component of connection. Emotional breakthrough, so this is a cathartic release that's seen a lot in psychedelic therapy. A lot of times it's actually when people think about their relations to other people and realize different insights about the relations to other people, and that sometimes bring this kind of emotional catharsis. Communitas is a experience, a group experience that I'll kind of go back into in a few slides, but it's the experience of group harmony and connection. Entity encounter, a very common experience with DMT. People and some people explain it as like our innate social manifestations of entity, and then our relation to those entities. So it's like almost a social hallucination, entity encounter. And animism, animism is the idea that there is a spirit in living things, so spirits in the trees, or spirit in nature. And a lot of times when people take psychedelics in nature, they actually feel that the tree is breathing, the nature is alive, and they feel connected to that. So it's kind of, they socialize the nature in the experience, and then they build a connection with it. But just also to mention that a lot of like these are like kind of positive experiences, but a lot of times there's social negative components. So paranoia, alienation, separation, these can also happen with psychedelics. So again, this is a context-dependent matter. So the idea of connectedness is very kind of seen in many qualitative research with psychedelics. It's seen in ayahuasca for substance dependence. Roz Watts, or lead clinical therapist from the depression trial, saw this as one of the key components of healing for people in our depression trial. So people felt more connected. Alex Belser saw it with psilocybin for end-of-life anxiety. Noorani saw it with psilocybin for smoking cessation, ayahuasca for addiction, and in ayahuasca retreats, and in psilocybin group therapy as well in UCSF. And all of those qualitative research reveal this component of connectedness as something that's important. So together with Roz Watts, we have this paper that's not published yet, and I co-authored this with her, and we kind of created the connectedness scale, the Watts connectedness scale, and we saw that people describe connectedness to self, which is emotions and body, connectedness to other, which is family, community, and connectedness to the world, which is connected to humanity, to nature, to something kind of bigger. And we see that for many people in just a natural observational study, so a large sample, we see that many people show a significant increase in connectedness in 2 weeks, in 4 weeks, and also up to 6 months. We see those increases for all of those connections to self, to others, and to the world. We see also that this experience, the change in connectedness is mediated by the experience itself. So people who had more mystical type experiences during those, this is from a ceremony study, so during those retreats and ceremony, also had higher increases in connectedness, also emotional breakthrough predicted increases in connectedness, and communitas, so the kind of group harmony in the ceremonies. Now we took this measure and we took it to our clinical trials, and we measured that compared to escitalopram. And this is our the same trial that was published in New England Journal of Medicine. And we see here that escitalopram does not increase connectedness, while psilocybin does increase connectedness. So while both can be antidepressants, psilocybin seems to work differently on the long term. So these are long-term changes on the long term of how it is clinically beneficial. And we think that connectedness might be one of those key differences between psilocybin and escitalopram. Slow me down if I ever speak too fast, because there's a lot of information here. So I'm going to move into kind of different realms of like where we see sociality, where we see this kind of psychedelics and relations, increases in relations or group formation. So we start by kind of self, person, and then interpersonal, and then group and culture, and so on. So in the personality, we see that there are different components that change in personality: pro-social, openness, social connectedness. There are many studies actually that show this. This is a meta-analysis that was published this year of 21 papers from the last 2 decades, showing these pro-social or social changes from psychedelics. So some of those papers showed increased altruism, higher social functioning, better relationship with family and friends, and increased empathy. But just always good to remember that most of them in a therapeutic setting, in contexts which are maybe not ideal, this is not necessarily what's going to happen. I'm not saying anything inherent to psychedelics, right? Context is crucial. And this is one example of this. In the same paper, they go back to a study from the 60s in autism spectrum disorder. And they kind of, in that study, they wanted to use these like pro-social or social connectedness aspects of LSD to treat autism spectrum disorder, which was usually good, and usually the improvements was for most of the population. But for some of the patients, actually, there was worsening of the symptoms, including aggressive behavior, including suicidal behavior and suicidal ideation. And then the researcher at the time said that it's related to the interpersonal relations with the experimenters. So the quote here is, "Greater improvements were observed when the therapist was more actively involved with the children, when they were given the possibility to experience meaningful interpersonal psychotherapeutic interactions, and when the setting were free of artificial or experimental restrictions." So if there's no healthy interpersonal interactions, then actually there's a risk in the psychedelic experience. Okay, so it needs a safe, trustworthy kind of relations to guide the experience itself. So studies kind of looking at interpersonal stuff between people speak about increases in love, in the therapeutic alliance, in empathy, in closeness to other, and trust. And also a few studies show increases in oxytocin under LSD. So for example, Dolder from the Swiss group shows that under LSD on a high dose of 200 micrograms, there's increase in openness, increase in trust, increase in I want to be with other people, and feeling close to others. So this is even an experimental study. In the same trial, they looked at empathy, and they showed people photographs of people in emotionally charged situations, and they measured empathy, and they saw that with a high dose of LSD, emotional empathy increased. Okay, so just the reaction to the photos. Now, the therapeutic alliance is also considered the crucial element of any therapy, but also of course, very much important in psychedelic therapy. So this is the report, the connection with the therapist. One interesting study of this in the 60s, the study of Hyde. Hyde did a study in a hospital, and they manipulated a lot of different things that relates to the context. So he did a whole year in which the relationship with the experimenters was very cold and dry and not empathetical, and a lot of requirements from the experiment. And then did another year of the trial in which there's a warmer interpersonal relationship with the patients. And they saw that a supportive environment also had, people had better experiences, but also less risk, less anxiety. So the impersonal environment can create more anxiety with the psychedelic experience. So again, from our New England of Journal Medicine, so from the same trial, we looked at the therapeutic alliance. This is also accepted but not published yet. Roberta Murphy, who is a therapist in this trial, she looked at the therapeutic alliance and how it's related to clinical outcomes. So we see here that the therapeutic alliance, again, the connection with the therapist, and predicts the report. It's pretty much a similar measure, but in different time point. And that predicts the intensity of the experience of emotional breakthrough, of the emotional release in the therapy. And that emotional breakthrough predicts the clinical outcomes, so the reduction in depression. So we see here again, this kind of like in this nice model, how the rapport, the relationship with therapist is crucial for the process of psychedelic therapy. Also, people who had stronger emotional breakthrough also had increased in the therapeutic alliance. So sharing intense experiences together creates a stronger bond with the therapist also. So reduces the depression and also increases the bond with the therapists. This is on the interpersonal realm. Now we move to groups, and this is kind of the maybe classical example of like doing a ritual together in groups, but also there are festivals and raves and group therapy. And generally, recreational use of psychedelics is a lot of times social. There's something that just using them for fun, which can be a good thing as well, is a lot of times social and a lot of times very meaningful for people. So I'd actually think that the pleasurable aspect of psychedelics is crucial for why people want to use them. We can talk about them as being therapeutic and being like, we need to deal with challenges. But in the end, a lot of people use them because they also like the bliss and the pleasurable aspect of them. And in a situation like this is a social aspect. So in group therapy, this is a review of 27 papers from the 50s and 60s of group therapy. And approximately 700 participants went through LSD group therapy. This review doesn't have any concrete conclusions, mainly because there's no concrete measures that were used across studies. They pretty much concluded it was safe, but they do present few studies from the 50s and 60s that say that LSD enhanced the kind of group relationship and allowed the kind of the group dynamics to be an important part of personal therapeutic processes for people. So in the same with the same direction, we looked at the concept called communitas. So communitas is a concept from anthropology, from Victor and Edith Turner. And they studied rituals in Africa and then in different places in the world. And they showed that a lot of times the ritual is in order to reach this kind of group experience. So these are not psychedelic rituals. And what happens that in rituals, the ritual has its own rules, its own social rules. And when a group goes to a ritual together, there is a liminal state in which the regular social dynamics collapse. At least that's what they claim. And then there's a strong experience of togetherness in the group. That's kind of described as moving from social structure to anti-structure. But that anti-structure, this togetherness is experienced as a collective joy, as a togetherness, as a solidarity as well. So they describe that this experience can happen in many liminal situations of in-betweenness. This can be in natural disasters as well. This can be in revolutions, in festivals. So if you maybe think about your own situations like this, usually people describe it as there was magic in the air. So you're in a festival and something else is happening and people relate to each other in a different way. And a lot of time people use there was magic in the air, there was a vibration in the air between us. So that's communitas. So we measured this with questions like, I felt that social status became relevant. I experienced all participants as equal based on a basic shared humanity. I felt a sense of sharing with other participants. I felt a sense of harmony with others. And this is the kind of the data we have from our ceremony studies. So for people that are going into retreats and rituals. And again, we do this kind of process-oriented analysis. So in the middle, we see this kind of increase in comunitas due to mainly psilocybin and ayahuasca. And we see this increase in comunitas that predicts the kind of group and that retreat comunitas and the self-disclosure. So people who had more of this experience were also more willing to talk about their own personal emotions in the integration sessions after. So there was like more, they created more bonding, and that led also for long-term changes in well-being and in social connectedness. You can also note here that report, like in therapy, the relationship with the facilitators was also mediating this experience. Okay, so what's called the container, the trust in the people around you also helps these experiences to happen. Another example of this is the setting questionnaire for for the ayahuasca experience, and they developed this questionnaire with more than 2,000 participants in their trial, so observational trial, and they have those components in the questionnaire: leadership, decoration, infrastructure, comfort, instruction, and social. And then they looked at different ayahuasca settings. So Santo Daime and UDV are ayahuasca churches, and your shamanic setting is Western kind of ayahuasca use. And they show that in all of those settings, those components lead to stronger experiences and also to better outcomes and well-being. So things like the social component and the leadership, but also just the comfort in the ritual allows these certain experiences to happen. So we saw it in therapy, we see it in rituals, and now we move to raves, and we see that as well. So Martha Newson, she actually was in Oxford, a PhD of Harvey Whitehouse in cognitive anthropology, and now she's in Kent. She studied identity fusion, which is very much a concept that Harvey Whitehouse is studying. And they showed that when people take drugs and dance, so usually most of them psychedelics, in a rave or free parties, that can lead to social bonding between fellow ravers. But that social bonding is mediated by the experience of awe. So sharing these like transformative experience on the dance floor with others also leads to this stronger social bonding. They also show that that bonding is also related to pro-social behavior. So they actually measured how much people are willing to give to a rave charity. And they showed that people who had more of a transformative experiences on the dance floor, more social bonding, were also had a pro-social behavior. So donated more to a rave charity, but not to a universal charity. So it was rave specific, okay? So it's like your in-group, right? It's the people that you share the experience with, not necessarily everybody. And moving to festivals, Forstmann looked at 6 large festivals. Again, a very large study published in PNAS. And psychedelic use, again, leads to, again, you see those like process-oriented studies. Psychedelic use leads to transformative experiences, which relates to also social connectedness, and that's also leads to a positive mood in the end. So with that idea and that kind of notion, also this understanding, I also did an observational study of Israelis and Palestinians who drink ayahuasca together. And most of my research actually is directed right now to this study. What I'm showing now is an observational study I already did, but there are a few more studies planned. So we interviewed Israelis and Palestinians who drink ayahuasca together. I also had participatory observation. So I went to those ceremonies. All of them happen in the underground. They're organized by Israelis, and there's a Palestinian minority in them. So I could give a whole talk, obviously, about this, but I'm going to shorten it here. These are not peace rituals. They're just regular ayahuasca rituals for psycho-spiritual intentions. But there's like an underlying kind of intention, collective intention in those rituals for like oneness and human connection and as well peace. But the main reason that people go there is just their own personal kind of psycho-spiritual growth. These are not retreats. So these are regular practice that happens on a regular basis. So communities are formed around them. It can be going to drink together every few months or once a year, but actually the people who participate in this also go to just develop friendships beyond just going to the ritual together. And there's a participatory practice, which means that not only the shaman sings for the participants, but also people that can bring their own music and prayers to the ceremony. Usually like after half of the ceremony, people share their own music and their own prayers. And that allows like also this kind of intercultural and interpersonal flow through music. So we found 3 themes in this study. One is this kind of theme of oneness, and I'll kind of spread those themes during my talk. So for example, this was like an obvious experience that people had. This is kind of the experience of comunitas. For example, there were moments of love and open-heartedness that we're all together. There is no, you are Jewish, Arab, Muslim, Christian. Everything was stripped. All this nonsense was out, and only acceptance and love were present. This is kind of an experience of identity dissolution, human-human connection, social hierarchy collapses, comunitas. This is a beautiful experience for many, and it's maybe an experience that also creates the kind of initial bonds that are required between enemies. But also good to remember that the conflict in Israel is asymmetrical conflict, is Israelis that occupy Palestinians. It's not just about becoming friends. There's also another cultural and political exchange that needs to happen if psychedelics are used for peace, you know, this idea. So I believe that this oneness and communitas are beautiful and important, but they're not the only thing. There are other things that need to happen as well. So in peace research, researcher actually call this, there's a problem, they call it the irony in harmony, which means that actually creating kind of a harmony in small groups in asymmetrical conflict, in conflict in which there is inequality and injustice, actually can prevent change of reality because it creates a kind of illusion of equality. And I have a lot to say about this in different papers, but it's just a tangent here. So moving to culture, a lot of anthropologists, psychedelic anthropologists, speak about the importance of cultural setting and cultural priors, the importance of music and art, and I'll kind of go through this. So, for example, you can see here in the hospital, the quotes start actually the it. It has transformed the entire hospital because the whole atmosphere about LSD. It has transformed the entire hospital. Sorry, I'll pause for a moment. This is a quote by Sanderson in 1959. And this is a conference about psychedelic therapy. If you're interested in the history of psychedelic therapy, it's quite cool to read those transactions because it's just like conversations between psychedelic therapists, and there's a lot of like openness of ideas over there. A lot of them are actually seen also today in the psychedelic renaissance. So LSD has transformed the entire hospital because the whole atmosphere engendered by LSD is spread throughout the hospital and in fact forms an essential part of hospital culture. If LSD is given in a large institutional setting, treatment will be ineffective unless this transformation has occurred. Okay, and we can see also now with psychedelic clinics, a lot of times it's about designing the environment. We don't want them to happen in the regular hospital settings. So the hospital needs to change. There's a change of the hospital culture as well of how to treat patients, how to relate to patients. So psychedelics in other cultures and in ours as well, a lot of times are used also with the idea of like a cultural revitalization. So if you see how peyote was used from 120 years ago until the formation of the Native American Church, we see that there's a lot of cultural elements in the practice. So peyote was a small practice in Central America, but then it starts spreading across North America 120 years ago. A lot of times the idea was to bring people back to the tribe from acculturation or from kind of cultural poverty, from the kind of challenges that happens through colonialism. So there's a very anti-colonial sentiment in peyote, at least in the beginning. But they kind of, what they did is also create a ceremony, a ritual, music, and all of that was part of how they treated also healing for people who were kind of suffered from colonization. So the idea of also the Native American church, if we said that Native Americans suffered a lot from addiction of alcoholism, the Native American church not just treated them themselves with psychedelics, but also with music, with ritual, with community, and all of these were important part of treatment of addiction that was formed by Native Americans. So beside the kind of those cultural elements, a lot of researchers discussed the kind of how cultural priors affect the experience itself. Weston LaBerre, who studied peyote cults already in 1938, was already kind of saw how visions, hallucinations are related to kind of cultural input. Anthony Wallace, who also kind of claimed that we need to look at cultural setting, and actually said that we should do cultural control trial. So like we do placebo control trials, we can do trials in which we manipulate culture elements between conditions. Marlene Dobkin de Rios, studying ayahuasca, also talked about kind of the cultural visions, how they're affected. Obviously, if you go to drink ayahuasca in a Santo Daime church with a lot of Christian background, people will have more visions of Jesus and Maria. But while you go to the scanner in Imperial College, you have more ego dissolution. And you know, there's a lot of cultural priors that happen that lead to certain experiences. So Ido Hartogzon showed that in American trips studying the 50s and 60s, kind of the psychedelic research, and showing that studies that were like psychotomimetic, so kind of trying to study psychosis through LSD, were actually creating psychotic episodes for people, while studies that were psychedelic, they created mystical experiences for people. David Dupuy also studied the same kind of idea, called it the socialization of hallucinations. And in a retreat addiction treatment center called Takiwasi, he showed that the kind of framework, the ontological framework of the retreat is to treat through exorcism. So they see illness as a possession of the spirits, and ayahuasca helps people to exorcise those spirits, and they actually see those spirits come out of their body. So this is another kind of to make this point, which is also was obvious for lots of psychedelic therapists. This is from the same conference Transactions by Hartman. In our group, for instance, which consists of 2 Freudians and 2 Jungians, the latter will get a transcendental experience in the patient much faster than the former. The 2 Freudians, on the other hand, will evoke the patient's childhood memories much more quickly than the 2 Jungians in the group. So we see that a lot of times the framework of the therapy feeds into the patients, and then it feeds back into the therapist, because the therapists see that in the patients. So if a therapist worked with some modality, that modality would lead to certain experiences, and then suddenly the therapist would start believing that, let's say, psychedelics are about bringing childhood memories, all right, and bringing repressed memories. But it's a lot about this intention and the framework in which they're used. Clear? Yeah, any questions? A lot of information. But I hope it all ties to this kind of idea of social component of psychedelic use. With that kind of Israeli-Palestinian context, we actually saw that although people had these experiences of identity dissolution and oneness, there were also experience of connection to the other through music and prayers. A lot of times there was very meaningful, awe-inspiring moments of intercultural connection. So this is an Israeli person saying, almost every retreat, there's a moment in which they, speaking about a small group of Palestinians, are comfortable enough to sing in Arabic. This is always an amazing moment. Suddenly you hear your most hated language by far, maybe the only language in the world that you really didn't like, and suddenly it sends you to light and love. Okay, so the idea here and what people speak about is that the ayahuasca allowed them to kind of relax their own narratives and ethos and identity, and that allowed them more openness to connect to the other culture. So it's known that groups that live in intractable conflict develop this kind of rigid, maybe expectations in the world, if we want to speak about like in the terms of plasticity or and things like this. So we can also think about it neuroscientifically. If you live in a intractable conflict, you develop this kind of rigid expectations of who is the other, what is your narrative, and the plasticity induced by psychedelics can allow the connection to the other in the retreat. So a lot of times people talk about expansion in this moment. They feel expanded. They feel that the group is expanded in the moment. People move from their own personal healing into something unique that happened in the group, which also relates to larger reality. So, but it's kind of good to remember Nicholas Langlitz, who's a psychedelic philosopher, anthropologist, brilliant guy, did some studies in the past about the Zurich lab. So he did an ethnography of scientists. So he studies scientists in his ethnographical work. So the psychedelic scientist. And in a recent paper, he speaks about kind of the idea of psychedelics for moral psychopharmacology. And he just reminds us that while members of the American counterculture used LSD, mescaline, and psilocybin as psychopharmacological tools to liberate individuals from the ills of their society, Huichol youth ingested peyote buttons to become full members of their own society, and Native American church worshippers consumed peyote to foster indigenous resistance to North American colonialism. In the 60s, psychedelics were taken to experience a mystical union that users claimed foster a sense of universal love, but anthropologists have also described Amazonian societies that use them in rituals to prepare for violent intergroup conflict. In all of these examples, actually, we see that LSD, whether it is brings a group together, whether this group is a counterculture group like LSD, or whether this group is trying to form into a society like the Huichol example. But the psychedelic use kind of brings a group together through a shared practice. Now we move into a revolutionary, and there's a revolutionary history with psychedelics. For some reason, LSD is the kind of the obvious one, but peyote and ayahuasca also have some kind of in their history, some of that. A cool example is actually by Hyde, so the same guy who manipulated the context in the hospital. And another manipulation he did was looking at individuals taking LSD just with an experimenter or a group taking together. And he says, and this is not therapy, so he's like a very of experimenters with like a very scientific kind of context. Group subjects tended to be friendly to co-subjects and hostile to the experimenters, whereas the individual subjects seemed positive toward and dependent upon the experimental staff. The group subject seemed to utilize his own co-subject as a substitute source of support. The group tended to become a somewhat deviant subculture within norms of behavior particular to itself and negative attitude towards non-psychotic outsiders. So kind of a very anti-establishment. This is before, you know, the rupture, the psychedelic LSD revolution, but he kind of sees maybe a precursor to that, in a context in which there's a clear hierarchical dynamic with the experimenter and relying on the experimenter for the kind of the safety, then there's a good relationship. But in groups which can support each other and the experimenter just wants to take data from them, they actually create a deviant subculture and oppose the kind of establishment represented by the experimenter. Same kind of anti-colonial history is kind of resistance, revolutionary history can be found in peyote cults. I actually mentioned it before, but peyote has been used in many times with anti-colonial sentiments. Actually, the tipi, that is a very kind of famous symbol of rituals right now. It's a good anecdote is that a lot of rituals, not just the peyote rituals, were outside. So Native American rituals were outside, but then they were banned, not just the peyote rituals, they were banned by their colonizers. And then they started to move the ritual into the tipi in order to hide actually the ritual. So the tipi is almost like in the beginning was a place that's related to their oppression and the need to hide their religious practice. Ayahuasca has also these sentiments of anti-colonial resistant sentiments. If you don't know, ayahuasca was in very few places in the Amazon until 150 years ago. 150 years ago was or at least, less, 120 years ago, was the rubber boom, in which a lot of atrocities happened to indigenous people in the Amazon. So in that very intense political period, this is also when ayahuasca spread between tribes in the Amazon. It was used for healing from colonialism, but it also had this kind of anti-colonial sentiment and kind of creation of indigenous unity through the practice. And LSD also has this kind of hippie, turn on, tune in, drop out, anti-establishment background. So I saw this also in the Israeli-Palestinian project. So again, observational research. And we saw that sometimes there were revelations or moments of insights or visions that relate to collective trauma, mainly the collective trauma of Palestinians. These can be insights of war, of intergenerational war, of the Nakba, the deportation of Palestinians, of just the house arrest of Palestinians, visions of the bleeding land, so very intense politically. Few Palestinian women actually felt the pain of the land. And a lot of times what happened in those visions is that they activate people. So unlike the kind of what we think about the kind of the unitive experience, which is an experience of acceptance, letting go, compassion to all, these experiences, a lot of times they bring this energy of like activism or anger or resistance. And in a way, the action is intertwined with it, the insight is intertwined with action. So they break people from the kind of normal way of perceiving the world. It reveals some injustice, and the action is also kind of almost wants to go out in these moments. So this kind of action intertwined with insight, I have a whole talk about this thing, but basically they kind of almost push people in wanting to change reality around them when insight like this occurred. So if you see things in a new way, or if you see injustice, you also want to change the old way. So a lot of times they even intervene in the ceremony itself, but also they try to, these people who had these revelations try to become like actors or activists, or kind of try to bring change. So this kind of idea of revolutionary revelations is very much, if you think about the archetypical prophet, you know, a prophet is a person who has a vision, but then it's tried to lead a certain movement. So the prophet kind of operates on the kind of stage of history, tries to change society. And psychedelics also have this kind of a prophetic element in them. Okay, so I'll summarize now. And I just want to say that like we need to look at psychedelics from a biopsychosocial perspective. The bio and psychological are obvious for many people, and the kind of the psychedelic neuroscience and psychedelic therapy is a lot about the biological and the psychological. And just a reminder, this talk is just a reminder of the social. It doesn't mean that the psychological and biological are not crucial. It's just like these all things together. And when we think about mental health, we need to think about this whole spectrum of human life. Also, when we think about neuroscientific theories, it's good to incorporate social into the theories. And for example, actually Rebus incorporates that, right? So Rebus, the idea that the expectations of our world, reduces, which gives space for more surprise and more sensory input, more being influenced by the context. So actually through Rebus, the idea of cultural priors, of suggestibility, of changes that depends on context, are also through this kind of explanation. Also, a few examples of plasticity are all actually also incorporating biopsychosocial, and I kind of think that's why plasticity becomes this kind of main thing right now in psychedelic research. There's a convergence into discussing about plasticity, mainly because it gives a good neuroscientific explanation for enhanced influence of context. So for example, from the lab of Rachel Yehuda, who studies trauma. There's this, they kind of describe the therapeutic process. They something similar to critical period plasticity. So if you know with animals, if you know those studies of blocking vision, binocular vision. So if you go to kittens and you block their eyes when they're very young, and then you take those, when they become adults, you take those, the eye masks, and they cannot develop sight anymore. Right? So there was a critical period in which vision is developed. And if that period is blocked, then vision will not develop in their life. So they suggest, Rachel Yehuda, that psychedelics are this kind of plasticity period that allows change, but that change is always context dependent. It depends on what's happened in the therapy, and that allows change. I think that one kind of beautiful example of this plasticity is actually if you see a lot of patients in trials, what they say, and what a lot of times happens to people who go to psychedelic rituals or festivals or therapy is that their musical orientation is changing, right? They start listening to the music that they listened while they listen to other psychedelics. And I think that relates to re-enculturation, okay? Music is our culture. And we get used to listening to certain music, we develop our musical taste, but then we listen to psychedelics under music in a highly plastic state, and we change our musical orientation. And that relates to changes in cultural input, okay? So if we're thinking on enculturation, enculturation is learning. We learn about our social environment. So we can also change these enculturation as we change learning. So human mushroom coevolution. This is kind of a theory that I'll finish with this thing of psychedelic sociality of Michael Winkelmann and Arke. And Winkelmann is like studies of psychedelics from evolutionary perspective for the last 30 years, I think. And they argue this thing. I'm not sure I completely believe that idea, but they suggest that humans and mushroom co-evolved and mushroom helped human evolution. The way they argue that is to show that the effect of psychedelics is very much human, is a very much human thing. So when you think what psychedelics do, they increase our flexibility, creativity, they change process of enculturation, they bring sociality, there's altruistic behavior. A lot of things, the examples they give here, symbolic behavior, so how we are affected by symbols and narratives and stories, meaning, all of these they argue relates to human sociality, and we develop this skill with the help of mushrooms. So it's a concept of a social cognitive niche. I won't get too much into this, but why I think it's interesting is to just think about it as that the 5-HT2A receptor, whether system and receptor, whether it's developed evolutionary through mushrooms or not, plays a crucial role in culture, sociality, human relationships, and so on. So to think about the 5-HT2A receptors is something which kind of crucial for something that's unique about humans. So we know that all drugs are context dependent. You know, it's not just psychedelics. If you drink alcohol in a bar with friends in a party and dance, or you're just with one friend and kind of crying about something that happens, this is already a different way alcohol it becomes context dependent. And every substance is, but it seems that psychedelics are a bit more than any other substance context dependent, especially if you think about visions and how they change through cultural priors. So based on this, I kind of think that the 5-HT2A receptors are very much related to this something that's unique to humans. So I'll finish with, actually I have time for two things, two speculations, okay? One speculation is about let's actually go to this one first. I have time, right? Yeah, 5 minutes. So this is kind of a speculation, kind of continuing based on what I said right now. So it's a kind of a neuroevolutionary, neuroanthropological speculation about the 5-HT2A system. We see that the distribution of the 5-HT2A receptor I promised some neuroscience, so I have to do those slides. Now we see that the 5-HT2 receptor is in high-level regions, in like kind of the highest parts of the hierarchy, and frontoparietal regions, DMN, kind of transmodal regions. But we also see that there's high density in the primary visual cortex. Okay, you see this here in the back of the brain. So not in all primary sensory modalities, but high density in the primary visual cortex. We also know that psychedelics are very visual. We know that they reduce alpha power frequency. So usually alpha power frequency in EEG is something that's reliably reduced whenever people open their eyes. So moving from eyes closed to eyes open reduces the alpha power. Also psychedelics, if you're with eyes closed and you take LSD, DMT, there's a reduction in alpha power. This suggests that there's activation in the visual cortex. So here, and it's the most replicated results in psychedelic research. Like undeniably, there is a reduction in alpha power in dozens of studies from the 50s and 60s, and also the kind of modern studies. For example, we see in a DMT injection, we see in the Chris Timmerman study, this is the intensity of DMT, subjective intensity, and we see here this band of alpha power reduces immediately in the beginning because it's an immediate reaction. But once the response goes down, we see also that the alpha power kind of comes back. Okay, suggesting that there's activity in the visual cortex. Also here with bold studies, we see activation under ayahuasca. We see activation in the visual cortex with bold, measuring with bold. So kind of looking at eyes closed, eyes open in a block design, looking at the activity in the visual cortex and showing that people are with eyes closed, there is still activity in the visual cortex, while it's usually when we close our eyes, it drops the activity. With ayahuasca, when we close our eyes, the activity stays in the primary visual cortex. Now, so this is activation. Now we look at connectivity. So we see that, and this is kind of what I'm going to suggest now, that a lot of the kind of meaning enhancing things behind psychedelics are related to vision, and they're sensory. They are sensory. So, for example, the idea of like stories become visions, their cultural priors become visions, or our connection to our surrounding a lot of times has a visual component into it. Okay, things are enhanced, things are moving, things are alive, but there's a visual component to it. And I argue that this is because that 5-HT2 system is related to both high-level regions in the kind of transmodal regions, but also to the primary visual cortex. And actually, if you look at it a lot in neuroscientific studies, we see connectivity increases between the primary visual cortex. So here, this is a between network analysis, and we look at co-fluctuation of regions, so how they operate together. And visual cortex, primary visual cortex, operate with many higher level vision regions. And we see this also in our PNAS paper. So connectivity in the primary visual cortex becomes more connected to many other regions. And also other studies from Preller lab in Switzerland, from Zurich, and the Liechti lab, so Muller in Basel, and the lab in Amsterdam, sorry, not in Amsterdam, the Netherlands. They also showed this connectivity between primary visual regions and higher level regions, frontoparietal or DMN. Enzo also showed it with our data. So what I argue here is that these regions, which have high 5-HT2A receptors, they operate together, and they bring stories into visions. They create in a sensory way, and that's enhanced the meaning of stories, and that brings a certain way of being in the world, an altered state that has a function for humans through rituals, through religiosity, through enhancing meaning in different, through different practices, and probably serve the function maybe in our evolutionary advantage for culture and acculturation and sociality. So this is a suggestion and theory I'm developing. It might be flawed. Another speculation is regarding psychedelic therapy. So just kind of thinking about how psychedelic therapy would be in the future, hopefully how it will be in the future, is hopefully a hybrid between a clinic and a community center. This is, first of all, cost effective. Working with groups is cost effective, obviously. Hopefully this will be local, so not a retreat that people go to, connect to some strangers and come back, but also something that creates communities in with your neighbors. It helps in peer support, so there's less neglect. One of the problems of psychedelic trials is once that they build a kind of a strong connection with the therapist, the trial ends, and some people feel neglect after the trial. So if a community is kind of created around the practice, a therapeutic communal practice, then there's no need for necessarily for the relationship with the therapist as the thing that grounds you in the integration. So people can just help each other integrate. It can be diverse. It can be people from different, with different issues, different problems. It doesn't have to be therapeutic. It can be just playing music together and enjoying each other. So people with different skills can contribute to each other. It doesn't have to be everybody are depressed coming together. Hopefully participatory, so means that people engage in this. They don't just receive things from therapists, but they also engage. And knowledge moves through apprenticeship. So knowledge moves from the facilitators into the people that go, the patients, let's say, but then the patient can become a musician or can become a facilitator later on once they're more involved in the practice. So this is obviously how if you know the kind of how the underground practices operate, they operate pretty much in this way. And I think this is the organic way in which psychedelics work. Right now, there's a challenge of institutionalizing them, but hopefully this organic way of how to work with psychedelics would also happen in the institutions. Thank you very much to all. Maybe I thank Robin Karat-Harris for supervision in 9 years and the whole Center for Psychedelic Research, Michael Wilkelman, Catherine Peller, and Evgenia Fotiu for psychedelic sociality research topic we edited together in Frontiers, and for Natalie Ginsburg, Dublin, Antoine Saka, and Nadim Karkabi for the peace research. Thank you.
Alrighty, we're now going to start the last lightning session of the symposium, the ethics of psychedelic medicine. We're going to be starting with Julia Bornemann, who's going to be talking about shaping the future of psychedelic research with public and patient involvement and engagement.
Hi, everyone. My name is Julia. I'm a PhD student at the Center at Imperial, and I'm going to tell you a little bit about why I think using the patient voice in psychedelic trials has not only helped us, but I think is an important part of all subsequent trial designs. So first, I'm going to define patient involvement. So the term I'll be using is PPI. It stands for public and patient involvement. And basically what that means is that the focus is shifting from doing research on people and instead doing it with people. So it's moving towards a space of equality and integration and kind of moving away from objectification of patients. So this can include anyone with lived experience. So for example, it can include patients themselves or their carers, or also lay people, community members, or PPI experts. And it informs all aspects of the research cycle. So it can be important whether it's part of conceptualizing a study or undertaking the research itself or disseminating it later on. There are a few reasons that I think it's important to do PPI. It produces efficient and cost-effective and relevant research outputs, and it creates these nuanced treatments, which I think prioritize accessibility and patient empowerment. And this attention to detail, I think, nurtures a trusting relationship, and particularly this is relevant in healthcare. And then the overall outcome is that patients tend to fare better, and they're more likely to actually comply with recommendations. And this is particularly interesting in chronic conditions like the ones that we've been discussing all day today, such as depression, but also what I'm going to be giving you an example on, which is chronic pain. there's many types of PPI, so the choice will largely depend on the specific intention or purpose for your trial, but general impressions can be obtained from patient groups online, for example, on Reddit, to more specific questions which might be asked in a workshop setting. And then the most direct method is implementing a research oversight panel. And if you're interacting closely with patients or public collaborators, they can help you with very specific actions. So for example, in my trial, which I'll be telling you about shortly, we have a research oversight panel of people with lived experience who co-produced all of our patient-facing documents, so the patient information sheets, to make sure that it's readable and useful for people. And they also can provide kind of practical feedback. So for example, things that you wouldn't think about yourself if you haven't had that lived experience. So we are putting our patients with chronic pain into an MRI scanner. I would have never thought that, you know, you'd have to put pillows around them to make that comfortable. So little things like that are also really important. So I'm going to give you an example of what PPI looks like in action. And the example is for my trial, Silopain. And this study investigates psilocybin in a chronic pain population, specifically in fibromyalgia. Fibromyalgia produces widespread pain with serious impairments in function, but it also has significant mental health comorbidities, with about 60% of fibromyalgia patients also suffering from depression. The aim of our study is to learn about the central mechanisms of psilocybin therapy, so in the brain and psychologically. And we're going to be using a mix of neurophysiological data through EEG and fMRI. And we're also going to be including psychological, behavioral, and qualitative paradigms as well. But when we were thinking about actually designing this trial, we encountered some challenges because the background research of this specific subset of psychedelic research is incomplete, and there isn't a whole lot to go on. So I was thinking, but at the end, I thought it was important to not try to reinvent the wheel. So there's already a huge amount of information out there of people who have been self-medicating, people who have chronic pain who have been self-medicating with these substances, who have been posting their trip reports online and sharing this information. So I thought, why don't we harness this knowledge of people with lived experience, and perhaps this could help influence our trial. So we conducted a series of interviews or in-depth discussions with the aim to find out what is important for people who self-medicate in their outcomes. What do they find is actually helpful? And perhaps how could we use this information to improve our design and our protocol? We did 11 one-hour discussions with people who self-medicate with psychedelics, who have chronic pain. And the interviews were transcribed and thematically analyzed for major themes. And so here you can see some of the themes on the like little wheel picture. This isn't all of them, but it's kind of a condensed idea of them. There's a couple of very classic psychedelic themes here, like trusting and being open, and including tenets of mindfulness and breath work as important things that might facilitate a positive experience. But then I think there are also interesting, more pain-specific things that came up as well, such as including movements like yoga or qigong in the sessions themselves. So our general understanding of what psychedelic therapy looks like is that people tend to lie down, close their eyes, put on their headphones, and not move for 6 hours. And this is obviously not really going to work in a pain population because they'll be extremely uncomfortable. So that was really helpful. And then also finding ways to express physical and emotional manifestations of their pain through journaling or art. But one thing that I was particularly interested in was the fact that several participants highlighted the extreme physical intensity of their experience. And this is something that I hadn't encountered so much before. And this really kind of struck a chord with me. So I'll dive into that and show you how that influenced the trial a little bit. So here's a quote from the interview. Basically, one of our participants said, "I felt this intense welling of energy starting from the core of my body, and it started to overtake me. I started crying, breaking down, letting it all go, howling in agony. All of the suffering, I was just letting it all out. It was the biggest release of my life. I got to let go of so much grief, fear, and even some anger. I felt really healed. My entire body was tingling and I felt so light. I felt so relaxed and completely pain-free. So this is a kind of an archetypal example of somatic release or somatic processing or somatic discharge. There are a couple of names for it. And this is this idea that is written about by several important people in the therapy field, such as Peter Levine, Gabor Mate, and Bessel van der Kolk. And basically the idea is that stress or trauma is stored in the body and can produce unease or dis-ease through a dysregulated nervous system, and that this can result in physical manifestations such as chronic pain. And releasing this stress or trauma can be violent and extremely cathartic. And this is, I think it's a really interesting concept, but it's still largely unexplored in rigorous research. So for example, there isn't one even name for it. There isn't a clear definition. And this frustrated me a little bit, but spurred my curiosity on even more, especially seeing as several of the participants found this to be really important in their experience. And so this quote raises questions of presence in the body, the mind-body connection, embodiment, and specifically also about somatic processing. And it inspired some new investigation or investigative directions for the trial. So we added some new measures to explore this question. So first we added the heart rate discrimination task, which is a behavioral task to look at interoception or how people inhabit their bodies. Basically, this task has two sort of trials. In the first trial, you compare an internal stimulus versus an external stimulus. So this is your heart rate or heartbeats versus tones, and you have to decide or report which is faster. And in the second trial, there are two external stimuli, two sounds, and you have to say which is faster. And then those are compared, and you can use that to basically see the amount of, or the fidelity with which you can interpret your own internal goings on, if you will. We're also looking at embodiment through some self-developed items that are used in some other trials as well. And we're also using a pain-specific Rebus model, which was mentioned a few times so far today. We're calling it the RebQP. I forgot to update it here. Anyway, this looks at people's beliefs and their relationship to their body and their pain, and hopefully we can see that changing throughout the trial. Finally, I also, with the help of my friends and colleagues, created the Somatic Processing Questionnaire, creatively titled, investigating somatic processing by hopefully trying to catalog physical symptoms that people report and whether there are any links to meaningful experiences, to emotions, and if that in turn causes any relief or release or catharsis. So we want to see, does this phenomenon happen? If so, what happens? And can this contribute to some sort of a definition that, we can use for future research? And then if we zoom out, we can also look at how PPI influenced our trial more broadly in terms of therapeutic protocol development. So it helped us directly identify topics to address and developed values and therapeutic priorities. So we focused on 4 major points. We don't have to zoom in on all of these. I'll zoom in on one later. But basically the 4 points are experiential avoidance, alliance and trust, embodiment and somatic process, and finally creating a lasting positive change. And all topics are approached with the spirit of participant empowerment and partnership. And these are really core aspects and values of our therapeutic relationship. So I'll zoom in on the first one. So you can see this is just an example, but this is the process we went through for all of them. So first we had to define our problem. We know that desperation leads people to avoid their physical and emotional experience and disconnect with themselves and their bodies. And this creates an unhealthy relationship with themselves and their pain, and also just their general relationship to their condition. So the PPI that we did directly inspired this. So you can see the quotes all around it. Specifically, people speak about, finding acceptance of their situation, and that this really helped them realize that their pain was a little bit more manageable, and that they didn't have to perhaps suffer as much. And so this made us think about some insights. What is the opposite? What do we want to achieve? What is the opposite of experiential avoidance? So we were thinking of openness and willingness, acceptance of their thoughts, feelings, emotions, and sensations. We were thinking about the need for willingness and self-motivation as an essential part of achieving meaningful change. We were thinking that we already know from previous trials that avoidance is reduced following psilocybin exposure. And this all culminated in us deciding on one of our primary outcome measures. We have two, but this is one of them, which is the brief experiential avoidance questionnaire. So this is a major aspect of our trial now. And then this therapy generated a therapeutic intention, because we want to move, or we want to help people move towards acceptance and openness of the full experience, not just the happy parts, but all of it. And it's obviously extremely relevant in pain, and especially prioritizes intrinsic motivation. So just a quick summary. I hope you can see how including the patient voice allows us to ensure that our research stays relevant and accessible to the end users. Embedding patient perspectives empowers people and strengthens the trust in the system, and improves the efficacy of the interventions that we're trying to develop here. PPI can adapt to serve various research contexts and environments, and it can be as large or as small as you would like or need it to be. PPI has informed SiloPain throughout, and has strongly influenced our therapeutic protocols, and even inspired a new research direction. Beyond this, we've gotten useful input on nuanced aspects, practical aspects, such as how do we deal with the MRI. So hopefully you can see that PPI is a really important, and I would argue essential aspect to include in the design of future trials, and how we can prioritize such engagement, and this will contribute to a more equal and representative psychedelic future. Thank you. Oh, one thing, we're recruiting. If you know anyone who has fibromyalgia, please come my way.
Okay, our final talk of the day is Ibogaine and Reciprocity by Kiran Ahmad.
And it's just going through with the... Okay. Hi, everyone. Everyone's still in the room, last 15 minutes of the day. Okay, woo! Okay, so I'll give you a little bit about me. So my name's Kiran Ahmad. I'm an NHS doctor by background. I'm specialising in anaesthesia. So some of the talks earlier mentioning propofol, which I use in my day job, and ketamine, which I use as my day job here and in Africa, because I do some work with Médecins Sans Frontières as well. But I joined Imperial about a year ago now, working on a number of different projects. So I'm working on the psilocybin and chronic pain trial with Julia, and a ketamine trial in healthy volunteers looking at PET imaging. But during my time at Imperial, I found myself moving towards the ethical aspect of psychedelics. So last year, I joined Drug Science's ethics working group, where we're focusing on this concept of reciprocity. So reciprocity means the exchanging of things for mutual benefit. And I'm looking at that in relation to Ibogaine. So for the purpose of the talk, I'll be explaining what ibogaine and iboga are, an overview of the Western perspective, just with an outline of what's happened in history to current date, the impact that this has had on the indigenous perspective, what the Nagoya Protocol is, which is a legal framework that exists around reciprocity, and Blessings of the Forest, which is a community interest company that I've recently joined for ibogaine and iboga advocacy work. So the map that you can see highlights Gabon, which is a country in West Central Africa. And this has been the main source of ibogaine for the last 100 years for the West. So what is iboga and ibogaine? Because, you know, sometimes they use interchangeably. So this is a picture of the Tabernanthe iboga shrub. This mainly is found in the rainforests of Gabon in Africa. And it's quite distinctive. It's about normally 1.5 meters in height, can grow to a 5-meter tree, has these distinct fruits on them that animals in the forest eat, and then they propagate the seeds, allowing the trees to regrow. Now, if you focus on the root of the iboga shrub, that is where ibogaine is isolated from. So there are a number of alkaloids within the root, around about 13, 14. And ibogaine is a principal alkaloid. So that can be extracted in a number of ways. It can be a total alkaloid extraction where everything's taken. It can be isolated as ibogaine hydrochloride, which is the most common way that we use it in the West, or it can be used as iboga, the shrub, so the actual root itself, either eaten directly, this is how the indigenous people of Gabon use it, or as shavings or ground into powder or in decoctions in teas and so forth. So this is just a picture of some of the indigenous people in Gabon, and they use iboga in their traditions. It's been a center part of their traditions for thousands of years. We think close to 100,000 years. The Pygmy tribe, which are one of the oldest heritages in the world, who still live in the forest, say that it's been with them since the beginning of time. And it's used in their religion, the main religion in Gabon of Bwiti, and it's used as a sacrament and also in initiations. But safe to say, it's very central to their entire way of life in Gabon, iboga. So this is just an overview of what's been happening in the West. So in 1819, it was the first Western reference of iboga, where a British explorer noted in his documentation the natives eating a plant and taking long walks at night because they were able to stay awake for a long time. Then in the 1900s, ibogaine was isolated from the actual root, and it started to be developed in France, and it was used as a muscle stimulant, so treated for influenza and sleeping sickness. And then in the 30s, it was marketed as lambarine. And then it was the 60s where the anti-addictive properties were first noted by an American scientist. So it's the addiction properties that is what the West are focusing on currently. And it was an American scientist, Lotsoff, who basically, he was a long-term heroin user himself. And he ingested some ibogaine and noted a decrease in his cravings and a lack of withdrawal symptoms. So he then went on to patent it in the 80s. And then in the early 90s, there was FDA approval for the first study in humans, but it got stopped after the first patient because of various issues with patent disagreements and concerns around safety, etc. But at that time, the anti-addictive properties had been noted. And so there were, even though it's a Schedule I drug in the UK and the US, a number of legal clinics exist around the world. So in places like Mexico, Brazil, Thailand. And there's a lot of underground work being done with ibogaine for substance use disorder, so mainly with opiates. And most recently, in 2021, a phase 1/2A trial has begun in Manchester by Deborah Mash and her team, looking at opioid use disorder. Now, the exact mechanism of ibogaine, we know it works on multiple transmitter systems. Preclinical studies have shown that ibogaine, which is one of the main metabolites of ibogaine, works at the kappa opioid receptor, thereby blocking the endogenous dynorphin, reducing that, which is one of the things that contribute to withdrawal symptoms. So that's about as much I'm going to mention on that perspective, because I want to go into the indigenous perspective now. So 85%. That is the number of trees, the percentage of trees, iboga trees, that have disappeared from the rainforest in the last 50 years. Just take a sip of water. And we know that that is mainly due to the international demand in the West. So what that means is it's driven up iboga poaching in this region, because then they can sell this on the black market for up to 4,000 times the market value. The commonest route for this is Cameroon, which actually borders the north of Gabon. So traffickers come into Gabon and go to the heart of the forests and national parks, and they plunder the resources. Normally, the traditional people, if they want to use iboga, they would simply go into the nearby rainforest and they would just remove roots from the tree in a sustainable manner and let the tree grow. But that's not what the traffickers do. They plunder the resources. And then they sell it to the highest bidders on social networks. And we think, and again, we've worked with NGOs in Africa who monitor like trafficking arrests, that we estimate that up to 90% of ibogaine in the West has come from a poached source, but it doesn't necessarily, it's not posed as that, it's posed as ethically sourced through various routes. And these figures, by the way, are taken from the Ministry of Forest and Waters from Gabon. Okay, so sadly, with iboga poaching comes elephant poaching, or elephant killing for the ivory. This is because elephants eat the fruits on iboga trees. So what the traffickers do is that they follow the roots, the track marks of the elephants to find the iboga resource, plunder the resources, and then they kill the elephant for the ivory as well. So we're seeing a massive reduction in the elephants in this region, having a huge consequence on the natural ecosystems in the rainforests. So as a result of this, in 2011, the First Lady of Gabon, she recognized what was going on. There was a huge, not an outcrop, in the public domain, noticing that the deforestation, the effects that this was having on the indigenous people. So she commissioned the writing of a report to understand what was going on in the scientific world in the West and how they were using ibogaine, because this wasn't really being fed back to Gabon. So this report was led by this gentleman, so Jan Guinon. So he's one of the leading names in iboga sustainability. He's been working with the indigenous people in Gabon since 2006 and the authorities. He's an international sustainability consultant, and he was given a mandate by the Gabonese Ministry of Culture to defend the rights of the people. He was supported by Professor Gasita. He is an 85-year-old pharmacist, psychopharmacologist, and a world authority on iboga. He has said for decades the importance of using traditional medicines to look after them as a way of working themselves out of poverty, and the issues of sustainability around the iboga resource, noticing that it was becoming diminished. So the report that was written was shared with the authorities in Gabon, who were absolutely shocked with the lack of supervision of the sector and what was going on. And as a result of that, the Nagoya Protocol came into action. So the Nagoya Protocol is from the UN Convention on Biological Diversity. And it's an international framework, a legal framework that exists, which is about access and benefit sharing of genetic resources that arise from a country, and any profit that's made that's shared in a fair way. So Gabon has been the first country in the world to sign up to the Nagoya Protocol with a psychoactive substance, that being iboga. So what that means is if iboga, it leaves the country and is being profited by someone internationally as ibogaine, then they have to give back or benefit share with the people, with the indigenous people. Now, step one is signing the protocol, and then step two is actually getting it implemented. And that's what's happening at the moment. We're waiting for the Gabonese government to implement the protocol, but these things take time, they take money, you need legal framework, you need Nagoya teams, but that's all starting to happen now. In response to the Nagoya Protocol being signed, it was recognized that projects and things that were happening in Gabon needed to be aligned to the Nagoya Protocol, so i.e. ethically sourced iboga, not trafficked iboga, and sharing of benefits. So in response, Blessings of the Forest was created. So this is a community interest company. It's not for profit, and they have an NGO sector in Gabon. And basically they act as intermediaries. They have several roles. They act as intermediaries between the indigenous people of Gabon and the international communities. And between the founders, so Professor Gasita and Jan, they have over 50 years of experience really understanding what's happening on the ground with the indigenous people. So one of the main projects that they're focusing on is creating community iboga farms. So encouraging villagers to plant new iboga shrubs so that they could be grown in an ethical way, and then supply chains can be created for it to be sold to international markets. And in between, it needs to be converted to ibogaine. And again, all of that requires infrastructure and frameworks to be set up, which is what Blessings of the Forest are helping with. And like I said, they act as intermediaries. So from Gabon is a French-speaking country, and to engage with international communities, again, it requires connection, it requires a bridge between them. And reciprocity. So I recently joined them in Blessed the Forest a few months ago as the medical reciprocity manager. And my role there is to engage with international communities who are either developing ibogaine or who are working with ibogaine, to encourage them to give a percentage of their profits back to the indigenous people, so that they can then, Blessings of the Forest can then support them to build these community iboga farms. The villagers can then directly sell their iboga as ibogaine eventually to the international communities. Then those villagers can generate their own income and then become self-sustained. That's the idea. So, as it was International Women's Week, Women's Day this week, I've put up, this is Mama Mazembe. She is a respected elder in the Gabonese community as in one of the ethnicities. And she obviously supports Blessings of the Forest. She's wearing a t-shirt. But there are 53 different ethnicities that exist in Gabon. And Blessings of the Forest have support from over 30 of those 53 ethnicities through their leaders. They're known as Negangas, the equivalent of a shaman. And that number is growing. But it obviously takes time to build the connection and relationships and trust with the local people in order for Blessings of the Forest to support the communities. And this is just an example of some of the iboga farms. They're planted right next to the rainforest. And on the left is young iboga bushes growing. And on the right are some that are ready to harvest. So just to know, it takes 5 to 7 years for an iboga shrub to grow so that the roots can be harvested for ibogaine. So again, Blessings of the Forest play a supporting role at the moment until those trees can grow and be sold to the international communities. And that's what we're helping with. So I'd just like to finish by saying, yes, ibogaine is being developed and there's really promising results for addiction. But at the same time, for us to remember the indigenous perspective and to hold gratitude for the indigenous people who have actually stewarded and looked after this medicine and the knowledge for thousands of years. So now that it can be shared with the West for these treatments. And now it's our turn in the West to be able to support these people, the indigenous people, to use their own medicine as a way of working themselves out of poverty. And that is what reciprocity is. Thank you. I was just going to say there's no time for questions, but if you want to contact me for anything, my email is up there, so you can feel free to get in touch.
Or if you guys want to hang around for a bit to chat, you're also more than welcome to. That brings an end to the symposium. Thank you so much, everybody, for coming. I'd like to thank the 14 extraordinary speakers that we had today. You guys all had really, really impressive presentations, and I thought you all brought your A game. This, I believe, is the first ever symposium on psychedelic science organized at Oxford. I hope this will just be the beginning of more events at Oxford to foster dialogue about this incredibly important topic. This was a joint event co-organized by the Oxford Psychedelic Society and by the Cortex Club, Oxford's Neuroscience Society, and it couldn't have been possible without everybody on the organizing team, Antara, Aaron Lafer, Iris May Morse, Tolu Duckworth Silfi, who else am I forgetting? CM Wilcox, Sam Libana Garcia, and there's definitely somebody I'm forgetting, but and Jess Myatt, yeah. Antara, did you want to say anything? All right, cool. Thank you so much, everybody. Yes.